having the connection with the Noida. A very good morning to all respected dignitaries, eminent speakers, esteemed delegates, researchers, academicians, and dear participants. It is a great pleasure to welcome you all to this technical session 5 of our 3 days international conference, which brings together global experts to deliberate on cutting edge advancement at the interface of science, sustainability, and healthcare innovation. The theme of today's invited lecture addresses one of the most transformative directions in modern pharmaceutical sciences, precision therapeutics driven by sustainable and biomimetic materials. As healthcare continues to move towards personalization, there is a growing need for drug delivery systems that are not only effective and patient-centric, but also environmentally responsible and clinically translatable. In this context, the topic of today's lecture is precision therapeutics using sustainable green materials like albumin for personalized and biomimetic therapy. We are honored to have with us Dr. Ankit Rochani, an accomplished academician and researcher who will be delivering this insightful lecture. Dr. Ankit Rochani is currently serving as an assistant professor at the Wegmans College of Pharmacy, Rochester, USA. He earned his PhD in bio-nanotechnology from Toyo University, Japan, and subsequently completed his postdoctoral research training at both Toyo University and Thomas Jefferson University, Philadelphia, USA. As a dedicated faculty member, Dr. Rochani is deeply committed to training the next generation of pharmaceutical professionals. He actively mentors PharmD, PhD, master students, integrating hands-on research, critical thinking, and real-world problem solving into his teaching philosophy. His academic approach seamlessly blends translational pharmaceutics with a sustainable biomaterial research, ensuring that students are well prepared for careers in industry, academia, and clinical practice. Dr. Rochari's research program is focused on formulation science, analytical method development, advanced drug delivery technologies. His work emphasizes the development of innovative dosage form using sustainable green materials, including albumin-based nanoparticles, hydrogels, films, and related biomimetic systems aimed at advancing personalized therapeutics. In addition to his formulation research, Dr. Rochani's lab plays an important role in supporting hospital pharmacy practices by evaluating the stability of compound preparations and establishing beyond use dates for both sterile and non-sterile compounded products. This work significantly enhances clinical reliability, regulatory compliance, and patient safety. Furthermore, he contributes extensively to bioanalytical characterization of biosimilars, developing robust assay and analytical methodologies that support product quality assessment, biosimilarity evaluation, and regulatory readiness in an efficient and scientifically rigorous manner. Without further delay, I warmly invite Dr. Ankit Rochani to deliver his expert lecture. Welcome, sir.
Thank you, Dr. Tiwari. Thank you for this introduction. Hi, everyone. I am here, and this is a wonderful opportunity to interact with so many faculty members who are seasoned faculty in their esteemed institutions, and also to connect with the young generation, the young blood, the budding scientists who may be interested to pursue some of these areas that I'm going to talk about in next few minutes or maybe in an hour. I would request people to switch on their cameras if possible, the students, so that like we can have a healthy discussion. I assure you that I will not bore you with my discussion on rheologies or the classical pharmaceutical sciences that you actually would have seen in your textbooks, but I will more talk about like the ideas of exactly how it can be used to help patients to make better therapies and maybe make a better life for patients. And that's what the dream has been for me when I did my pharmacy at MS Ramaya College in Bangalore. and then coming over here and working and supporting all the clinicians so that patients can actually get benefited with some of my work that I do. That gives me a sound like satisfaction. So if someone is keeping their cameras on, that's that would be good so that like we can I can see you and we can discuss more about what I'm going to talk about. So my topic for today is about precision therapeutics using sustainable green materials like albumin. So much of my talk is going to be centered about like how this albumin protein has shaped the world of pharmaceutical sciences and how it is being used in clinics as well as in modern formulation development, and what are its how is it beneficial to us as an academician, and how can we use it to further our career, also to help patients so that it can be useful to them, because ultimately the goal is for them to use it. And the material has to be sustainable, it has to be simple and easy to use and apply. When I say precision, I'll just mark some of the points over here. When I say precision, one second, am I able to connect my pen? I think so. Yep. When I say precision, the precision meaning is that like the system has to be precise about um, its area where this particular formulation is being applied to. Many times, what happens is that, like, we develop nanoparticles and delivery systems, like most nanoparticles, solution dosage forms, oral dosage forms. You take it by oral route or give it by IV infusion or by other different routes like intranasal, intravascular, intraocular, or at any other areas. But the conditions like cancers, which are they're chronic in nature, you expect the blood vessels the drug to undergo an ADME, like absorption, distribution, and metabolism, and hope that the drug would actually reach to the site where it has to be giving out a pharmacological response. To avoid that, we thought that maybe it would be a good idea to have some kind of an injectable gel or maybe some type of formulation that can be applied directly at the site of a tumor or something when the patient is undergoing a surgical resection. Thinking from that perception, these gel-like systems actually gives us a precise location where it can be utilized. It also provides us a personalized way of developing different types of formulations. So you can use a single material and make different types of dosage forms. And I'm also going to put some light on exactly what do you mean by biomimetic. That's the whole idea. Like I'm mostly going to talk about different sustainable materials and mostly focusing on albumin for getting a personalized or biomimetic therapy. So the albumin, one second, there's some problem. I'm not able to connect. Yeah. So albumin is basically the protein that you're seeing rotating on your screen. It's a heart-like structure that is abundant in the human body. And our goal was to create like something like this. The next video that you're seeing is the gel, a transparent gel that is injectable in nature and it can be applied at a localized site. And maybe this particular matrix can be used as a delivery system for drug delivery. So that's the basic theme of my whole lecture that I'm going to talk about in next 1 hour. Before I start with the discussion, I just wanted to give you an idea where do I live. I mean, like I'm in United States. Everyone knows Dr. Tiwari actually gave a wonderful introduction of me. I live in Rochester, New York. I work at Wegman School of Pharmacy. Um, to give you a perspective, if you see the screen that I have shown, I am very near to Niagara Falls, which is like roughly 1.5 hours from my home. So, guys, if you are interested to come in over here, you can drop down and stay at my place, and probably I can drive you to Niagara Falls. That's how I've been selling this place to most people who are coming from India and visiting me over here in the United States. And I'm also nearby to uh, New York JFK, which is like 5 hours drive. So Times Square. Technically speaking, the reason the mostly I like about this place is because I am living in the area where we have Rochester Institute of Technology, which is the motherland of for the Xerox machine. If you know, that was actually invented in RIT over here. So I have some collaborators over there. I'm working with them. It's a place where the Kodak, the camera that we used to see. I do not know most young kids. I do not know if you know about Kodak, but like in our days, Kodak used to be a big name when we had to have those cameras. And it's a place of Finger Lakes where we have abundant growing grapes and wines that are most produced in this region. It's a cold place, so it has like a temperature of -7 to 2 degrees C. So just thought to give you some idea where do I live in, and if someone is interested, I'm looking at chats. If you have any questions, just feel free to drop in a chat, and I'll be happy to answer the questions regarding the place where I live and what we can do over here. So there are a few couple of learning objectives that I have over here. That is like I'm going to talk about what are sustainable green material. I'm going to talk about the biochemical nature of albumin. I'm sorry about changing the slide. I'm going to talk about like exactly how the biochemical nature of the albumin is going to be helpful in making different types of dosage forms. I will be talking about like the role of albumin in aging and age-related diseases. Its role as a xenobiotic for a natural nano carrier. I'll be talking about a clinical use of albumin solution as drug for treatment of chronic hypovolemic conditions. Then I'll be talking about my research on albumin nanoparticles, gels, 3D films, and 3D bioprinted structures that we are trying to develop in our lab over here. Feel free to drop any questions if you may have, or if you want me to slow down, I can do that too. But I wanted you all to understand exactly what it means to play with the green materials like albumin. Going to the next slide, sustainable green biomaterials for drug delivery. There are so many of them. Like usually they're classified into three parts. That is either they are polysaccharides, they can be protein, lipids, and also these are the three main categories in which most of our pharmaceutical excipients, which are used as which we use as like pharmaceutical excipients, are classified as a green material. In case of polysaccharides, we usually use chitosans, carrageenan, alginates, hyaluronic acids. These are some of the polysaccharides. I have written carrageenan over here because one of my project was on the development of carrageenan-based gels, which were used for the development of a non-drug containing contraception for females. This was funded by Gates Foundation. It was one of the contract work. So if someone wants to have more details, we can talk about it in the later phase because I want to focus more on the protein-based system that is albumin, which is the material that I'm using for making gels. So to understand, like if these materials are to be sustainable, they need to have three characteristics. Number one, they should be produced naturally and it should be structurally conserved in all the living organisms. Um, Second is they should be biocompatible and they should be non-toxic in nature. And third, it should be acceptable and it should have been standardized as per United States Pharmacopoeia, Indian Pharmacopoeia, or any of those regulatory bodies so that we can use it for developing different types of dosage forms. In this talk, again, I'm going to circle back to our discussion on the albumin. If you look at the table that I've shown over here, it is from one of the cover story that we published in AAPS News magazine. I have listed four different types of materials that are which are used commonly for drug delivery application. The first one is albumin, which is a protein. The next one is collagen, which is again a protein. Hyaluronic acid is an example of polysaccharide, and I've also shown a very niche material that is DNA, which is an oligonucleotide. And this material is it's used in the research works in developing different type of nanoparticles, but so far we do not have any formulation that is which are approved by the FDA, which shows that you can use DNA as a pharmaceutical excipient. But I want to circle back to our discussion on the albumin. If you notice, it is 180 grams that is present in an adult human. We can have a source of bovine or uh, have a lecture is going to be okay. So the source of this albumin protein is usually bovine or human. Mostly, we use bovine serum albumin as a source for creating model formulations, and if then they are characterized, and if these characterizations in the animal models are working well, then you actually use human serum albumin for making your formulations. That's how the traditional trend looks like. But even if like you're looking for human or bovine sources, the structurally these two proteins are homologous to each other. And if you overlay the three-dimensional structure of both the proteins, there is nearly 82% homology in the three-dimensional space, meaning the structure of albumin that is coming from human versus that of animal, that is bovine source, they are so similar to each other that like if you're making a formulation using the bovine source can be easily be transferable or translatable to make a human albumin source formulation. So we have discussed about like its abundance and like the nature already creates it for us, and we are just going to extract it from the bovine source or the human source and use it for our benefits. An adult human has like roughly 4.5 liters of blood. So if you look at over here, I have mentioned in the bottom over here, the human blood is like 4.5 liters. The average concentration of the albumin in the human body is like 40 milligrams per ml, and we are talking about like having 180 grams being produced by the human body. We have lot of albumin in the body, and we do not know how to use it and how can it be helpful for us for the formulation, and we are just getting to scratch the surface and create better formulations out of it. So coming to the discussion of the chemical nature of the albumin, we need to know the excipient before we start making any kind of formulation. So albumin is a globular protein. This protein is like it covers nearly 60% of the human body proteome. That means it is significantly abundant. Just imagine like your human plasma that is extracted from the blood, nearly 60% of that is albumin. And remaining 40% is basically other proteins that are useful for doing other functions. So if you have so much of albumin, nature creates it. There must be some reason why nature needs 60% of albumin in your human plasma. So, and the concentration of that, like typically ranges from at 440 milligram per ml. And if you translate that into percentages, it accounts to 4% weight by volume. That is like a normal amount of albumin that is present in the human body. It is usually synthesized in liver, and it is approximately 13 to 14 grams of the albumin is present in the it's produced daily in the human body and circulated for actually its use. Now, one of the albumin's most advantageous properties is that it has a long half-life and extends the circulation time of the albumin-bound drug. So this ties back to your knowledge of pharmaceutics that like if you have a high protein bound drug, it will have a longer half life. It won't be easily available for its pharmacological use. But this information for us as a pharmaceutics people, we use this information. Oh, if the drug is able to bind to a protein, maybe we can formulate it and maybe we can create a formulation that has a extended release type characteristics. That's the difference between how a pharmacokinetic person thinks about it and how a formulated person or a material science person looks at it. So it extends the lifetime of the albumin bound drugs by improving its pharmacokinetics. The interesting part is because this albumin is a natural protein, it binds to the drug, it carries the drug from point A to point B. It functions by transporting the drug into the cell using these two proteins, GP60 and SPARC. That is a natural mechanism by which when, let's say, like if you have a drug which is over here, and you have a giant protein, it would actually combine with this and create like a complex structure. It will then go on the GP mediated receptor, which is, I'm sorry about that. So it actually binds to create a complex like this. It goes to the receptor that is GP60 and gets into the cell membrane inside the cell and creates this pharmacological effect. So you'll need these two receptors, that is GP60 and SPARC, that is responsible for transporting the drug into the cell for creating a pharmacological response. How does that look like? If you look at this particular picture, which is a depiction of exactly how the structure would actually go into the cell through creating these small vesicles. So this is your GP60 receptor. The green color dots are the complex of albumin bound protein to the drug, and it creates these type of membrane internalization, which then actually releases the drug into the tumor interstitium. The reason I am showing you this particular picture is because to give you an example of a cancer cell, because ultimately we want to use this idea of using albumin as a drug carrier for the treatment of conditions, chronic conditions like cancer. So if the cancer cell is actually expressing a lot of GP60 and lot of SPARC receptors, then we can actually help to have a precision medicine to the cancer cells. And in case of the cancer, because it's a food hungry uh, ball of tissue, and because it just keeps on absorbing too much of nutrients, you would expect that the drug would actually go and bind to these receptors and then translocate itself into the tumor interstitium by interacting with your GP60 and SPARC. And this is the reason why we call albumin as a natural nanoxenobiotic carrier. It on its own has this functionality of carrying the endogenous as well as the exogenous molecules inside the body from point A to point B. That is from your blood circulating vessels and coming in contact with the the cell membrane, and then transporting the drug inside the cell. Um, the next thing is that, like, why is this albumin so important? Like, why are we even talking about it from a pharmacological perspective? It was observed that the albumin has a really good correlationship with aging. And it was observed that if you look at this graph, the albumin in a young adult is significantly high. As the aging happens, it causes a significant drop in the concentration of albumin. And to think it this way, imagine like albumin is basically a buffer that is floating around in the blood, and it is carrying the drug from point A to point B, from one compartment of the tissue to the other compartment of the tissue. If this medium is actually depleting, there will be a lot of trash going to build up in the human body that is going to lead to a lot of inflammatory problems. So, from a pharmacological perspective, albumin is one of the most important protein, and that's the why the nature has created it and made it like 60% of the whole human proteome. And there has been a significant positive correlation with the drop in the albumin concentration in the body with the other diseases like arthritis, chronic diseases, heart failure, glaucoma. It is also responsible for the cancer prognosis, and it also is correlated to the neurodegenerative diseases. So all even my father has a significant drop in the albumin, and if you look at your blood reports, many times like hospital would look at the albumin concentration, The reason behind that is that if there is a decrease in the concentration, no matter how much amount of drug you're giving to treat it, that effect will never happen because the albumin has dropped and there is a significant increase in the waste material in the body, which is not getting circulated, and that leads to inflammation and chronic conditions. So that is the reason why there is a significant correlation of the albumin with the aging and age-related pathologies. Now, that is one thing. Like if you have an age-related condition and you are using albumin as a and there is a you have a drop in the concentration of the albumin with aging, and that leads to age-related diseases like cancer and other conditions that I just mentioned. How should you treat it? So we did a systematic review about exactly how these conditions are being treated in the patient in the clinical scenarios in USA and as well as in India. We found that like there was like a simple intramuscular or IV infusions that are given of the albumin, which is usually 25%. I've just shown you an example of 25% solution of the albumin USP that is usually given by IV routes for the treatment of conditions like hypoalbuminemia, hypovolemia, treatment of the ovarian hyperstimulation syndrome, acute respiratory distresses, acute nephrosis, hemolytic disease of newborn. These are some of the conditions that, for which this is commonly prescribed for treatment. In case of cancers, usually the infusion of the albumin is given as a supportive therapy to the chemotherapy so that like there is enough blood plasma volume developed so that there is no buildup of the waste material in the body that can aggravate the cancerous condition. So, technically speaking, albumin actually helps to improve the colloidal osmotic pressure. It drives the interstitial fluid into the vascular compartment and increases the volume of circulatory system. And that's the reason why it helps to make your body run. So you can think of like the albumin working as a fuel that actually just keeps on buffering your system and also cleaning it at the same time. So this like how the albumin is used. And to give you a perspective over here, the last point, the small tiny thing. I'm not a businessman, but I just thought maybe it would be more relevant to most pharmacy student. It says that the global market size is valued as approximately $7.23 billion in 2025, and it's projected to grow to 13.33 billion by 2034. Honestly, I usually don't trust most of these market surveys. So, because I had a privilege or I was lucky to actually work with some of the clinicians at Jefferson Hospital and also over here with Rochester URMC, your Rochester Medical Center, I was able to know that like these albumin IV infusions are most like usually used for the treatment or giving the support therapies for treatment of these conditions, including cancer, making it one of the most used preparations in the clinical settings. Now, if you notice, if you have like most students and like when we are discussing many of our pharmaceutical excipients that we use for making tablets, gels, all different types of formulation, have you come across any formulation that actually works as a drug as well as an excipient at the same time? I mean, to my knowledge, there was none. Albumin is actually providing that sweet spot where it is used as a drug on its own in the clinic and also used as a formulation excipient for making the nanoparticles and gels, films, and other delivery system. So we argued about this point with the United States Pharmacopoeia and US over here. One of my colleagues is actually sits in the expert group for the excipients, and we wrote this to them and we published this as an atypical excipient, where an excipient works as drug as well as formulation material at the same time. That was one of the key reason why we actually focused our attention towards the use of albumin for making different types of formulations so that the body does not recognize it as a foreign material and starts giving out hypersensitivity reactions. So we saw that albumin is actually dropping in the aging population. Then we saw that like albumin is used for in the clinics for making this for treatment with the infusions. The next thing is that like we know that albumin is used as a drug. How can we use that as a formulation? So this is a classical example where the albumin was first explored as a drug. So if you see over here that the drug name is called as ebrexane, which is an albumin a nanoparticle. If you guys look at the literature, it's a nanoparticle made from paclitaxel. So you have a nanoparticle where the drug is actually encapsulated in the shell of albumin, creating an albumin nanoparticle. Currently, there are three marketed preparations that I've shown over here, which are already FDA approved, where albumin is now used as a pharmaceutical excipient for making the nanoformulations. So here we have two stories. One, albumin used as a solution for the treatment of hypovolemia, and it's used as a drug product. On the other hand, it is used as a biomaterial that can be used for making these precision medicines. So it gives us an opportunity to explore and tap into this, and there are a number of clinical trials that are going on in developing these type of nanoparticles using albumin for drug therapies for treatment of various type of cancers. Looking at this is my favorite chart. Over here, you can see like a single material, like a simple structure like this heart-like structure, albumin, which can wreak havoc if there is a problem, but it can also form different types of formulations that are suspensions, hydrogels, composite film, nanofibers. Dr. Vasha, I hope I'm not running late on the lecture, right? Just let me know.
No, no, sir.
Okay. So we have like we are creating different types of form nano formulation over here. That is, we are making nanoparticles, nano emulsions for different types of drug delivery. We have hydrogels, which is currently one of my favorite topics, and I'll tell you why it is important. We can make composite films, and I'll also show you the example of how to make the composite film and how do you make the gels out of albumin. And then you can also create nanofibers through electrospinning techniques to actually make nanofibers for wound healing applications. One of my earlier work was actually, as you noticed, that like albumin was used for the delivery of paclitaxel. We actually were while I was in IISc Bangalore, I was introduced to this particular drug, luminesipib. It was a heat shock protein 90 inhibitor that was explored for the cancer therapy, and we made nanoparticles out of it using the albumin. That was my first experiment when I was doing my first postdoctoral fellowship. That's when I was like really convinced that like this is one of the coolest material I've ever seen, which can work as drug as well as an excipient if I want to play with it, if I know the chemistry how to play with it. So after having knowing about like how the nanoparticles are being developed, I just want to show you how these nanoparticles are developed. So here I have a video, like a short video to show you how the nanoparticles are developed using the desolvation. So you put the drug in the alcohol, like maybe ethanolic solution of the drug, and you put it into a dropwise solution into the solution of BSA. It actually starts to form nanoparticles through desolvation technique. It's like a controlled precipitation followed by cross-linking so that like the nanoparticles are developed. You can see over here how the nanoparticle suspension is being created. And these are some of the images that I captured through the SEM images. Here there is a better picture of the SEM image. So we had like the particle size of roughly around 2 to 2 nanometers. This was the SEM image that we captured, and these are the TEM images to capture and see like exactly how the nanoparticles were looking. That's the docking image of the doxorubicin that was actually binding to the albumin when it actually forms nanoparticle. And we explored it in the treatment of MCF7 cell line, that was breast cancer cell line. The reason I'm showing you the data that is more pictorial is because one of the reviewer asked me like, okay, you are seeing that like there is a bar graph of like exactly how the cytotoxicity happening. but I thought that maybe I can show it to you. MCF7 over here, when it is treated by only native nanoparticle, that is no drug in it, it forms these colony forms. Can you see this? It just creates the colony form and it's happy. All the debris that you see is like basically those nanoparticle chunks or some debris that are coming from the formulation. But if I use the drug over here, you can see this these cells actually starts to get transitioning into apoptosis, and it starts to form those blebs, which is one of the stages when it undergoes the apoptosis, when you are actually exposing to drug loaded albumin, which is a clear indication that these nanoparticle loaded drug loaded nanoparticle systems were actually working for the treatment of cancer. And this work was actually published in 2020, and it was one of the editor's choice paper. It was an interesting study that we published in one of the scientific journals. So once I did this nanoparticle study, the story goes into the idea that like if the material can form nanoparticle, can it be explored for making gels? That was my question. So we wrote an interesting review article recently got published in IJP where we were able to show that these gels can be used. So the albumin can form gels and it can be used for tissue regeneration, wound healing, anti-cancer therapy, and bone cartilage healing. But the problem was most of these gels that were published and that were actually available, they were composite systems, meaning albumin was combined with some other pharmaceutical excipient to give that rigidity and those flexibility to the or viscoelasticity to the system so that it can create these type of biomimetic structures that can be used for tissue regeneration or wound healing or for cancer therapies. But our agenda was that, like, what if the albumin structure itself can be actually molded into gels without any excipient? That's the goal. If it can actually self-assemble to form a nanoparticle through a simple desolvation method, can it actually be somehow by changing the temperature, pH, or some cross-linking that I can do to make this albumin solution into a gel instead of nanoparticle? That was the idea. So before I go into this, I wanted to give you the reason why I want to make the gel. Was I was working at Jefferson Hospital, and you can see this weird, ugly looking picture of the brain dissected for the treatment of glioblastoma and the insertion of these discs over here. These are called as glial wafers. And to our knowledge, this is the only implantable disc that exists on the planet for the treatment of glioblastoma after surgical resection. Meaning you're removing the tumor from the brain tissue, and because there is a cavity and because the surgeon is not able to find out that there is a cancerous tissue left behind or not, they will actually put these discs inside the tumor cavity and seal it. And if you look at closely, you can see these lacerations over here. These are unavoidable. So no matter how much they did, they were still not interested to do this. But because they did not have any choice, they had to go for these glial wafers for the treatment of the tumors. So they said that like, hi, why don't you just try out like some kind of formulation that can be injected over here? And that was the actually primary reason to make a gel out of albumin so that it does not create these type of lacerations when they're actually sealing it. And it is matching to the viscoelastic behavior of the brain tissue. These were the two main goals for which we were actually experimenting in developing different types of gels using the albumin. So going into the albumin gel, so here over here we have the representative figure. It can be injected like imagine like a surgeon removing the brain glioblastoma tissue and applying these gels through a syringe. So it can be injectable. That is how we were able to modulate and change the chemistry of it to make this thing more viscoelastic in nature. Over here, you can see that it can also form different structured system. And then we are able to put drug into it to make a delivery system for more effective and site directed application. So how does this work? We have albumin solution that is made in salt, common salt. No, nothing fancy. We have salts in the kitchen, right? That's the same thing I'm using. Albumin solution with common salt. I'm changing the pH of this particular solution. I'm writing over here pH 7.4 over here. And I'm heating it at 85 degrees Celsius for 20 minutes and it starts to form this gel. These gels can be it can be thick, it can be hard by changing the concentration of the albumin. You can make it hard or soft depending on your needs. So you can personalize the dosage form. So for the glioblastoma, we wanted it to be similar to that of the texture of the brain. We started with a 6% system, but if you really want a hard system like a implantable block, you can make different type of cubes or circular shapes out of it to make it like an implantable system. So, playing with the moldable characteristic, this was the moldable system that we had, but I wanted to play it a little bit more. So we actually use silicone molds to create something like Happy. One of our master students just went so happy with the gel formation. He said like, okay, let's make Happy out of it or make a name of the university using the gel. I was like, this is an interesting thing. I can put it in one of the published papers. So I was thinking of putting Dr. Rochani in the albumin gel and putting it on the manuscript. That is one of the wild ideas that I have right now. So, it you can actually make these printable gels, and what you're seeing over here is that like the same story can make a structured system, and if you just heat it for 20 minutes, you can make it like a soft injectable gel systems. And the key is that 85 degrees C, 20-minute heating, and with sodium chloride you have a clear, transparent, injectable gelling system. We characterized this using the dynamic light scattering. We were like interested into why this thing gels. Can we change its chemistry? How should we modulate it? We found that like when you take a solution and you put it in the zeta sizer, the particle size is roughly 3.6 to 7.5 nanometers. When you give a heat shock, it starts to aggregate. So now the albumin structure is getting denatured and creating like a aggregation system to create those gel like structures. So you can see that like with the increase in the particle size over here, as well as like as the time increased, the number of average particle size is also proportionately increasing, meaning the transition of sol to gel state can be actually be mapped using a dynamic light scattering experiment. How did it look like in the electron microscopy? This was never done before. And we were really excited to see like how the albumin structure looks like. So what you see over here, this thing, this giant crystal of like 1600 by 600 micrometer particle size, this is native albumin. This is just albumin powder that you get it from any vendor that would be there, like Fisher Scientific or whatever company that you, or Sigma Aldrich. This is how the native particle would look like. But when you actually transform that into a gel, it looks like these honeycomb-like structures. You see this? The porous structures where the different layers of the solutions are getting. This is how the cross-linking look like in the real-time situation when you actually put it under electron microscope. This is how we were able to map and characterize like exactly how the albumin would look like and how can we manage its viscoelasticity. So that's the NaCl gel that we made. Now, remember I said that like I wanted to make a gel that was matching to the brain viscoelasticity. So I literally took the brain and breast tissues and did the rheological experiment to see like exactly does this viscoelasticity matches to the gel that I'm trying to make. Ladies, if you have used or made uh, sugar syrup at home, my mom used to do that, put the sugar syrup between the two fingers and used to lift these two fingers and see those threads being formed. That is the kind of example that my mentor gave it to me, that is called a spin barkite behavior that actually says how viscoelastic a gel should look like. This is how the brain tissue was looking when it was crushed between the two system, and this is how the gel is looking. So it has to mimic that type of viscoelasticity before I implant that into the brain tissue. So what you're seeing over here is the data of like the storage and the loss modulus, showing you that like the bottom line over here, this brown color line is for the 5 and 6% gel, which is showing that my gels are slightly more um, viscoelastic. I mean that it's more softer compared to the brain tissue. Your brain tissue data is somewhere over here, this place. This shows that your it's a storage and loss modulus is a measurement of exactly how the hard or soft a gel can be. You need your gels to be softer and so that it doesn't create the friction when it's applied to the brain tissue, and it is easily able to solve the problem. So that is the data for like the comparison of 6% and 5% gel compared to that of the brain tissue. We did a lot of stability studies with this, and we found that with the if you look at this, this is the storage modulus data, and this is the loss modulus data. We could see that like at 37 degrees C, there was a drop in the viscoelasticity for both the storage and loss modulus data, indicating that the gels were stable for approximately 24 days. If you do it for longer time, it may not be helpful, which is kind of a good in our case because we do not want the gel to be present in the brain cavity or the breast cavity forever. We want it to actually give a controlled release and then disintegrate over time. So this gives us an idea of like how long can we actually make this gel to last, or because this is a 6% gel, if I had 7% gel, probably it would last longer. But in my case, I was more interested to match to the viscoelasticity and create a controlled delivery system so that we can have some data to actually move forward with the animal trials. Now the gel is good, so wanted to see if this can be working as a delivery system. So we added doxorubicin to it, and it was easier for us because doxorubicin A, it is water soluble. So it gets solubilized in sodium chloride solution. It creates this bright red color gel, which is visible easily, and you can characterize it and handle it easily for doing the experiment. And that's the reason why we used doxorubicin. And the other thing was we also found that doxorubicin, when applied, it can also be useful because we are thinking of putting it into the breast tissue for trying it for the breast cancer and also for the brain tissue. When it's applied locally, it can actually provide that anti-cancer effect. So that was another technical reason why we wanted to use doxorubicin to try if these gels can be used as a matrix for drug delivery. Now, the question was when I mentioned to you that when you use albumin in NaCl solution, heat it for 20 minutes at 85 degrees C, it gels. So you need 20 minutes to gel. What we found was with the doxorubicin, this thing actually gels in 15 minutes. This was the first time that we were able to see that somehow the drug itself is working as a cross-linker. We have no idea why it happens, but because we are right now characterizing it. But this was the first evidence to show that the viscoelasticity of that 15-minute gel was significantly better, and maybe the doxorubicin is actually helping it to gel. If you look at this 10-minute thing, the albumin solution is completely liquid, but it is gelling in case of the doxorubicin, which actually feeds into this idea that maybe the drug itself is working as a cross-linker. So we took the SEM images to see like exactly how similar or different we are having differences, similarity or differences that we are having with the doxorubicin versus native gel. You see this, the comb-like structures are conserved in both native and doxorubicin like gel-like structures. These comb-like structures becomes more prominent so the 20-minute gel comb-like structure is similar to that of the 10-minute doxorubicin albumin gel, which also goes into the point that like maybe doxorubicin is actually helping it to gel faster because of the ionic cross-linking that we are trying to see. We went into checking for the stability studies of it, and we developed a calibration curve to quantitate the degradation profile of the drug, We know that the albumin gel was actually degrading at 24 days. We do not know if the drug itself is also getting degrading earlier or maybe later on. So we developed a stability indicating assay to study this, and it was done using high performance liquid chromatography. And we developed a stability indicating assay to map this degradation. And what we found was the doxorubicin itself also gets starts to degrade on roughly around day 10 at room temperature. So you see over here, it starts to degrade, and by the day 30th or something like that, it is the degrade like nearly the percentage change has dropped to roughly around less than 40% in the degradation profile, which shows that like ideally speaking, I would actually store the drug and the drug loaded albumin gel at 4 degrees C, probably for maximum 4 to 5 days. But this is also a good point that like if the gel itself is actually disintegrating, it can also help the release of the drug over a period of time. And that was confirmed when we actually did the I'm sorry about that. We did the release kinetics of it to see like exactly how the percentage release is happening over time. And we were able to see that the saturation of the 15 or 20-minute gel is happening around 20-hour time point to show like a controlled release. Whereas on the left side over here, you can see that like 15, 20-minute gel, they are like showing the straight lines. When we looked at the anti-cancer effect of this, the with the gels, if I just focus over here, this is like 100 microliter of the gel. You can see the MCF7 cell lines were actually growing really well on the surface of the gel, and the morphology was conserved. If you look at 100 microliter gel with the doxorubicin, you can see those tiny blebs being formed over a period of time, indicating the apoptosis happening, which shows that like the gels were actually releasing this drug over a period of time and creating an anti-cancer effect for the MCF7 breast cancer cell line. Now, an interesting part that we are trying to focus right now is that, like I mentioned to you, that the drug itself was working as a cross-linker, and we were using NaCl as the cross-linking agent for most of the gels that we made, and I showed you previously, like all the data that I had shown was about NaCl. One student came to me and said that, like, hey, Dr. Rochani, we can actually make gel faster when you use a divalent cation. And maybe instead of 20 minutes, probably we can just make it in 5 minutes. But the problem is the gel is opaque, and I'm not sure if this is going to be a good idea. I said, like, okay, this is interesting that the gel is becoming faster, and I don't like this white color blob because it's basically like your egg white. And that is like not structurally appealing to me. But it was an interesting situation where the gel can be made earlier so that like if like doxorubicin could handle heat of 85 degrees C for 20 minutes. I do not know if other drugs can also have this problem. Yes, Dr. Tiwari?
Dr. Ankit, can you summarize in 10 minutes?
Yep, it's almost done.
Yeah, okay.
So we started exploring this idea that maybe if we use a divalent cation, maybe we can actually make it gel faster. So that's why we first did the characterization of how the viscoelasticity looks like, whether it is forming hard gel or soft gel. So over here, you can see the data of NaCl where you are increasing the strain and it is becoming more um, compressed and it's denatured. When you reduce the strain, it starts to go more elastic and come back to its original state and make it more elastic in nature. It's like your rubber. You stretch it, it becomes more deformed. You release the rubber strain and it come back to its normal state. So this drop that you see is basically the explanation for its elasticity. So that's our standard NaCl gel, 6%. If you see the KCl, we can see that this thing is more softer in KCl. And if you look at the MgCl2, this is coming all the way back to its down. That means it's more hard and more rigid in nature, like a rubber. So the NaCl is making a good matrix, but we thought that maybe some divalent cation can also help us make gel faster, maybe. That was one of the hint that I had from this particular experimentation. So an year ago, a student came in and said that like, hey, Dr. Rochani, you can see over here, he used CaCl2, a divalent cation, and he said that like if I heat the solution of albumin in divalent cation with a concentration of roughly around 5 millimolar, we can get it gel in less than 60 seconds. This was one of the biggest thing that we were. So NaCl was making gel in 20 minutes. CaCl2 was able to make a gel in 60 seconds. Where we can actually now explore like maybe some heat sensitive drugs can be also be used as a delivery for using this particular thing. So to summarize, clinically, albumin is used for the treatment of hypoalbuminemia, volemia, and all kind of clinical disorders because the drop of the albumin, it is basically helpful in creating a natural homeostasis so that the life can be improved. If there is any perturbations in these homeostasis, we have a solution where you can actually take IV infusions of the albumin, making albumin as a natural drug for the treatment in clinics. As a formulator, I use this nature of the albumin where it is an albumin solution. Played with the chemistry of the pH, with the salt and the drug molecule. We were able to manipulate its chemistry and make a gel out of it and nanoparticles out of it. These gels were injectable, and we found that it can be injectable or it can have those three-dimensional structures molded out of it. We saw that 6% gels were injectable, whereas if you increase the concentration to 7%, it can make a structured system. We used doxorubicin as a model drug for delivery, and we also characterized it for stability indicating assays. We did the rheological evaluations of this. We checked its therapy against the breast cancer. We found that like maybe use of divalent cations can help us make gels faster, and that's what we are doing right now. One of the student is working on the CaCl2 gel, and another one is working on this. Showed to you that the albumin can form three-dimensional structure. We are now working on 3D bioprinting, where you can actually print the structures using the albumin gel and can be used as a scaffold to see like how the cells can grow on top of it and maybe can be used as an implant. The other picture over here, that's the example of how you can make albumin using films. So maybe after this, if you anyone is interested to look at like how to make a film, we can schedule an appointment and we can talk about it. Thank you so much for listening to me, and I'll be happy to answer any questions you may have.
Thank you so very much, Dr. Ankit Rochani, for such an enlightening, insightful, and thought-provoking lecture. Your presentation beautifully highlighted the role of sustainable green biomaterials, particularly albumin, in advancing precision and biomimetic therapeutics, and clearly demonstrated how transitional pharmaceutics can align with the clinical needs, regulatory expectation, and environmental responsibility. The depth of your discussion on formulation science, advanced drug delivery system, hospital pharmacy support, and biosimilar characterization has provided our audience with a valuable perspective that bridge fundamental research, clinical application, and real-world pharmaceutical practice. We truly appreciate your valuable time and scholarly contribution to this conference. Dear participants, if you are having any question, the session is open. In the meantime, I would like to ask some questions. What are the
Yep,
desirable characteristics of drug that can be incorporated within the albumin?
fascinating. So albumin actually in the physiological pH exists as a negatively charged protein. And we would like to have proteins that are positively charged that can actually interact with the albumin and create a complex bonds. But that is at the charge state. But to answer in more detail, most drugs are either hydrophobic or hydrophilic in nature. And the BCS class 4 drug is the notorious class that I call, and one of the biggest problems in the pharmaceutical world is that BCS 4 class, where the most drugs are hydrophobic and difficult to formulate. It doesn't go in water. It doesn't get. We can't make tablets out of it. We can't make anything out of it. It's just like given with the DMSO for IV infusion and creating more problems in the patients. So what I did was I showed you an example of a water soluble drug that was doxorubicin, and it has it's positively charged and it binds to negatively charged protein. It forms gel. We are all happy with it. I also worked with one more that drug, which are two drugs actually, which I have not shown data over here. One was ifosfamide and another one was temozolomide. Both of these drugs are hydrophobic in nature because the ultimate goal is to play with the drugs like paclitaxel, which is BCS class 4 and hydrophobic, temozolomide hydrophobic, ifosfamide hydrophobic. Most these drugs can be actually incorporated to make gels. I've not shown the data over here. Maybe Dr. Tiwari make one more session and I can show you that data too. So it can be, it's a universal material that can be played with, and you can create different type of formulations with it by using techniques like co-solvency, where you can you can dissolve the drug in the lowest concentration with DMSO or ethanol, and then put that into albumin such that the drug doesn't precipitate out. And then you can actually make a gel that is more uniform and injectable in nature. So you can do both for the hydrophobic, hydrophilic, and preferably I like positively charged drugs that can bind to negatively charged protein.
Right. Apart from albumin, what are other cost-effective proteins that can be used?
Other materials, you mean? The excipients. I have worked with carrageenan, chitosans. These are the polysaccharides that are mostly used. The other one is hyaluronic acid, which is like commonly used in cosmetic industry in United States, and all the girls are really fan of like hyaluronic acid lotions and solutions, and they are like sold over the counter. So the recent material that we are working on is hyaluronic acid, and you can use gelatin. That's another protein, or I won't call it as a protein, it's a polypeptide. So collagen actually degrades into gelatin and we make capsules out of it. So you can also use gelatin, which is readily available for making these type of gel systems. But then you will get into the problem of, as an academician, we always look for novelty, right? Something new, something innovative that is not done before. Gelatin is like curcumin, which is like there in all the kitchen, and we use jellos and like it's all there. But carrageenan, chitosan, hyaluronic acid, these are some of the interesting materials which has gained a lot of more momentum from the sustainable green material. And my personal favorite, which is like not touched and like really good one, is that DNA material, which is again not explored much, and it's really novel in nature, where you can use DNA as a biomaterial for drug delivery. That's fascinating.
Right. Now I would like to invite our chairperson, Director of MIT Institute of Pharmacy, Professor Dr. Anuradha Mishra, ma'am, for words of appreciation.
Thank you, Dr. Prasha. Thank you so much, Dr. Ankit. Throughout your session, I was there, and I come to know personally so many things. Just like your students, you are showing your research, and bit by bit, starting from the percentage, in which percentage you have started. Just I felt that the research is going on in front of me, and you are sharing your knowledge. Thank you so very much for sharing your inputs and to think beyond the traditional way how to deal and what are the novel ideas how to make these gels, whether from its moderate form to advanced form, that is in the nano gel or nanotechnology, which can be again investigated in any other directions. Like I found most interesting in the glioblastoma, the you are prepared and you are working on that to introduce really that are the novel fields and that needs research, and researchers like you can think beyond that. We are really proud to have you here, and definitely in future we will ask you to join us, and whenever you are planning to visit India, please be visited to Amity University so that you can interact. Thank you so very much to be.
Thank you, Dr. Mishra. Thank you so much. It was pleasure.
And thank you, Dr. Ankit, for accepting my request. And I know it's almost 11 or 12 in night there.
Yep.
So during due to this hectic schedule, also you have accepted my request. I am very thankful to you.
Thank you so much. And it's good to talk to you guys. I mean, like, we are doing good science, and if it can somehow get something really cool out of it, then nothing beats that. And I'm always happy to contribute. So let me know. Yeah.
Yes, Dr. Ankit, we can plan for collaborations. You are
Yep.
asking our faculty
I'll share my
members or
email
someone.
to you guys if
Sure,
you
sure.
we can talk. Okay.
As a
I'll
token of
share my.
appreciation, love, and gratitude, we would like to present e-certificate. I would like to request our IT team to present e-certificate.
Thank you.
Kindly accept it, sir. On behalf of the organizing committee, I extend our sincere gratitude for sharing your expertise and inspiring future research and collaboration in this important domain. Thank you so very much.
Thank
With
you.
this,
See you.
I formally conclude the lecture, and I will now request my esteemed co-host, Dr. Jishan Fatima, for the technical session 6. Over to you, ma'am.
Thank you so much, ma'am. Am I audible and visible? मैम यहाँ से मेरा background नहीं change हो पाएगा. Just give me 2 minutes. Thank you so much, ma'am, for giving the platform to me. As I take upon the next speaker for the session today. Dr. Ravi Maharajan, sir, are you there? Dr.
Yes.
Ravi?
Yes. Yes, I can hear
Dr.
you.
Ravi, are you there?
Yes, I can hear you. Yes, I can hear you.
Sir, am I audible to you?
Yes, yes, ma'am, please continue.
Mm,
Dr. Ravi, am I audible to you?
Yes, I can hear you. I can hear you, ma'am.
Oh, okay, so you're not visible. Sir, I cannot see him. I cannot see Ravi, sir.
Uh,
IT team, can you please look into this matter? Sir is not visible. Participant, please wait for some time. We're just getting the thing resolved. But sir, is it possible for you to share the slides so that I can proceed?
Yes, I will do.
Sir, please put the slides in a slideshow mode. I take this privilege to introduce Dr. Ravi Maharajan, who is a distinguished researcher in biopharmaceutical manufacturing and has a specialized expertise in applying artificial intelligence and machine learning and this digital twin technologies for the development of mRNA, siRNA lipid nanoparticles vaccine. With more than 26 publications in reputed high-impact journal and active involvement in editorial roles, he has significantly contributed in improving the stability, scalability, and therapeutic performance of RNA-based medicines. His research is centered on optimizing lyophilization process and advancing innovative drug delivery system to enhance the efficacy, safety, and accessibility of RNA therapeutics, while promoting sustainability manufacturing process. Dr. Ravi also plays a vital role in mentoring emergencies research and collaborating with international scientific experts, driving progress in pharmaceutical sciences. His proficiency in microfluid nanotechnology and advanced analytical techniques positions him as a key contributor to next generation pharmaceutical innovation. Today, he'll be delivering his lecture on a topic, Sustainable RNA LNP Vaccine Platform, AI-driven optimization for green nanomedicine. May I now request Dr. Ravi to proceed with the lecture? Sir, the platform is your.
Thank you, ma'am. I'd like to extend my warm greetings to all the professors and viewers attending the conference. For the conference, today I'll be talking about the applications of RNA LNP vaccines and its continuous manufacturing using the AI/ML model. So I will be covering on the general brief descriptions relating with the green nanomedicines for the vaccine manufacturing using microfluidics techniques and the incorporation of different stabilizers to make the vaccine stable at the room temperatures. And for this, I'll be using the implementation of artificial intelligence and machine learnings to optimize the stabilizers formulations and make it stable in the GMP scale higher batches. And after that, I'll be incorporating all the data to fit the digital twin structures to make it possible to make at a large scale at a lower cost and less more efficient times. And I'll be concluding with the presentation with future perspective that we can go further with this with the studies. So the first chapter is about the green nanomedicines. In the vaccine world, it's very difficult, challenging to have storage at the room temperature because we need to have storage in the cold chain facility that consumes lots of energies, and also storing at -70 degree requires dedicated facilities, and it not be possible for the less developed world, and the resource may be wasteful because we need to have more ethanols and buffers and resources to make it the preparation. So my study is focusing on the reducing the cold storage need, less burden for the storage time, and make it production in less resources. So the objective is to, since the market size is growing each year, so the objective is to make the packaging to the lyophilization process and eliminate this cold chain necessity. And to do this, because of implementation of artificial intelligence and ML, it will reduce the time, cost, and the manufacturing operations. So using the minimum cost is the one of the goals. So in this, we'll be reducing the waste with less hazards and using the safer solvents and with the minimum degradations for the prepared vaccines. And for this, in the LNP manufacturing structures, it will be continuous manufacturing within having more than 95% encapsulation efficiencies with less use of ethanols and less minimum use of the buffers. And it will be stable for the ambient temperatures. And the use of AIML will help us reduce the solvent waste and have predictions of its stabilities at different temperatures, preferably at the room temperature, with a fewer number of experiments and optimizations. And in the lyophilization steps, we attempt to reduce from -70 to up to 0 degree refrigerator temperatures. So with a less introduction of biodegradable excipients and stable at the room temperatures. So for this, in the preparations of RNAs with the lipid nanoparticle technology, we are using combinations of four lipids. First one is ionizable lipids, and second is phospholipid with the cholesterol and PEG lipid. The combination of four lipids and the diluted RNA LNP, and we will be preparing using the microfluidics technologies. And the key parameters that we will be evaluating will be encapsulation efficiency, biodegradables, and room temperature stabilities. And in this study, we'll be discussing mainly about the siRNA, and the second one is mRNAs. So in this process, in the microfluidic process, we'll be feeding from one channel the combination of four lipids. From here, the diluted siRNA or mRNA in probably in the ratios of 3:1 or 5:1. And this chip will mix the two lipids and RNAs. We can have an instant dilute with the buffer to make it stable, and we get the finished product here. Since it's stable in 7.5, 7.4, we diluted with the fresh buffer 10 or 20 times, in between 10 and 20 times. For this, we attempted to make with different chips. We can have chips like hydrophobic focusing or SHM or mixer or flow microscope and toroidal bifurcated microscope. And among these, we found more processing wise, more efficient for this one, TFR. So the next goal is, since we mixed this lipid in the ethanol and this is buffer, we need to remove the ethanol portions. So the mixing at lower ratios and doing the fast mixing will help us reduce the concentration of ethanol initially also. And with the dilution, it also decreases the percentage of ethanol. And once we have the dialysis, it can remove down to less than 2%. This TRM, this microfluidic chips has more advantage over other chips, since it consumes less than 40% less solvent consumptions, and the manufacturing conditions are precise. We can define the flow rate ratios and total flow ratios, and it doesn't have much deviations. Once we change the batch size, we increase the batch from 4 ml to 40 to 55, more scale up to up to 240 ml. It doesn't have much change in the characteristics, so it's I prefer the TRM chips while making the lipid nanoparticles using microfluidics. So in the conditions, uh, in our experiment conditions, the optimized parameters was came to the flow rate ratio of 12 ml per minute, and the flow rate was in between 3:1 and 5:1 FFR ratios, and the flow rate is in between 12 and 20, and the optimized was within 20 and 15. So this was based on the 150 formulation screening through the chips in the TRN, and we got the optimized forms with these conditions. And after we get the LLPs, the pyrolysis kit we used was 10 kDa cassettes for around 5 ml samples to make the buffer exchange, and the reconcentration was possible using the tangential free flow TFF concentration capsule pulse. The next one we tried was we used these machine characters for the manufacturing conditions, for freeze drying conditions, and this is for the characterization aspect. And for characterization, we mainly use the DLS for the zeta potential, particle size, and PDI, and encapsulation efficiency using uh, the cartridge chips. And nanodrop was initially checked to check if the particle size was remain constant or not. During comparison, we also try to compare with the traditional mixings with the two syringe and also compare with the extrusion method, and we found that microfluidic mixing was efficient with aspect to solvent consumptions or energy consumptions or process flow. It was very easy to use and waste was minimal during that production scales and reproducible. We can scale up to 45, 40, 50 times scale up. It's possible. Next, we compare the this nanoscale with the recent machines available in the market. We generally use to compare with the nano-assembly ignite. This is the model we used to do during the experiment, and later we use also contact with the in-cell masters. It depends on pressure difference and mixing in the same principle. So one of the machines extruder and entity for the scale up and the mixing procedure. This one is which is cheap, cost and efficient to make. But for the long term and at scale up purpose, we can use the N-cell and Ignite Nano. This both instruments are preferred for scale up and with minimum deviations. For formulation aspects, we attempted to screen with the phosphate buffers and trace buffers during the dialysis times, and we could found that the change was minimum when we used the trace buffer, and it was more comparatively stable uh, in trace buffer along with the tray halos. With this was the machine we used during the experiments at 2 flow rate ratios and 3 total flow rates, and we used the cartridge of 10 kDa. Sample was around 4 ml and 5 ml, and we used NanoCon tube to reconcentrate for small sample, and for scale-up samples, so larger samples, we use TFF. And we found that when we compare with the total flow rate and flow rate ratios, we can found that only uh, these ratios particularly affect the particle size formations and encapsulation efficiencies. And when we compare with the Incelt and Ignite machines, we could found that this was particularly effective in HCR. It was it changed it effect with the ratio affected with the encapsulation references. And scale up can be possible if you control the DFF and FFR in the optimized ratios. The next one we compare. This is the encapsulation efficiency change. This was the initial, and after the for freeze and thaw, we store it at the refrigerated temperature for 1 day. And another story is we kept it the sample at 80 degrees centigrade for 2 days, and we can found that uh, freeze and thaw mainly affected the particle size at that time, and temperature was mostly influential in changing the encapsulation efficiency and the RNA contents within the prepared formulations. We, to make it stable in the freeze thaw and temperature, we try to make it we try to add more stabilizers to make it more stable. In that, these are the conditions. In the stabilizers, we tried with the four amino acids initially with the glutamine, cysteine, glycine, and alanine. And later we tried with arginine and acetylation forms and hydroxyl forms. And among these with the 10% halos and additions of amino acids, it was possible to make it more stable during the lyophilization process. Uh, we believe it's stable because of these interactions of the OH bonds with the proteins during the interaction of dilysis process. It may be possible due to the trials and making it stable with the conjugation of these amino acids. In this case, glycine was more stable within the selected experiment conditions. The next one is out of the four lipid structure. The last one was the PEG lipid, and we try to change the amount of PEG to make it stable. We tried with 0.5% and 1% and tested at BI7 and 8 conditions. And when we tried with the freeze thaw and fluctuation of temperature at that time, for this we used the siRNA because it was more cheaper and uh, easily available at the time. And we checked with when we characterized the sample with 0.5% and 1% PAZs, and we checked for the generation of particle size after the conditions, after freeze thaw and after high temperatures. We could see that before agitation, it was change was minimum, and after the agitation in the 0.5% PAZ, it was fluctuating more. When we increased the pH samples to 1%, it was able to control the particle size change. Similarly, when we noted the particle size in flow cam within 2 and 10 micron and 10 and 25 micron and more than 25 micron, 0.5% pH addition clearly shows the visible aggregation at that time. So when we increase to 1% sample, there is no visible. It was more kind of stable and no slow particle was observed in the flow cam data. So we tried heat crystal and again tried with the salt additions if it would make it more stable. So in these conditions, we tried with the additions of sodium chloride salts in this is control samples and making introduction with the 25 millimoles, 50 and 100 millimoles. And we can found that higher additions of salt was not particularly beneficial to this its stability. So the flow can shows it's a bigger particle size aggregations here, and these are getting more bigger particles aggregation particle size here. So minimum of salt addition is only preferred. Then we consider the study of this heating or freeze cycles on the long-term stability aspects, and it can be seen that this untreated, we can see in the last figure, it was the minimum before the stress, it was unchanged, and after the stress, we can see only 1% PSC additions in the Olympic structure have more stable, could stabilize the samples. Similarly, when we change the freeze-thaw cycles, increase the freeze-thaw cycles like 3 times, 5 times, and 7 times, still it could provide more or less stable formulation cycles up to this cycle 7, and this 1% cycle is here. It was comparatively more stable after 1% addition of PAG. So we try to study to make it vaccine formulation stable in the room temperature. So we follow the vitrification theory to we find out the TG dash. If the TG dash is 10 and 15, minus 10 and minus 15, and we try to add the stabilizers, the amino acids, or some polymers as well to make it below minus 20 or minus 25 to reduce the TG dash to make it stable and keeping the temperature stable for 4 hours at in between minus 10 and minus 15 to make it more stable, and primary drying was faster. Here we tried with 3 stabilizers, mannitol, sucrose, and 3 halos. In this, when we tried the same siRNA formulations with the 3 sugars, and we could found that only the trihalos at the 10% concentration give us more stable forms, and we feel this is the desired one based on our this data, that we choose the 10% trihalos and attempted to add other uh, stabilizers along with the sugar content for the stabilization aspects. So when we tested during liquid phase preparations, we could measure the pH during different. This one is DW water, and this is single concentration of TE, and this is 1.5x phosphate buffer, and this is tris buffer, and you can see that it changes the pH during the lyophilization. So there we need to add, uh, besides stabilizers of sugars, we need to add other stabilized forms, additional stabilizers also to make it stable if you want to have a stable form during the powder formulations of LNPs. So here it's change. It changes water is more or less stable. And to make it stable in other buffer states or after lyophilizations, uh, we need to have secondary stabilizers. And the question we have at the time is, do we really need the buffer? And we try to do without buffer at the time, and we found that if you want to go to the powder form, buffer is necessary for the stabilization. But we can remove the buffer and stabilize in powder forms and add additional stabilizers or polymers or some kind of combination of polymers. So we tried this again with sodium chloride salts. The reason for not having stable after the addition of sodium chlorides, we think it's because of the osmotic pressures that it changes and extract out this uh, that remain in between the particle size and make it unstable, less stable during this process. We tried with different forms of stabilizers like acetylase forms or hydroxy forms and some with arginine or other forms to make it stable. And in this case, we tried this is the acetylation forms of arginines at 10 millimoles and 50 millimoles. And after lyophilization, the particle size was not significantly increased. It was more or less stable. And encapsulation efficiency-wise, also after lyophilization, it remained stable when the concentration was 50 millimoles and the amino acid was acetylation form. So we further check with the DSC data, and it was there was very less change, and it was not since it's within this monostudy, it's possible to do the analytic process to make it the stabilization forms. And the possible mechanism we propose for this stabilization is because of this NH2 of the amino acids and reacting with the RNAs to make it stable at lesser dosage forms. We tested the samples of these lyophilized RNA samples and the fresh samples in the cascade cells, and we could found that when we tested with the 10 millimoles nano samples and 50 mol nano samples, and before and after lyophilizations, it was more of similar type. So we didn't find much change after that. Uh, from this data, we are able to confirm that it was stable when the concentration was 50 millimoles and the acetylation form was used. And after a month, it was still remaining more or less. This is a particle size. It didn't change much, and encapsulation efficiency remained more or less to the higher concentrations in our desired concentrations. So then next step we moved on to is since we have the stable form of the RNAs with the stabilizers, with addition of stabilizers, and that one we tried with mechanical space if it still remains stable. So give the 100 RPM at sorry, this was little blank. We tried with this 100 RPM rotations at 25 degrees centigrade for 5 days. If it can, the stabilizers, the acetyl amino acid can still make it stable for long duration action. And it was found that it's possible to make it, the change was minimum with an acetyl arginines. Encapsulating efficiency-wise was also after stress, the in compared to phosphate buffer, DAPC, or this is just simply water or and arginine. Acetyl arginine was able to preserve the encapsulation efficiency. The duration was minimum compared to others, and the possible it may be the possible mechanisms like because of the crystallization formations, there are small particles entrapped inside within the LVPs, and it was able to make it stable due to interactions and prevent the leakage. uh, we initially recommended not to use the buffer for the liquid solutions. If for the lyophilizations, then it's uh, together with the secondary stabilizers, it's possible to use buffer like trihalose amino acids and combination of another one amino acids. It's possible to go to the doing buffer and make it stable in the powder forms. So after we get the formulations, we moved on the application of AIML to make it scale up to GMP batches with less time and more efficiency. For this, we initially tried the definite screening design. It's a kind of design of experiments to screen the parameters. Uh, in our case, we used for the evaluation of this composition, TFR, FFR, buffer conditions, and concentrations to evaluate, and the critical residual conditions. We we took the particle size, PDI zeta potential, and encapsulation efficiency uh, to make it scale GMP scales for the how we use design definite screening designs to generate the 18 different combination of formulations containing these five parameters and with four responses. Uh, and the conditions are the upper and lower conditions are set as this one. This one is lower is 3 and higher is 5. Similarly, flow rate 12 and 820. NP ratios within 6 and 10, and for lipid types they used four lipid types combinations, and the ratios we used Pfizer ratios and Genboy ratios to compare the, and for the output response we consider for four response. After having the design of experiment data, we fed the DOE model to the artificial neural network to see if it can predict the optimal conditions when we change tweak some of the characters, some of the parameters in the input variables, and still it can give the predicted output response or not. So we fed to the artificial neural network of the optimized DOE data. Then for this we used the bootstrap model and SVM. There are three models for the optimizations used in the model. First one is this XGBoost, boost optimizations, and SVM. For this, we screened our responses based on the 18 experiments and draw it in the design of experiment to have a sweet spots. And we could found that the particle size, PDI, zeta potential, and encapsulation efficiency, the desirable uh, uh, conditions are within. If we increase the TFR here, it was possible to have desired particle size, and with the encapsulation efficiency for this one it remains also same. And for the this one is second conditions for particle size, encapsulation efficiencies, and the this one is encapsulation efficiencies, and this is zeta potentials. And if we increase the TFR and FR, we can have a more the faster the process, we have the higher encapsulation efficiency and consistent particle size. So in this yellow zone, it was possible to have the optimized formulation conditions within these structures. So the next one what we did is we fed the conditions, uh, the the our experimental data with the predicted models and compare both experimental and predicted ones for the four conditions: particle size, PDI, G-transmission, encapsulation efficiencies. For this, we have made 18 data sets and run 15 experiments for training and validations, and run remaining three we used as a testing purpose if it gets the same predictable data or not. And we fed to bootstrap forest at these times And what we found from this is the green one is the bootstrap that we have a prediction this one. And what we found is it was more close to the predicted lines when fed to the this model. So we again tried this model with the support vector machines. The dark blue one is the support vector model, and we could see it can have the similar prediction with accuracy uh, for the optimized conditions and for the optimized LNPs. We tried with lasso, but it was not much accuracy. It was not much acceptable from this one. And this artificial neural network, in combining with the GOE, this gave the higher number of accuracy rates to predict the particle size, PDI, encapsulation efficiencies, and zeta potentials in these aspects. Uh, In this model, when we fed to the artificial neural network, we use the combinations from TANH1 and TANH2 first layer, and second is Gaussian second layer, and this is first layer, and this one is second layer, and we choose the condition from 0, 50, and 100, and out of this combination of six different and three conditions, 0, 50, and 100, this 50, 0, 0, 50, 0, 50, these conditions gave us the uh, stable form of that predictable LNP formulations. In combination in this first layer and second layer using an Gaussian second layers in these combinations, we draw its R square values, and so it has the this is for the validation purpose, and if it is accurate enough to have predictions uh, based on this one. So based on the R square values and this R square fit values and this SSE's training models and this validation models, we can say that for the higher prediction accuracies for these four parameters using ANN was a suitable model to have the uh, predictions in the optimized conditions. So we can have based on the predictions, we can have the experimental conditions. So this saves us a lot of time because when we use this one condition, we have optimized one condition, we can use another siRNA or another mRNA conditions and can do the same with less frequent experiments. So after having this DOE also and optimized model also, then we move on to next conditions to have the this develop this next model. This XGBoost is making the running with the minimum experiments to have the LNP samples, maybe siRNA or mRNA LNP samples, and optimizing use bind optimizations. For this, we again tried with combination of four lipids with the mRNA sample. Previously, we tried with siRNA, so this time we tried with mRNA. And the combinations, we took more input conditions. This time we consider six input conditions, increase the input factors, and also output response we increased to eight output response, including this hit rate cycles, recovery ratios, and encapsulated mRNA. Still, when we increase the input factors and increase the output response, still our optimized condition was able to give more or less predictable results. And when we use these conditions to make the samples in the real world experiment, still it gives the desired conditions. The output response was within the desired limits. So this one is used for the prediction of the conditions, and this one is used for the combination of lipids. This four lipid conditions was using this one self-validated ensemble model, and this factor conditions was optimized using XGBoost, and later this was fed to bind optimization to have the desired processing parameters, so that we can have a LNP samples in large scale with the same characteristics. Even we do change in machine in in-cell or ignite, it doesn't change much because of the validation and this fed data to the models and its prediction. It doesn't change much. Uh, Even if it changes a small change in batch size or factors or out of two machines, we choose. We consider this PLS for the positive affecting and negative affecting for the conditions based on these input parameters. So in this machines and from the PCA and in the PCA and PLS, we can found that the most affecting to the LNP structures is from this pH lipids and the this one pH lipids and the this phospholipid to pH lipid ratios. These two influence more during the formulations and in the long-term scale stabilities. So the next one we checked the both models and increased the batch size to check if it is more stable in the real world conditions and check the large scale with the CPK values. And since it was higher than 1.3, so it meets the GMP scale manufacturing conditions like Six Sigmas. When we used the model, and accuracy was more than 94%. In that, if when we took the small number of experiments. So next we do is we choose the optimal design of experiments to evaluate the 24 mRNA formulations based on the DOEN model and based on the XGBoost. And we found that the biooptimization models prepared from this was within the predictable range within the experiment conditions, the output response from the experiments and the predicted conditions were uh, within the desired and our predicted range. So we could see this desired range for the particle size, PDI, this is for zeta potential, this is PK, heat range, uh, all the eight conditions that we choose fall within the desired conditions after we had the XGBoost model optimizations and have the we used with the bind optimization to have the uh, consistent sample. So this one is and we uh, use the predictions to around 10,000 more samples, generated samples. Out of 10,000 samples, we selected few predicted conditions and run on the real. We did actual experiments from the selected conditions, and it was found that it was within the Six Sigma conditions that it meets the conditions within that ranges. And this one is the uh, predicted conditions, and the one is fed with the experimental data conditions here. So even if we do less number of experiments, the higher number of predictions and optimization will help us to give us uh, a stabilized form of samples with very few experiments after this stage. So for the for choosing the optimum lipid ratios out of four, which one is optimum lipid conditions, we use this self-validated ensemble model to find out this ratio combinations, and it was the machine accuracy was more than 95%, and the predictions generated stable forms in the and it was stable when we kept it for the stability studies also it was stable based on this prediction models, and uh, our sample was more stable in accelerated conditions. For this for the four conditions and these conditions, this four and these four conditions, we fed the experimental data into the model, and we found that when we choose, when we set our desired conditions from this model, and this desired this upper and lower range, and this average desired conditions for like particle size, zeta potential, we set for all eight conditions, and we run on the machines to have our what should we set for the experiment to get this kind of results in the desired run. And for the combination of lipids, we choose the ratio. This one ionizable lipids and this phospholipids and this cholesterol and pH lipids. If we choose in here the zone in the this side, and this combination lipid in this zone gave us the optimum vaccine formulations, which was stable at room temperature and for longer times. And it is applicable for both siRNA and mRNA as well. Based on this one, we do two separate experiments again to verify the applicability of this SVM, and we found that the upper response was within the range, within the predicted range, and within our desired range. So this was the accuracy, validation accuracy was within the desired range. So next one is after we have a stable formulations and after a longer time and in the room temperatures, we checked with different AI models to check uh, if it can also identify the smaller aggregated particles or foreign particles or degraded particles within the detection samples. So uh, in this we found this out of compared the model CNN was more uh, reliable and have higher accuracy to predict the this uh, aggregation particle size. At the time, we used four foreign particles to check. First one, silicon oil, protein aggregates, rubber closure or air bubbles, and try if it can identify out of four particles. And CNN, this convolution neural network was better at identifying the particles. At the time, we fed the image samples. At the time, we used 5,000 samples to the machine to develop the model. And based on the morphological structures and 18 diameter parametric conditions, we designed a filter set and fed it to the machine learning our algorithms. And in-house, we developed these two configuration to detect if it's the cluster is protein aggregates or foreign particles based on our in-house models. So the condition was this one: first set for aggregation to find out. This one, second one is for the foreign particles and determine if the particles were detected or not. After the design of all the initial formulations, formulation scale-up, long-term stability, and detection of particles, we fed all the data to our system to design the digital twin model. It's a hypothetical replica of the real-world experiments to help up design if it can be developed in the real scale in biopharmaceutical areas and have a continuous manufacturing. So we fed the data to based on the this one is real time data sensing with the sensors and helping with the uh, this one. This one, smart sensing, making the using the use of the tools like in our case Encell and the Ignites and the Packworks device. For live legend aspect, we use the one wireless sensors and temperature samples, and another is depends of pressure gauge to develop this real-time monitoring using real models. So it was helpful in uh, small-scale experiments have optimized replicating to continuous manufacturing and large GMP standard model manufacturing at a low cost. And it was possible when we incorporated our own fed data to the digital twin models. It helped us reduce the waste of like ethanol and other
Um,
solvents.
excuse me, sir.
Yeah.
So it's a request. Can you please wind up with the lecture as we have the next speaker waiting?
It's almost finished.
Okay. Thank you, sir. Thank you.
Okay. This help us design the Pharma 4.0 based on our AML designs and the digital twin models and develop the sensors and uh, automations in the continuous manufacturing process. So the key conclusions from this discussion is we can simplify how much buffer or if it's needed for the lyophilized or the selection of stabilizers for making it stable during the solutions and during the manufacturing into the lyophilized forms and using AIML models to have reduce the number of experiments, having faster optimizations and predictable output response in big batch size with minimum errors. So this digital twin models, it's helpful for the real-time optimizations using the sensors, pads, and having a during the preparations. It will help us to make the last scale possible at the minimum time, and the output response can be predictable at the real-time scenarios. So this was the next one is about the future scopes, and this will help us to make it. The purpose of this study was to make it stable vaccine at room temperatures at reduced cost and make it sustainable, and supply chain is controllable. Uh, this is the last slide of my presentations. Uh, I'd be happy to answer any questions if you may have, and if you want, if you need to ask any questions in later, I can contact at this email address. You can contact me later as well. Thank you.
Thank you so much, sir, for such a wonderful lecture. Now we have a question answer round. Where we have uh, I'm just taking up the question from the audience, sir. So it is one of the audiences asked that what are the key challenges in implementing AI, ML, and digital twins in RNA LNP vaccine manufacturing?
One of the challenge is to have the development of model. It needs a lot of data to have the optimized conditions. So if we collected enough data in our model, like we can work on collaboration of 2 or 3 laboratories to generate the data. If we combine all data from these labs, then the model will be more have accurate and the prediction will be more accurate. So working on in collaboration will help to develop such models. So another challenging is the resource, computing resource. So we can use the cloud computing resources to design this, to fitting the annual model. Next one is we can have a little bit of training to implement in our biopharmaceutical settings. So it will help at that time.
Uh, Thank you so much, sir. One more question to take up, sir. What makes lipid nanoparticles more suitable for RNA delivery than viral vector in context of green nanomedicine?
In my aspect, when I did my study, I chose different models, different experimental settings. It was easy to make and cost effective, and the characterization aspects also it was easy in our laboratory settings. So I suggest I prefer for the more LNP based for the viral vaccine manufacturing because it was convenient and more easy to use in the pharmaceutical settings than others.
Okay, sir, the last question to take up. How might digital twins enable real-time monitoring and control of RNA LNP vaccine production?
In our case, we used in the lyophilization aspects, and we use the temperature wireless sensors inserted in all the files. And second one is the gauge that we used for using the machines. So if we have the developed sensors, it doesn't matter wireless or wired, and connected to the real-time sensing to desktop, then it's possible to have the data at the real time and control from the central computers.
I hope the queries have been satisfied by the speaker. Now, may I now request our director, Professor Anuradha Mishra ma'am, to please say a few words of wisdom for sir.
Thank you, Jishan ma'am. I would like to appreciate Dr. Mahajan for this insightful lecture. Altogether, our delegates as well as students and faculty members have learned a lot about the AI and ML applications. So, as a token of appreciations, I would like to request my IT team to kindly share the certificate. Thank you, sir, for joining us.
Thank you.
Kindly accept this.
Thank you, Dr. Ravi.
Thank you. Thank you, Professor.
May I now take this opportunity to invite my co-host, Dr. Swati Prakash, to take over for the next session. Dr. Swati? Dr. Swati, you're not audible. Dr. Swati, we cannot hear you. कैमरे को नहीं खोलो.
Swati, you can rejoin. Leave and rejoin.
Till the time Dr. Swati joins, we can just have a break for 5 minutes. Audience, be there.
Yeah. So we welcome Dr. Sudarshan Singh. I think he joined.
Yes, sir. Good afternoon.
Sir, please switch on your camera so that, yes, yes, yes. Thank you, sir, for accepting our request.
Good morning, sir.
Good afternoon.
It is good morning, sir, here.
Yeah,
Good morning and welcome to, sir, this virtual platform on behalf of Amity Institute of Pharmacy, Amity University, Uttar Pradesh, Lucknow campus.
Thank you for inviting me.
Sir, am I audible now?
Yeah.
Hello?
Yes, you are
Yes,
audible.
Okay.
Swati, you
Okay,
are
sir.
audible.
Okay, ma'am. Thank you, ma'am. Sir, can we begin, ma'am? Can we start now?
Swati, please.
Okay, respected dignitaries, distinguished speakers, faculty members, researchers, and dear delegates, a very good morning to everyone. It is my pleasure to welcome you all to the Technical Session 7 on the day 2 of the 3 days International Conference on Green NanoCon, Pioneering Sustainable Nanotechnology and Chemistry, organized by the Amity Institute of Pharmacy, Amity University, Uttar Pradesh, Lucknow campus, from 16 to 18 February 2026 in the online mode. This platform highlights the growing need to integrate environmental responsibility with the scientific innovation. It emphasizes the development of eco-friendly nanomaterials, green synthesis methods, and sustainable chemical processes that may minimize the environmental impact. The conference aims to promote research that balances technological advancement with the ecological preservation for a healthier and more sustainable future. I would like to extend my heartfelt gratitude to our esteemed speaker, Dr. Sudarshan Singh sir, our HY ma'am, Professor Dr. Anuradha Mishra ma'am, all the faculty members and the participants for their gracious presence and valuable time throughout this 3 days international conference. I would also like to warmly congratulate the organizing committee for successfully planning and conducting this conference and for creating such a meaningful platform for knowledge sharing and the scientific interaction. It is now my privilege to introduce our distinguished speaker, Dr. Sudarshan Singh Sir, Frontier Proactive Research Scholar at the Faculty of Pharmacy, Chiang Mai University, Thailand. Sir has made extensive contributions to pharmaceutical education and research. He is currently working as Frontier Proactive Research Scholar at Chiang Mai University in Thailand. He has authored more than 10 technical textbooks for pharmacy graduate students based on the syllabus of PCI and Savitri Bai Phule University, Mumbai, India. He has also edited 15 professional books under the following imprints: Apple Academic Press, Springer Nature, De Ruyter, Elsevier, and Bentham Science, and has published more than 100 peer-reviewed book chapters. He has also published more than 300 research and review papers in high-reputed peer-reviewed journals. He has presented numerous papers and participated at both national and international conferences as a keynote speaker. He has also been ratified and approved by the Pacific University in Rajasthan to supervise the PhD students. He has registered 5 patents and an active editorial board member of Science Domain International, India, UK, and USA. He also serves as a reviewer for several international journals. He has 11 years of teaching experience in both undergraduate and postgraduate courses on pharmacy and 8 years of research and development experience. He has guided several UG and PG students and has also co-supervised 4 doctoral students during his postdoctoral research at the Prince of Songkhla University and Walai Lak University in Thailand. He is a life member of several professional bodies, including the Society of Pharmacovigilance India and Registered Pharmacy at the PCI. He has received several grants from the Government of Gujarat and Savitri Bai Phule University, Maharashtra. He has also received travel grants from the ICMR and the Center for International Cooperation in Science, Tamil Nadu, India. His current research focuses on the use of additive manufacturing, that is 3D imprinting techniques, to develop patient-centric implantable scaffold materials that are fortified with the active compounds or with polymeric biomaterials grafted bioactive compounds, which are superficially aimed at counteracting the dermal inflammation and development of engineered multifunctional natural-based biomaterials for the drug delivery applications. So today he will be presenting his work on biogenic metallic nanoparticles incorporated into biodegradable wound dressings that highlights their, highlighting their antimicrobial, anti-inflammatory, and wound healing potential. We are truly honored to have you here, sir, with us today. I hope the session to be enlightening and enriching for all the delegates. I also encourage the attendees to embrace this opportunity and ask the questions at the end of the session, and they may put their queries in the chat box as well. That will be taken at the end of the session. So on behalf of AIP, AUPL, I warmly welcome you, sir. And sir, it's over to you now.
Thank you, Professor Swati.
Thank you.
You have given a very broad introduction. Thank you so much for that. I would like to first thank Dr. Abhishek Tiwari, sir, who has given me opportunity to speak on one of my research what we have done previously, and that research has been vastly developed and currently in market for treating veterinary animal, that is pet animals, that those who are getting infected. So before we start, let me share the screen and then we will proceed. So just let me check, the screen is visible.
Yes, sir, it's visible.
So we can proceed, right? I will share here my sum of view. This work was led by me and under the supervision of my postdoctoral supervisor, Professor Supayang, who is a leading scientist in antimicrobial product development. She is currently working as Director for Center of Excellence related to natural product and Southeast Asian Center of Excellence for Antimicrobial Resistance. So we have been working on several products based on theme of this current green NanoCon. This was selected. Now, this is a very important point in today's era, that we are getting day by day resistance to various pathogens, and we need to find out something which can still show efficacy in terms of antimicrobial, and still it can be used. Though developing nations are not adapting metallic nanoparticle, mostly in the form of reduced that can be used as antimicrobial or not. But still, we are adopting slowly by slowly, and this is the one of the product patent which has been done here in Thailand, and we are using for treating the veterinary animal. It has been clinically tested on veterinary animals, so directly it was placed in the market for vet product. That is, all the veterinary pet animals visiting to veterinary hospital or Prince of Songkhla University, they are treated using this developed product. Now some technology was incorporated in this that we will see slowly by slowly. But before that, we will see what is this. And that is, several types of synthesis methods are available for metallic nanoparticle. Like everyone know, physical methods are there, then thermomechanical methods are there, chemically synthesized methods are there. All have their own advantage and they have some disadvantage too. Green synthesis of nanoparticle is nothing new. It has been bought almost 10 years before when we were started working on this synthesis in our lab. But I think it is more than a decade old when the concept has been bought for the synthesis of metallic nanoparticle using green extract or plant material that can reduce the metallic form of organic material or inorganic material to a specific reduced form, and then that reduced form can be used in for various purpose. Like some of the metallic nanoparticles are even used for biosensors. They can be coated over the nanowires and they can be tested in the form of sensors. Here we developed for checking it as antimicrobial efficacy. So we can see on the first slide that how the metallic nanoparticle was developed using eucalyptus extract. Now selection of eucalyptus itself is an important aspect. When paper industry comes, the eucalyptus is first choice of for the use of its pulp and other parts of the plant, while the leaves are wasted. So it has been considered that we need to use that waste and valorize in the form of component that can reduce the metal to its nanoparticle form, that is reduced form, and then we can test it for antibacterial properties. So before we go in depth of this research, we have done several aspects related to this metallic nanoparticle. Just we will go one by one, and then we will try to understand the basic work what has been done. So we are discussing here two things based on my research what has been done, that is metallic nanoparticle and use of eucalyptus, that is the waste leaf which is generated from the paper industry. So first is the metallic nanoparticle, where we have transferred that metallic nanoparticle in polymeric matrix and then we printed in the form of film and we used it in the form of antimicrobial on-site degradable wound dressing. So on-site degradable wound dressing, the concept was bought when we want to go for dressing again, we need to clean everything. So the concept was here on-site degradable is the wound dressing should be degraded with the pus which has been formed, and just we need to remove it with the help of drying the area of the wounded portion. Now, same metallic nanoparticle was transferred to gel form and we checked again the efficacy. So first was the composite we printed and the second one was conventional carbopol gel we prepared, and we checked the efficacy, whether it is showing the efficacy in the form of antimicrobial or not, and we found that it can retard the growth of all the tested microorganism, that is both gram positive and gram negative. Similarly, how it can do in its a reduced form without incorporating in any drug delivery system. Then we little modified that drug delivery system. We transferred that metallic nanoparticle in thermosensitive gel. Some of the disease like candidiasis which are happening in the specific portion of the body. There we require insertion of the gel, which should be transferred in the form of insertion of fluid, and which can be transferred in the form of gel when it's come in the contact of body temperature. So here again, we tried and we found that when we incorporate in the polyxomer based formulation, it can give a good transformation without affecting sol to gel formation, and the relative phenomena, that is, it can reduce the biofilm, it can improve the inflammatory responses, as well as it can show good reduction in the cytokinins when we tested for pro-inflammatory cytokinins. Now that was we tested for bacteria. Later we checked those green metallic nanoparticle for fungal, that is candidiasis. Sometimes we need to pay attention when it is happen, and we have again restricted number of medicine, and those medicines are also showing, those antibiotics are also showing resistance. So this is an image we can see where on the left hand side is showing TEM of the Candida albicans treated with silver nanoparticle, and on the right hand side we can see that is the SEM, scan electron microscopy of the treated Candida. So what we can see is that when it was treated with silver nanoparticle, the basic structure was rupture, and it is showing that itself the fungus was not able to propagate, or the colony was not propagating, and we found that the metallic nanoparticle are still active in the against the candida. That both figures are showing that when it comes to colony forming unit, as well as when it comes to structural integrity, both has been completely destructed by the metallic nanoparticle when it was treated. And we can see in the both image, that is in TEM and SEM, the silver nanoparticle at treated concentration showed an effective efficacy. Now, similarly, we try to check against on A bomb, that is from ATCC. Then we check for E. coli, we check for Staphylococcus aureus, and again for Candida. And we found that when they were treated against the MIC of the silver nanoparticle and MBC of silver nanoparticle, that is MIC is minimum inhibitory concentration, was checked, and we treated those metallic nanoparticle to specific bugs, and we found that their structure was denaturating, and then the broken structure itself showing that when it is treated at two different concentration, that is at MIC and MBC, it was an quite good effective. So this what we tried to perform over the various pathogen, those are related to infections. There are another kind of efficacy that we found, that when we go for post-operative or pre-operative procedure, in both case we need to very careful about infections. In case of post-operative procedures, there will be definitely incisions with the help of sutures. Now, alone sutures are not capable of showing antimicrobial efficacy, so we need to give some external medication with an antibiotics to control the infections. So in this case, we took two different types of suture. First is silk braided suture, and second one we will see in the next slide, that is nylon suture. Now, silk sutures was tested because it can absorb easily the metallic nanoparticle. So first what we did is we externally synthesize the metallic nanoparticle and deposit it over the sutures. And in second case, we did in situ deposition That is directly we took the sutures and we added the phenolic rich extract as well as the silver nitrate, and we try to reduce there only itself. Now in both case, we need to check the efficacy. So we tested the efficacy of both sutures against the bacteria, and you can see the antibacterial efficacy is showing almost 99% inhibition in bacterial growth with good log of reduction in bacteria when it comes to coated suture compared to uncoated. So this is a part what we found, that antibacterial efficacy can be obtained if we coat the sutures. By the time, we have to be also careful that it should not affect the healthy cells, that is the carotenoid cells or fibroblast cells. So we checked the cell proliferation efficacy in presence of sutures, whether it is showing any effect on those cells or not. So we tested those things and we found that now this was done a bit indirect method. That is directly it's not possible to check because of several factors. Contamination can happen. So we took the sutures and sterilized these sutures using specific gas sterilization, and then we transfer it to the sterile complete media, and the eluents what has came out was directly added to the 6-well plate where cell were already seeded and scratched. So we can see on the right hand side of the figures that cell were migrating properly, though it was bit less than untreated cell, but we cannot find out any significant effect that when it's affecting the bacteria, it's also showing the effect on normal cell or not. So it was not showing significant effect on normal cell. Normal cell were propagating as usual and they were showing the growth. So this was confirmed from here that suture which has been coated can be used, and this yet not had been bought in the market, but already patent has been granted on this. So from here we went for in vivo studies. We made an incision. It's not an incision which happened because of some injuries. It was an intentionally made incision over the chicks of the mice, and then there we stitch this coated sutures. So we tried to find out whether these skins are normal or not, and we found that on performing HNE staining, that skins were doing well. It's not like when it's affecting the bacteria, skins are showing some effect or not. Well, only the things what we didn't check, but it has been reported clinically that silver nanoparticle cannot infiltrate the kidney and it will store in the kidney for longer time, and then it can cause loss of working for the kidney. But we have not checked all those things because already several clinical trials has been shown on animals that it can happen. Then we further checked on a bomb, that is we tried to check the silver nanoparticle whether this metallic nanoparticle which is related to OMPA specific protein of a bomb can reduce their intensity or the way mechanistic way by which it can cause the further progression of the disease. And we found on the right hand side, you can see that OMPA protein, the density has been reduced and it can be seen against the various tested A bomb from the strain which has been obtained from the culture which was provided by Prince of Songkhla University hospitals, and then we concluded that yes, it can show efficacy there too. Now it comes to next, we coated the catheter, which is again an instrument which is going to be inserted inside the body for specific diagnostic purpose as well as the therapeutic purpose. So we coated that catheter and we tried to find out whether it is effective or not. So you can see the atomic force microscopic image with the left hand side blue color is showing and specific uncoated, as well as the green is showing the coated one, and this can be directly related with the SEM image where we can see the small nanoparticle has been deposited on the surface of catheters. So after depositing over the catheter, it form non-transparent catheter. It become black because the process was layer by layer deposition of silver nanoparticle, and then we went to check the efficacy against the A bomb again, and we found in the Petri plate you can see that it is showing the inhibition of the growth of bacteria, as well as when we went to check the specific structure for against the other bacteria, that is S. aureus, E. coli, and Pseudomonas, as well as Candida. Everywhere it has shown that the different types of pathogen are still inactive against the coated catheters, and this can be useful. Like there is no need of giving additional antibiotics when such types of catheters are used. Then again, we checked metallic nanoparticle on gloves. So this all what we have tested, we have related somehow it is affecting the patient, and from patient to patient transmission. That is, when a nurse use the gloves, how transmission can happen. So you can see from the figure on right hand side, figure A, B, C, first is showing imprint, then stamp 1, stamp 2, and stamp 3. So this is, we try to mimic the process. We coated the gloves with metallic nanoparticle, and then simultaneously we prepared different bacteria grown on the plate. So first one is the imprint, that is the gloves was directly pasted over the already grown bacteria. Then from that first gloves and second gloves was bought in the contact of first gloves, and then it was pasted over the plate which was new treated but no bacteria was grown. Then from the first stamp, second stamp was made similarly, and then third stamp. So we can see when it comes to coated one, silver nanoparticle coated, the growth rate was reduced from stamp imprint to stamp 1 and totally reduced to stamp 2 and stamp 3. That means there was no transmission. But when we checked on the uncoated one, we found that still there wasn't transmission for both gram positive and gram negative, as well as the candida which was tested. Now we little move with the previous work on A bomb that was tested for A bomb. Here you can see in the right hand side that A bomb were stained with DAPI, and then we try to check whether the structural change on the nucleus is happening or not. So zoom based image is showing that the structure has been already broken, and because of that the blurred color is coming from those A bomb which has been stained with dye. So this all what we have done on the medicated, that is the product which are related to health. Further, we tested this on food contact surface. That is, nowadays most of cities are making ready to eat or processed food. Now this food contain a lot of sodiums. Even if it is having a preservative, still it can show you contaminations, and we need to stop it. Because of this fast life and people don't have time, they are directly going and buying the packed food and trying to eat. immediately so they can save the time. So what we did is we prepare the metallic nanoparticle reduced using eucalyptus leaf extract and transferred to the edible film. That the film was made using chitosan. So you can see 4 different A, B, C, D on day 3, sorry, on day 3, day 7 and day 15. So the bottom one is showing the edible film which was not added with metallic nanoparticle. Then the different ratio between mid one and the top level of the packed food. So this food was ready to eat sausage. Sausage was coated and it was stored at 37 degrees Celsius, not at freezed refrigerated conditions. So on day 15, the uncoated has shown that molds has been already grown and the food is contaminated, no more useful. But when we come and see on the the different aspect, that on day 15 with the metallic nanoparticle, there was no growth. That indicates that it can enhance the shelf life. Now, this was tested against the food, but we need to check whether the added metallic nanoparticle in the film, when it's elute, it's going to affect specific cell that is related to kidney or related to colon cell or not. So we tested the efficacy, that is biocompatibility, and we found that dose dependent cytotoxicity is there. So at specific dose only this can be added as a food preservative, or else it is going to cause effect over normal cells. This was further transformed to the different form, that is here we valorize the waste paper, the copy shop machines as well as the other copiers. They produce a lot of waste of papers. So the eating tray, food eating trays are available, that is ready to eat over the plates and throw it again. So we try to mimic those type of plates by taking the waste paper and added some amount of starch, and with the help of specific mold we prepared similar type of plates, and then we kept over the food, ready to eat food. And then we try to check here two things. First thing, the plate that was made leach some quantity of metallic nanoparticle or not to the food. And second thing is whether it is effective or not. So it was leaching, but up to an extent which can be safe while it is showing efficacy. So we are not sure exactly about related to food and drug administration whether this is acceptable or not, but it is showing efficacy in terms of extending the that is shelf life of those foods. So this what we have, you have seen what we have done related to metallic nanoparticle specific to eucalyptus leaf extract. Those all project were done using synthesis of metallic nanoparticle, that is using eucalyptus leaf extract and synthesize the metallic nanoparticles, and then we tested on the various aspects. This is just a short part what we have seen. Now we will come to our basic talk what I have selected to prepare a wound dressing that can degraded on site, and then it can show efficacy also. So this is an abstract, I am not going to read. We will try to understand what is happening in the study. So this is an usual process. You can find out in any laboratory where extract can be prepared and specific content of the extract can be treated with specific amount of nitrate, and then depending on your process, it may involve heating, it may not involve heating, it may require incubation in dark, it may not require incubation. It depends on the process, how you are optimizing. But the basic phenomena is when we are going to green synthesis, we avoid heating, we don't go for heating, but we have to go for incubation in dark to make sure that all the nitrates has been completely reduced. If there is unreduced forms are available, that has to be removed out or washed out. So after incubation, we can collect those metallic nanoparticle. Now, whole process is very important to be performed using DI water. If you are trying to use mineral water, and the process will be not 100%. It may show sometime complications also, that metallic nanoparticle is not reducing. And you will find out those things is when you go to check the peak over the spectromax. That is when you try to go and check the lambda max is showing in the spectra or not. So using DI water, we can prepare, and then we can collect the sediment and resuspend again in DI water. That is 3 to 4 times washing is required to remove all the unreacted, unreduced form of material which is available. Either it may be plant extract or it may be nitrates. So everything has to be removed. And then it depends again whether you require sonication or not. Now, sonication step has been shown here to make sure that your nanoparticle should be at nanometer in nanoscale, and it should not be showing on coalescence. If you know already zeta potential is very high, there are less chance, but it is not exactly recommended to do sonication, but it depends on availability. If you want, can be done, or if it is not required, there is no need to be done. Then drying and is the process. If we want to perform some different category of fabrications, like if we want to increase the stability of those nanoparticle, we can freeze dry them, and then we can store it for longer time. But here again, we have to be very careful when we are going to reconstitute them. We need to reconstitute again in DI water only. By mistake, if it has been done in different mineral based water, then it will be in the suspended form. It is not going to be exactly show redispersed form. So this is the process what is generally used for green synthesis. Apart from this, radiation based, thermal based, physical based, several methods are available. Even chemical based, polymeric based. There are a lot of methods are available that can be used for reducing the metal to metallic nanoparticle. Now, first thing what we do is we need to check lambda max. So it should show a peak something around 380 to 420. If it is showing peak, then it is the nanoparticle has been prepared. Sharp peak, the particles are in good nanoscale. Curved peak, they are showing the particles are not good in nano shape. They may be coalescence or might be reduction has not been completed. Now, maybe if we need to further clarify, I would like to inform that this nanoparticle is different from polymeric nanoparticle. This is just a reduction of metal to an nanoform, that is in reduced form, which is called as metallic nanoparticle. Polymeric nanoparticle we prepare in our pharmaceuticals, that is based on different polymer. We incorporate, we use different method, and we will form a polymeric matrix, and inside the matrix or inside the core structure, there will be entrapment of drug. So here nothing is like that. This is just a reduction of metal, what we can see in the form of metallic nanoparticle. So we perform various characterization. You can see all the different types of characterization we are performed. On the right hand side, we can see particle size analysis, which is showing a specific curved like structure on the scale that shows that a specific range of particles were formed when it was reduced, and those can be again reconfirmed when we go and see the TEM image where specific spherical shape of metallic nanoparticle has been formed. Now technology has been far and far developed. We can prepare metallic nanoparticle of cube-like structure, like various structure we can form. We can prepare even flower-like structure that will mimic the structure of flower, and we can call them as metallic nanoflower. Now, that metallic nanoflower are not restricted to silver. They can be even prepared for gold, for zinc, and for others. So technology, that's why I said in the beginning that this is not an start of metallic nanoparticle. A decade has been already over and lot of advancement has been done. So after characterization, we went to directly formulation. We didn't went to check anything because as I have shown in the previous project, we already checked the MIC, we already checked the MBC, that is minimum inhibitory concentration, then bactericidal concentration. We checked the time kill assay and we know already everything. So we don't need to go and perform again those same thing. So we directly went to prepare a composition. So the composition, what you can see is already optimized. And then we took the composition and we went to, we went to check the color analysis, because when we prepare something which is going to be applied topically, it should be elegant, like it should be acceptable. If it is something like black in color or dark black, brownish color, sometimes patients does not accept those things. So we went and checked the color parameter and we found that it is lightish yellow in color that you have seen in the first slide. Maybe later we can go and see. Light brownish to yellowish color when after incorporating inside the film. So we then went to print it. So printing is again now nothing new. Just we need to work out on two things. That is, we need to know the rheology phenomena. If we understand, if we have good understanding of the fluid mechanics, there is nothing need to worried about 3D printing. In case of gel printer, if we are using different printer like like FDM or laser based, then we have to understand those things. But the gel printer is the economic one and it can be used. Only the thing we have to understand is we need to know what is the fluid mechanics of the gel, what we are going to print. So we checked the viscosity and other things, and then we went to print it. So using a Tinkercad software, first we prepare the design and how many layer we want to print. We exported that from CAD software to the 3D printer, and we need to just print it. After printing at the room temperature, it was dried and then it is ready to use. So apart from printing, we went to check something that is, we went to check some common aspects that we need to know. That is functional and structural analysis. We need to know whether it is interfering with polymer or not. If it is interfering, then how? So we need to check those things. So we can see from the bottom to top, that is the FTR of extract, then metallic nanoparticle, then incorporated in three different polymeric material. That is Xg is representing xanthan gum, HPMC is the hypomoles, and then sodium CMC. So we can see the peak which is related to OH was still in the dome shape. It's not changing or not disappearing, as well as the sharp peak at 1358 was little bit going on like broadening and disappearing. So that indicate that metals was incorporated in the matrix of polymer. That's why the peak was little disappearing, but not completely disappearing. And the sharp nature was lost. So that confirmed that metallic nanoparticle was incorporated within the matrix of the polymer selected. Now we choose here three polymeric complex, that is xanthan gum, then HPMC and sodium CMC. Among these three, we selected only one based on antimicrobial time kill analysis. So we will see later, but we will go little faster. I don't know if have much time or not. So when we prepare metallic nanoparticle, that nanoparticle can be further quantified using Burke's diffraction law. So when it is prepared, it should be highly crystalline in nature. That is the lyophilized form. You can see it is showing good peaks, and this all the numbers are coming from the that Burke's diffraction law. The CDD values can be directly propagated from the software, and it can be indicated here. So you can see on the top of the image that when it was incorporated inside the metallic nano, that is the polymeric matrix, the crystallinity was reduced almost to 50%, which is because it has been entrapped inside the polymeric matrix. So this is what the Burke's diffraction calculations, which is showing that how we can calculate the crystallinity, and we can further calculate the size of the crystallites. So the size you can see for SNPS, that is the metallic nanoparticle, matches with the TEM what we have performed and the particle size analysis via dynamic light scattering techniques. So the particle nature was still good even in lyophilized form, as well as when it was incorporated inside the polymeric matrix. Raman spectra is not usual when we do sensing analysis, then it's play a good role. But here we try to find out whether the spectra is still matching or not, and we find that the spectra is matching with the lyophilized metallic nanoparticle and compared to the metallic nanoparticle incorporated inside the polymeric matrix. So after incorporating inside the polymeric matrix, we went to perform FESEM based on EDX. So EDX analysis was performed to quantify the metallic nanoparticle inside the matrix. So as I differentiated, the polymeric nanoparticle is different from the metallic nanoparticle. In polymeric nanoparticle, we know the practical yield and theoretical point of view what we have added, but here we don't know what is the exact quantity is available inside the polymeric matrix. So we follow EDX and we follow ICP. So we can know in specific length of composite what is the content of metallic nanoparticles. So first we perform EDX based analysis, and we found that the metallic, that is you can see in the figure, is it showing presence of various metals as well as the silver in reduced form, that is iron, and that make confirmatory that the particle which is present inside the composite are metallic nanoparticle. So since we made a 3D printing, the printed surface of the composite should be not much rough, and it should not be too much smooth. Too much smooth means it should have some porous nature to make the passage of air when it is going to applied over the wound. Not much rough means it should be printed properly, not like somewhere bubble has formed or somewhere printing was lost and pore has been left there. So we check the uh, surface roughness using atomic force microscopy, and you can see the SA is indicating surface roughness, and the peaks and heights were indicated on the 3D, that is X and Y axis via atomic force microscopy. And we found that this have smooth structure, but we need to know the porosity. So we perform, you will see the results of porosity later. We perform the porosity too. Now, when we prepare some material which is going to be applied over the wound, it should be adhere over. So we check the hydrophilicity using contact angle analysis, and we found that they are good in nature, and the best one what we have optimized in the latter case, that is SPMC based film incorporated with, showed 44.60 degree when it comes to hydrophilicity. They showed good hydrophilicity, like they can adhere over the wound. It's not like it will show anti-adherent nature. Then we check the physico-mechanical property. We check porosity, as I said. So we found the porosity was higher among all tested composite was with HPMC. We find solubility because all the polymer are soluble and biodegradable, we can say, because sodium as well as cellulose based and xanthan are from nature derivative from nature, and they can be easily solubilized in water. That's why we have given the name that is degradable. So we check the solubility, we check the swelling index, then tensile strength, as well as the related mechanic property, that is Young's modulus and elongation at break. And we found that except the tensile strength, all the tested parameters were perfect for HPMC based composite. Then the further supportive we did was we checked the optical property, that is all physico-mechanic property. We check moisture content as well as moisture uptake, then this vapor transmission, and the thickness. So we found from here that because optimization was not done on the basis of all this physico-mechanic analysis, optimization was done based on the time kill assay. But still, when we compare the HPMC incorporated metallic nanoparticle composite with other, we found that it was not much differing. So we checked the further is scavenging assay as well as nitric oxide. So when we come to talk about wound healing or wound dressing, these two are most important concept that we need to perform, that is whether ROS based any mechanism is there or not. So this is a very primary test. ROS based cannot be directly detected via a scavenging assay. We need to perform ROS based specific experiment, but we checked the DPPH and ABTS, and we found that when it comes to alone composite without incorporated metallic nanoparticle, it can show good scavenging assay, as well as when it comes to reduction in nitric oxide, it can show good inhibitions. That is, it can retard all the cytokine, because we already tested the pro-inflammatory cytokinin in previous test. So we didn't went to perform cyto-inflammatory all the tests, but we checked only very basic, and we found that both tests were okay. Now, when we comes to wound dressing, another important thing we need to know is clotting, whether blood clotting is at appropriate time or not. So we check the blood clotting at 2 time duration, that is 5 minutes and 10 minutes. We took the blood from specific animal, and then we perform the well-defined assay, and we found that the metallic incorporated are showing good clotting efficacy at a reduced time compared to control composite. Now, this is the test where we optimize which composite is perfectly suit for the further testing on animal. So we perform time kill assay. Now this time kill assay was indirectly performed, since metallic nanoparticle was already transformed inside the composite. Before going to print the composite, we took the specific quantity of composite and we dissolve in the media where we dilute the different other thing. That is, when we perform the MIC and MBC, we have serial dilution. So we took here DW water, that is sterile DW water, and we tried to dilute the polymeric incorporated metallic nanoparticle solutions, and we tested the we performed the time kill analysis. So you can see we perform against the basic pathogen which are generally available when we talk about or when we want to treat bacterial infected wounds, that is A-bomb, E. coli, Klebsiella, Pseudomonas, and that is again Staphylococcus aureus, then epidermitis and Candida. So from here we found that the hypermolars composite was far better than xanthan gum incorporated as well as the sodium CMC incorporated. From here, then hypermolars was considered as an optimized composition for further test. So after performing that analysis, we went to check the biocompatibility on human skin cell. That is a different cell you can see, HYCAT cell, fibroblast cell. And since we already performed the nitric oxide on macrophage, the results are here against the macrophage also. So we found that the specific tested portion of metallic nanoparticle incorporated composite were good in compatibility use. Only you can see on the figure that is xanthan gum based has shown viability less than 90, but still more than 80% are considered as biocompatible and thus it can be used. Since we performed previously EDX as well as we perform ICP, and here we don't know exactly what is the quantity of metallic nanoparticle present. So we tested those things and then based on those things we perform the diffusion. So when we apply the wound dressing over the wound, we need to know what is the quantity it's going to be released. So after 10 hours of release study, we found that almost 1.4 microgram per milliliter was released. This was correlated with MIC and MBC, the test we have performed in various studies where we try to check the structure of bacteria are retaining or breaking. So those studies were performed at specific MIC, half MIC and 1/4 MIC, which relate to this content what has been released. So before going to animal study, we tested cell migration assay. That is, we tested similar where the sutures were coated and we tested here also. And you can see like cell alone where no treatment was done and the treatment has been done with composite elutes are not showing significant inhibition. Like cell are dense, cell are not scattered. So it shows that it's not much affecting the normal cells, that is tested HYCAT cell. But when we compare in the form of graph, we found that the HPMC based showed 40% migration during the tested hour, which is much higher than the other two composites. So considering all these results, we went for animal studies. Now this wound you can see is not wound created by a doctor. This all animal who are visiting in the veterinary hospital, there we applied. So it was a random trial, but we have kept all the animal in sequence ways. Like the first animal which was treated, it should be continuously treated for next 30 days. And on the last day, the same wound was calculated. That is, what is the shape and area of wound on the 0 day and what is the shape and area of the wound on the 31st day. So this was done on 30 animal, but at the end of experiment, only half of the animal were still visiting the hospital. So we consider only on those 15 animals and we optimize and presented the result, the best what has been obtained. So you can see test dressing has shown almost in the form of wound area and in the form of wound length up to just 10 that is of wound was left on 31st days. But when it comes to control dressing where no metallic nanoparticle was a bit higher, so it defined that the tested material were effective. It was not effective against the normal cell, so it is not showing cytotoxicity against the normal cell, while it was showing efficacy against all the bacterial infections. So what I have not presented here is at every visiting of animal, we took the swab from the wound and we try to check the pathogens, the probable pathogen which is available. So animal were visiting was some from accidental wound, some were visiting because of some different case. So when it comes to wound, all wound don't have similar kind of pathogens. So we have to make record of that, and all those records are very necessary when it comes to filing a patent. Those records are confidential and that's why it cannot be presented here. But we tested those things also and we found that yes, all the pathogen which was present at the day 0 has significantly reduced, and when it comes to the end of the treatment, it was completely vanished. So it was a specific test. We need to swab it and then again go and perform the microbiological assay. And then depending, because we are pharmacists, we are not a pure microbiologist. So microbiologists can identify from the color only which type of pathogens is available there. So that has been done, and then finally we concluded that dose dependent form of metallic nanoparticle can show an efficacy in the form of wound healing, and not only in the form of wound healing, since we have tested on the various aspects also. That is, we tested on sutures, we tested on catheter, we tested on gloves, like transmission from where can happen, we tested on all, and we found that this can be used in the form of dose dependent form. It is not like that we can use it at higher dose like antibiotics getting resistance. Higher dose will not filtrate from the kidney and it will retain in the kidney, which has been already proved. So again, it is having some advantage while it is having some disadvantage too. But still, let's hope that it can show some positives that it can bought in the market. Already in some country, they are making it available in the market, but they are not making very clear in the media that we are applying such kinds of, like when we perform the animal study, we bought the similar patch from the China and we try to compare it. So they have the product, such type of wound dressing incorporated with metallic nanoparticle, but it's not, we know everything that is available or not available. Let's hope that the resistance what we are seeing day by day can give some hopes in the form of metallic nanoparticle. If it is going well, maybe we will see in the market soon. So this is what a small portion of work what I have presented here. We are doing a lot in drug delivery too. At last, I would like to thank the team and organizer of MIT, that is who has given me opportunity, and I am very thankful to Dr. Abhishek, because we have not met yet, but we are doing a good communication, and hopefully in futures, maybe we can do good collaboration too, and we will bring up with some good research work. Thank you.
Thank you so much, sir. Your presentation was really exceptional, enlightening, elaborative, and actually in this, when the cases of antimicrobial resistance are on rise, So we also wish you luck and hope that our students could learn from such an enlightening session, sir. And the session is open for the queries now. Sir, some participants have raised some questions.
Yes, sure.
Sir, there's a question that can we minimize the use of preservatives by using this type of films?
Preservative is different thing, and this is antibiotic, which is something
Sir,
acting as
actually,
an
in
antibiotic.
yes, sir. In one of your slides, you mentioned that the preservative, you checked the efficacy with this film that the
Like we
preservatives.
tested the food packaging, you can see here, we can claim that preservative can be removed and metallic nanoparticle can be used, but FDA does not accept that. No FDA accept that metallic nanoparticle can be consumed. So I will even say that it cannot replace the preservative. Yes, it can be an antibiotic, but it cannot replace the preservative.
Sir, can this film be used for the biomaterials like DNA or the vaccines?
Yes.
Okay. Sir, there's one more question that is there any specific criteria for the development of silver nanoparticles with the plant extract?
Uh, Plant extract must be rich in phenolic or flavonoid content, so it can reduce. And specific criteria is the solution where we are going to do reduction should be deionized. If it is not deionized, there is a chance of not complete reduction. There is a chance of highly presence of unreducted metals. So those will still show change in color from specific brownish, dark brownish to yellowish color. But these two things are very important. And apart from this thing, like people have tested so many things, even we have tested varying the pH, but varying pH does not make any sense because we are using a specific DI water. Varying temperature, if your extract is enough in phenolic or bioactive constituent, there is no need of temperature. It will reduce it immediately. But some plant may have lower content of those active constituent. Then in that case, heating is required. Now heating need to be optimized in those cases. If we don't optimize, then again we will have a problem that 100% reduction is not coming, and it will directly reflect the cost. When we go and convert in the form of cost, then it will affect. Then apart from this, the content of nitrate, what we are going to reduce, we need to optimize based on specific ratio. We need to optimize that. It's not like that what I have reported, same will be going to be reduced when you use different plant material. So that need to be again optimized. And again, content of plant extract. It is not necessary that what I have used content or quantity will work for you. You need to optimize. Now see, optimize means that plant contact may vary. Like I will give you an illustration. When we work with eucalyptus, around 1 milligram is enough to reduce a specific molar of nitrate. But when we use guava leaf extract, when it's come in contact with DI water, they change the pH of DI water. And then we feel that reduction has happened because color might slightly change. But when it comes to mechanistic inside, guava leaf have reducing capacity lower than the eucalyptus because of its own presence of bioactive compound, which is changing the pH of solution where we are performing. Now this thing cannot be immediately visualized, but when we store, now nanoparticle, if we are talking about lipid, then the basic what we need to understand is stability. So stability means we need to go and store somewhere and then we need to observe. So after observing sometime, we will feel the difference. We will see the difference. And because of that, things needs to be optimized, but it has to be understand very carefully what to optimize. It's not like you are optimizing and you are using demineralized water in place of deionized water. So nothing is going to work. That we need to understand.
Okay, sir. Thank you, sir. So there's one more question that what are the research constraints of the silver nanoparticles?
So we have a basic problem, what I will share here. If you are working with silver nanoparticle and working with hand without gloves, if you are not wearing gloves and working on the synthesis of silver nanoparticle with any method like physical, chemical or green, any method, the silver will deposit on the skin, and slowly by day it's going to be penetrate, diffuse inside the skin, and it will go and retain in the kidney. This is the first and foremost thing need to understand anyone who is working on metal. Then second thing, if we are working on metallic nanoparticle, we have to be very careful with the utility, like the glassware. If we are using the glassware and there if you are synthesizing, you need to wash it 10 times if you want to use same glassware for different purpose. Still the traces of metals will be available there. Like an illustration, I prepared a metallic nanoparticle on 100 ml of volumetric flask. I thought that, okay, twice I rinsed, everything gone. And then I took sterile DI water and then went for dilution while I am performing the MIC. The traces of metallic nanoparticle will affect my results. So like this, there are many more things, but these two are very important that we need to understand while we work with metallic nanoparticle.
Okay, sir. Thank you. So, sir, one more question I would like to ask that what are the key challenges in translating the nanoparticle-based dressings from laboratory to clinical or the veterinary purposes?
When it comes to application, people are not accepting. When we say we have to fill the form from the owner of pet animals, or when we go and give to patients, we need to take their consent. So they don't accept. They say that this is something new and we don't want to accept. That is the first and foremost problem with the metallic nanoparticle. If you will say this is a drug, then people may accept. But when it comes to new thing, people don't accept. And when they know, like being a medical professional who know the problems and challenges with the metallic nanoparticle, they don't accept. So transforming a metallic nanoparticle to dressing, then make it sterilize, and then make it available to the doctor, and again go and instruct to the doctor that you cannot apply it with your naked hand. You have to wear a specific gloves where transmission should not happen, and then only you can use. So it's not like an exactly antibiotic what we can do. Like if we go and take tincture, iodine tincture, and we can use it with the help of naked hand and apply over the cotton gauze and apply over the wound. It's not like that. We cannot do such things. So it's highly unsophisticated, and still it's showing good efficacy, but it's need a lot of attention for further. Like when we publish this paper, a reviewer has asked, your particles are spherical in shape, and you are claiming that this is dose dependent, biocompatible. What happens if your particles are rod like structure or octagonal structure? How it is going to behave? And then he suggested some paper, and we found that at lower concentration, when we change the shape of metallic nanoparticle, they are more toxic. Because when it comes to the peptidoglycan layer of the microorganism, those structure can penetrate very easily than the spherical shape. So this is like when we work, and then we will understand that how it is going to be challenging. If it is not challenging, again you won't feel like excitement something I am doing. So it's still a lot of challenges are there, and that needs to be understand by doing. By just reading we will know hypothetically, but when we perform, then like people will never tell you that metallic nanoparticle will deposit on your fingers if you are working with naked hand, and that is very toxic. So it has to be understand very, very carefully.
Thank you, sir. Thank you so much for answering all the queries very patiently. And we have to come to the end of this session. So on behalf of the Amity Institute of Pharmacy, Amity University, Uttar Pradesh, Lucknow campus, I would like to extend a special word of thanks to our respected speaker, Dr. Sudarshan Singh sir, for delivering such an insightful and informative lecture. Sir, your expertise on the topic biogenic metallic nanoparticles incorporated into biodegradable wound dressings have truly enriched our learning experience, especially for our young researchers and students. Your valuable knowledge, research experience, and perspectives have greatly enriched this session. We deeply appreciate your willingness to be a part of this conference and to contribute to its academic success. Thank you, sir.
Thank you very much.
Now, I would like to request our HY ma'am, Professor Dr. Anuradha Mishra ma'am, to kindly give a few words of appreciation to us, sir, in recognition to
Thank
his
you,
contribution.
Dr. Swati. Thank you, sir, for this wonderful talk about the green nano formulations, and specifically you have used herbal are the sources like whether eucalyptus or the others what you have said, and your field of expertise mainly what I found that in case of your wound healing as well as in case of your pet foods, so that is really interesting on which directions you are showing your research here. There are challenges as some of our delegates they have informed here. Definitely there is challenges when we are going for formulations. The quantity of formulations will be lower in compared to the polymers or the synthetic formulations what we are preparing using new generation polymers. So in that way again I must thank you that you are carrying out your research in these directions, and wish you all the best for your future, and you may again go for novel researches in these directions in other way. Thank you so very much, sir, for gracing this international conference and sharing your thoughts as well as your expertise in the field of nanoscience. Thank you so much. And as a gesture of appreciation, I would like to request IT team, please share the certificates. There will be a certificate which will be given to you in virtual platform. Please do accept it.
Thank you so much, sir.
Thank
It's
you,
a
sir.
wonderful lecture.
Thank you, sir.
Thank you once again, sir, for this session.
Thank you.
With this,
Thank
we
you
come
very
to the end
much.
of the technical session of the day 2. There is a lunch break now till 2 p.m. I request all the participants to rejoin sharp at 2 p.m. We will proceed with the evaluation of the scientific posters at sharp to 10 p.m. So kindly join us back at 2 p.m. Thank you everyone for joining the session. See you post lunch. Thank you.
Post-lunch session of the online conference Green NanoCon, Pioneering Sustainable Nanotechnology and Chemistry, organized by Amity Institute of Pharmacy, Amity University, Lucknow campus. This session includes the poster presentation from various researchers, and to evaluate the participant, we have an external guest with us, Dr. Rohit Bhatia. Sir, I warmly welcome you on behalf of organizing committee. I welcome you for sparing your time in joining this platform with us. Sir, I may also take liberty to introduce you with our participants. Dr. Rohit Bhatia has over 13 years of diversified experience in academics, administration, and the field of research. Sir has a specialization in medicinal chemistry, computer-aided drug designing, and bioanalysis, and his area of research interest includes anti-cancer drug discovery and paper-based microfluidics. He has been granted with 12 patents by Government of India, has published more than 150 research and review articles, more than 15 book chapters, and Sir has also authored 8 books. He has delivered various guest lectures at national and international platforms. Sir has also qualified GATE exam 4 times and has guided 32 postgraduate students and has 6 PhD students under his guidance. Recently, Rohit sir has been received a grant of 20 lakh by MSME Government of India for the development of point of care device for detection of glucose in human saliva. Sir has also been awarded with Young Scientist Award twice at international platform. With this brief introduction, sir, I again welcome you as an our external expert for
Thank you. Thank you
accepting
so
our
much, ma'am, for the
request.
wonderful introduction. And it's my privilege to act as examiner with a reputed institute like yours. So thank you so much for inviting me for this.
Thank you, sir. Thanks a lot. And the dear participants, you have 5 minutes with you to discuss your work, and which is followed by question and answer round of 2 minutes. With this, I would like to inform you that you are evaluated on 4 different parameters. The first one is presentation. The second one is scientific content. Third is your delivery and communication. And the final one is response to questions. Beside this, I also request to you to kindly switch on your camera while presentation and question and answer round. With this, may I now request the participant with code 01 to share his slide with us. Participant with code 01, are you there? I'm again saying participant with code 01, are you there? The topic of your presentation is integrated computational design and evaluation of novel imidazole analog as antiretroviral agent. Are you there? Hopefully she had not joined. So let me move on to the second participant, the one with the code 02. Participant with code 02, exploring the anti-inflammatory property of pumpkin seed. Please respond if you have joined this conference. Participant with code 03. Participant with code 03 AIP GNC 202603. Abhishek sir, can you please intervene? Do we have the participants with us?
Yes, ma'am, I am there.
Your code is
04.
04.
Yes, ma'am.
Okay, so kindly share your presentation with us.
Okay, ma'am. Ma'am, is my screen visible?
Yes, your screen is visible to us.
Okay,
Please
Yes,
ma'am.
present
visible. Please
yourself.
start.
Okay, ma'am. Can I start?
Yeah,
Yes, yes,
sure.
please,
Please.
please.
Okay. Good afternoon to respected judges and all my panel members and my fellow participants. I am Ankun Chatterjee from Birla Institute of Technology, Mishra, and today I am going to present on my topic of advances in precision medicine and AI-driven personalized diabetes care, a review. So coming to what is diabetes? Like diabetes, can we define diabetes as a common group of endocrine diseases which is characterized mainly due to increased glucose level in our bloodstream, and it is mainly caused due to basic two factors. Like one is the irrespective or inhibitory property of insulin to be produced, or another is the body cannot use the amount of insulin produced in the body in an effective way. So this leads to various types of symptoms, which in short we can classify as three P's, that is polydipsia, and that is excessive thirst, polyuria, excessive urination, and polyphagia, that is excessive hunger. So together with weight loss and blood vision, there are some of the effects which are side effects of diabetes, which also includes like diseases like kidney disease or retinal infection or neurological disease. Now coming to what are types of diabetes? Diabetes can generally be classified into two types along with other some kinds of types. Like the basic two types are type 1 diabetes mellitus and type 2 diabetes mellitus, where type 1 diabetes mellitus refers to the kind of diabetes which occurs due to the inability of pancreatic beta cells to produce insulin, and type 2 diabetes mellitus is a condition where the amount of insulin produced by the beta cells is unable to be used by the body, which causes an increase or decreased amount of blood glucose level in our body.
Okay,
Type 1
Ankan,
diabetes.
you can move to next part, stat
Okay,
and
ma'am.
precision medicine. Yeah.
Okay, sir. So,
Yeah.
according to the statistics, in my y-axis, I can show the number of people in millions worldwide who have developed diabetes over a period of from 2000 to 2030, which is a predictive model. And by 2030, we can see that the worldwide population of diabetes will reach to 643 million people, which is not very small, and it really causes a very healthy issue or a very unhealthy issue about the thinking that how it can be controlled. Now, diabetes care, like what are the basic cares that we can do for preventing diabetes? Like we can have medical nutrition and therapy, regular physical activities, smoking, reducing the amount of smoking and drinking by people, self-monitoring and education, sleep management, stress management, waste management, and etc. Now, the recent development or the recent therapy of diabetes has developed through precision medicine. So, what is precision medicine? Precision medicine is generally an innovative idea or an approach to a disease treatment and prevention that tailors the strategies to individual variability in genes, environment, and lifestyle. It mainly aims for the concept of moving away from one size, from a concept of various therapy to moving to a concept of one size that fits all the therapy. Now the process of medicine, precision medicine, can be like be continued as like first we select a disease group, then after selecting a disease group having the people with all kinds of diabetes effect, we select a control group, the people who do not have the kind of diabetes disease. Then all the data collections and data sets are collected together and they are sent to our computational system, which then sub-classifies the people according to different types of subheadings or different types of classifications. Then after classification, we take them towards characterization, which is based on mainly five factors like genomics, which means DNA analysis, epigenetics, which means gene expression and modification, transcriptomics, that means mRNA profiling, metabolomics, that is metabolic analysis, and proteomics, that is protein composition. Now, after we have classified the patients according to these five classifications, then we go for biomarker discovery. And after biomarker discovery, we go to pharmacogenetics, that can be told as precisionized drug selection. Then, after we classify the patients with precisionized drug selection, then we can proceed for personalized treatment. What happens in this cause that every people can be treated according to a different types of classification rather than an overall treatment, which helps in better understanding of the disease and better diagnosis and reducing side effects or toxicity. Now, if I come for my AI-driven precision, like what are the AI, how effect of AI for diabetes, it helps in how AI helps in diabetes. It helps in diabetes management and treatment, diagnostic and imaging techniques, health monitoring systems, developing predictive models, public health interventions like awareing public about how diabetes can cause serious health problems, prevention, lifestyle and dietary management protect for prevention of diabetes, enhancing clinical decisions and making, and patient engagement and self-management, which can help people or patients for better analysis and reducing risk factors. Now, coming to the application and devices, which by one of the MNCs, that is Abbott Healthcare, they have developed a device by which we can continuously monitor the glucose. It is called as CGM, that is continuous glucose monitoring, where we can continuously monitor the amount of insulin or the amount of glucose present in our body through our smartphone wirelessly, and by that we can adjust like increase or decreasing glucose level, and we can adjust to a uh, controllable level so that it reduces my risk of diabetes. Now, coming to conclusion, I can say that in the generation of AI, we cannot only specify to like certain old methods of following all the old traditional methods. As technology revolutioning day by day, we should also make our approach more better, like toward making it easier for our patients to have a better diabetes care and better diagnosis, thus reducing the risk of patients towards toxicity and reducing the amount of diabetes that can occur in the future days. And I would like to, in acknowledgement, I would like to thank my college, Birla Institute of Pharmaceutical Sciences and Technology, for allowing me to participate and helping me in completing my poster presentation. And this is my references. So I would like to conclude my presentation. Thank you.
Okay, thank you, Uncle. It was a nice presentation, and the title you have chosen is the need of the hour. But I would like to ask you the data you have shown. What is the reference for them?
So the reference I had
You
obtained
have
this
you
from.
have you have cited something like Abbott Freestyle Libre and Libre Assist. Is it a marketed one?
Yes,
Is
I
it
have
available in the
obtained.
market?
Yes, sir, I have obtained the data from US FDA. So it has shown,
Okay.
US FDA has shown a status of statistics of the predictive statistics of the amount of people that can be, can be diabetic towards in the next 10 years of time.
Okay, so can you comment upon the reliability of such kind of precision medicine approaches? Yeah, what is the success rate of in the current time?
So the success rate is not much high like 90% or 95%. The success rate is currently 75 to 80%, as there are still some kinds of that development needed towards my precisional medicine, as so the use of precisional medicine right now is not very much familiar in our country, but in foreign it is
Okay,
quite familiar, as people are using.
can you cite some example that those are with the at the clinical stage now and derived from precision medicine approaches? Is there any clinical status of such kind of therapeutic candidate?
So like any drug or something else you are asking?
Yeah, any approach or something drug which is in currently clinical status.
So currently the drugs that I can say of like sorry sir, like one of the drugs that is recently being used or for the type of uh, sorry, for the type of diabetes type that is being really used in precision medicine, that is the my classification which shows a rare DM body, that is mutant obesity a mutant or sorry sir mutant obesity diabetes in young patients, that is generating in patients below the age of 25 years. So in that my precision medicine is under clinical stage, that it is treating on some inhibitors like GLP-1 receptor, where the drug is like semaglutide, or on
Okay, I
SGL2
got it.
receptor,
I
that
got
is
your point. So
empagliflozin.
can you summarize some current challenges or hurdles in application of precision medicine to the market? What is the current scenario?
So the challenges is classifying people according to their disease. Like it is like all the diseases are not monitored properly or not classified. Like in our country, the pharmacovigilance is not as strong as the other countries. So the proper report generation or proper reporting of the disease, that is it really helpful, or is all the type of people are really classified in this type of drug, or my diagnosis is not correct. So that is one
Okay,
of the big
so
challenges.
if you would get a chance to work in this direction, so what will be your primary rationale?
Sir, my first primary rationale is classifying or getting all the data that will be available, correct data that will be available for all the patients worldwide, or basically for my country. Like I have to summarize all the people having different types of diabetes symptoms or like that, and like that time I can classify the data, and then based on the type of receptor on which the diabetes is acting, I can classify drugs accordingly and provide the personalized medicines.
Okay, but what's your background, Ankan?
Sir, I am currently studying masters in pharmaceutical and medicinal chemistry.
Medicinal chemistry.
MPharm in pharmaceutical medicinal chemistry.
So why you are shifting toward this precision medicine instead of synthesizing new derivatives? Is there
Sir,
any
I can
problem?
syn sir, no, sir, there is not any problem. I can synthesize medicine on diabetes also. If there is a new drug, I can go for a novel moiety that I can find out. So this is just one approach.
Okay, okay. Great, great, Ankan. Thank you. You can leave now. Thank you.
Thank you,
Next
sir.
candidate, please.
Yes, sir. Koina, are you there? Koina Chakrabarti? Koina Chakrabarti, have you joined the conference?
Yes, ma'am.
Yes, yes, Koina.
Yes, please share
Place it
your
on
presentation.
slideshow mode.
Is my screen visible?
Yeah, start please.
So, good afternoon, everyone. My name is Koina Chakraborty. I am a student of Birla Institute of Technology, Misra. So, today my topic on poster presentation is the role of COX-2 in cancer pathogenesis and its significance for drug discovery. COX-1 and COX-2 are the vital enzymes that create prostaglandins, which serve as crucial signaling molecules in the body. Nowadays, growing evidence indicates that COX-2 plays a key role in starting and advancing various cancers. Prostaglandin, when it is synthesized by COX-2, fuels tumor growth and spread by enhancing cell multiplication, tissue invasion, and metastasis. COX-2 is secreted into the tumor microenvironment by cancer cell, and it also promotes cancer development by angiogenesis, invasion, and proliferation and apoptosis. So now we will see that how the role of prostaglandin occurs in cancer. So this image illustrates the role of prostaglandin in cancer. Firstly, in this image, we can see that the cancer cells or the surrounding cells produce and release the signaling prostaglandins. Prostaglandins latch onto its dedicated receivers into the surface called EP receptors, EP1, EP2, and EP3 and EP4 receptors. And once when the EP receptors gets activated, they turn on several relay switches inside the cell like PKA, PKC, ERK, and MAPK. These switches are part of pathways that control growth and survival. These active switches travels to the cell's nucleus. They activate master switches on the DNA called NF, K-beta, and CREB. So this diagram also shows the separate pathway that links prostaglandin to growth receptors. Prostaglandin signaling can ultimately lead to activation of EGFR in promoting cell growth and survival. All these activated pathways converge on the nucleus, lead to an overall increase by the migration, invasion, immunosuppression, proliferation, and immunomodulation. So this is the COX-2 PGE2 pathway in tumor immunology. So this image shows that how the COX-2 PGE2 pathway promotes immunosuppression in the tumor microenvironment. Tumor cells upregulate COX-2, leading to the increased production of prostaglandin, which acts as a central regulator. It inhibits dendritic cell function and reduces their activity to recruit and activate effective immunoresponses, while also impairing normal natural killer cells and cytotoxic T lymphocytes, which are responsible for killing tumor cells. At the same time, it also promotes it also responsible for the expansion and activity of immunosuppression cells, including regulatory T cells, Th17 cells, myeloid-derived suppressor cells, and the M2 macrophages. It also shifts the macrophages from the anti-tumor M1 phenotype to the pro-tumor M2 phenotype. These suppressor cells produce thought factors such as TGF, beta, IL-10, and additional.
Okay, fine, Koina. Move to next part. Scroll it down. Yes. Just summarize the anti-cancer mechanisms of COX-2 inhibitors.
Okay, sir. Okay. So the this diagram of the anti-cancer mechanism of COX-2 inhibitors shows that how COX-2 inhibitor exert anti-cancer effect. Normally, arachidonic acid is converted by COX-2 into the prostaglandin, which promotes tumor growth, angiogenesis, cell proliferation, migration. When COX-2 inhibitor blocks this pathway, the conversion of arachidonic acid to PGE2 is reduced. As a result, angiogenic factors and migration of vascular endothelial cell decreases, which limits angiogenesis and tumor formation. The inhibitors also downregulates beta-1 integrin, reducing cancer cell adhesion and metastasis. At the cellular level, they trigger apoptosis by promoting cytochrome C release, PRP cleavage, and BAX upregulation and caspase activation. These effects suppress cancer cells proliferation, migration, and angiogenesis and metastasis while enhancing apoptosis. So here I conclude my presentation. COX-2 and PGE2 represent highly promising therapeutic targets. Using COX-2 inhibitors, it can suppress mainly the tumor growth and also inhibit the metastasis and apoptosis. COX-2 and PGE2 pathway is a central driver of malignancy and also promotes the pharmacological effect for inhibition and prevention of the cancer. So these are the references for my presentation. And
Okay, Koina, it was a nice presentation. And Koina, what is your postal code?
Sir, 05.
05.
Yes, sir.
Okay. You are a chemistry student or a pharmacology student?
Sir, I'm a chemistry student.
M.Pharm?
M.Pharm in chemistry, pharmaceutical chemistry.
Chemistry. So you are just focusing on COX-2, right?
Yes, sir.
So can you name any selective COX-2 inhibitor?
Yes, sir. Here I have shown that the involvement of the COX-2 in carcinogenesis. So some selective inhibitors like BCL2, TGF, beta-2, these are very helpful for inhibiting the carcinogenesis.
These are the targets, na? Can you name any marketed drug which is specifically inhibiting COX-2?
Sir, cyclophosphamide.
Which one?
Cyclophosphamide.
Cyclophosphamide.
Yes, sir.
So what's the fate of COX-1? How you will segregate? Yeah, how your drug will segregate? Yeah, what should be the specific structural features so that the drug only bind on COX-2, not on COX-1? How will you specify the specificity of the drugs?
Sir, COX-2 actually gets more attention in the cancer research, while COX-1 is.
I got all of your points. You have very well explained the role of COX-2 in carcinogenesis and in tumor progression. So, as a chemistry student, what will be your plan to inhibit COX-2 so that you can prevent carcinogenesis in future? Actually, you have not shown any evidence, yeah, any research paper work or any clinical trial drug which is already working on COX-2 and it is well established relationship. You have just explained the mechanisms. So have you shown any kind of this?
Actually, sir, I have only worked in the review part of this, but not in the research part. So that's
No,
why.
I got that. Review is okay. But you have just summarized the mechanisms. You have not shown any kind of therapeutic candidate based upon the literature. That's why I was asking. So, what is the clinical status? Is there any drug currently in clinical trials which is binding to COX-2 and treating cancer? Have you any idea about this?
So nowadays, celecoxib is recently using for the COX-2 as COX-2 in carcinogenesis.
I don't think so. Celecoxib is in clinical trials for celecoxib. Derivatives are there, but not only single celecoxib is being used against carcinogenesis. So you are in first year or second year?
So first year.
So you can design a project, you can modify celecoxib, you can use the QCR approaches to prepare celecoxib derivatives, which may be beneficial to inhibit cancer targets. So all the best for your future. Thank you so much.
Thank you, sir.
Next
Thank
candidate,
you,
please.
Koina. The next presentation is Nishant Sharma. Nishant, are you there?
Present, ma'am.
Yes, so please share your presentation with us.
Nishant, what is your postal code?
So 06.
06.
06, okay.
Yes.
Yes, start please.
Sir, is my slide visible?
Yes,
Hello.
Nishant, it's visible to us. Please proceed.
Good afternoon, sir. My name is Nishant Sharma. My presentation topic is silk fibrin dissolving microneedle for insulin delivery. Diabetes management is still largely depends on repeated subcutaneous insulin injections, but these are painful, which can cause needle phobia, which is associated with the risk of infection, poor patient compliance. Therefore, microneedle plays a very important role. It is minimally invasive, patient-friendly, and alternative for insulin administration. Microneedle represents minimally invasive transdermal delivery of insulin, as it only penetrates stratum corneum without reaching deeper layers. This enables painless delivery while maintaining therapeutic effectiveness. Among various biomaterials, silk fibrin has gained significant attention due to its biocompatibility, biodegradability, mechanical strength, and ability to sensitize biomolecules such as insulin. Additionally, secondary structure allows controlled and sustained release. The main objective of this is to develop sufficient mechanical strength for effective skin penetration, only the stratum corneal layer, to evaluate their have ability to achieve control and sustain release, to access their biocompatibility and sustainability for long-term transdermal delivery. Silk fiber was prepared and used to fabricate microneedles through molding technique. Insulin was incorporated into the polymer matrix before casting into the molds. The fabricated microneedle were evaluated for mechanical strength, morphology, insertion capacity, and in vitro release studies were performed to assess the insulin release behavior and biocompatibility. The developed silk fibrin demonstrate adequate mechanical strength to penetrate the skin effectively. They show controlled, sustained release, which is advantageous for maintaining stable blood glucose level. The dissolving nature of microneedles eliminates the sharp loss and reduces the risk of infection. Furthermore, silk fibrin stabilizing properties helps maintaining insulin bioactivity during formulation and release. Silk fiber dissolving microneedle offers promising platform for transdermal insulin delivery by combining biocompatibility, mechanical strength, and tunable release. Their ability to provide painless and controlled, potentially long-acting insulin administration positions them as a strong alternative for conventional injections for insulin delivery. Okay, sir. Thank you, sir.
Thank you, Nishant. Sir, do
Nishant,
we
you
have
are
any
a
question?
pharmaceutical student.
Yes, sir.
So why microneedles only? What are the
So,
specific advantages of microneedles over the
so
other conventional drug delivery devices?
they are minimally emissive. They do not need trained personals, can be self-administered, and they are able like they do not cause pain. They are painless.
What's about the delivery content? Can they specifically deliver the drug insulin to the administration site as the insulin traditional injections?
Yes, sir. They need to bypass stratum corneum layer, which is mainly responsible for the barrier.
for insulin.
This is your research work?
No, sir. This is a review.
So you have mentioned to develop silk fibroin based. So what are the advantages of silk fibroin?
Silk fibroin, sir, it has good biodegradability, good biocompatibility, good mechanical strength, and it is able to protect the insulin for long-term storage.
And what's
And
about the insulin release as
sir,
compared to the other delivery systems?
so we can make it controlled and sustained release for longer duration.
Controlled and sustained release. So is there any meter device available in market to
No, sir.
deliver insulin?
Sir, currently microneedle is in clinical trials only.
Have you seen the literature? Is it in clinical trial? You
Yes, sir.
haven't presented here. You must present here.
Yes, sir.
And the references format is not proper, and first reference is too much old. 2002 you have cited. Second one is not complete. So what's your plan? You are M.Pharm student?
Yes, sir.
So what's your plan in the second year about the project? You will work on microneedles only?
No, sir. I will try to find something else.
Okay, Nishant. Thank you so much. All the best.
Thank you,
Next,
Nishant.
please.
Yeah, we can move on to our next presentation. Shreyashi Goswami.
Yes, ma'am.
Yeah, please
Am I
present.
audible?
Yeah, you are audible. Please
Okay.
share your slide. Nishant, can you please unshare yourself? Nishant,
Is my screen?
no, just wait. Nishant, please unshare your presentation. Yeah, you are visible now. Shreyashi, please share your
My skin,
presentation.
my skin is visible?
Your screen is visible. We can see you, but your presentation is not visible to us. Please share your presentation.
Ma'am, just a minute. Now visible?
Yes, perfect. Please proceed.
Okay.
Shirashi,
Good afternoon.
what's your postal code?
07.
Start.
Okay. Good afternoon, sir and ma'am, and good afternoon, all the participants present over here. Myself, Sreshi Goswami. I am from Birla Institute of Technology, Misra, and I am presenting the topic on regulating of deceleration. Ma'am, is it visible?
Yes, we can see your presentation. But there is a dialog box on the right hand side. Can you
Yes, ma'am.
please?
So why it's not? Ma'am, just
Try to reshare and close everything else.
ma'am, is it visible now?
No, it's not visible. We can see you only.
There.
No issue, Shirashi. You can continue. No issue. Yes, please.
Okay, so, mm,
Yes.
so just a minute. So just a minute, just a minute. Mm. Now my topic, my topic is on regulation of decellularized extracellular matrix from pig tendon and 3D skin model recapitulate auto antibody
Shirashi,
rating
now screen is not visible. Please reshare it. Yes, it's visible.
Yes.
Continue, dear.
Okay. Now, my topic is on, as I told earlier, regulation of decellularized extracellular matrix from pig tendon and 3D skin model recapitulate autoantibody driven pemphigus pathomechanism. Now, what is decellularization? Decellularization is the process where the cell is mainly removed from the tissue, but the ECM structure of the skin is intact. Then coming to the extracellular matrix, what is this extracellular matrix? Extracellular matrix is a 3D structure, and it's mainly biomaterials like things like chitosan sodium alginate, which is mainly used for the tissue healing property. Now coming to the third one, pig tendon. Why we use this pig tendon? Because pig tendon is commonly used in this decellularization, and it's a mainly very strong collagen materials, and the extracellular matrix like pig tendon, which is also known as porcine, is has mainly biosimilarity with the human ECM. That's why we use this pig tendon. Now coming to the next one, autoantibody driven by pemphigus. Now, what is this pemphigus vulgaris? It's an autoantibody driven by blistering disorder. Then coming to the introduction, then coming to the pathophysiology of this path. Then coming to the pathophysiology of this disease, first we can see that it's a healthy human skin. When this attack, when this pemphigus vulgaris occur, the first step is occurring through autoantibody formation, which is mainly occurred by the IgG autoantibody against the desmoglein 3, which is a glue-like protein. Desmosome act like a glue. After that, binding to the desmosome, and then the loss of cell adhesion, which is also known as acanthasis. After that, intraepidermal blister formation occurs, and then the cell break, and it can occur in the mouth and causing a blister formation. After that, we coming to the methods. Firstly, we collect the pig tendon, then decellularize it, then the preparation of DECM gel, DECM hydrogel. Now, how we prepare this DECM hydrogel? That at first we collect the tissue from the pig tendon, then we decellularized it. After that we lyophilize it at minus 80 degrees centigrade to get the minus 80 degrees centigrade to 24 to 48 hours to get a powder formation. After that enzymatic digestion occurs for 72 hours approximately. As a result, we get the DCM pre-gel solution, and then neutralize it, and the final step is gelation. Then we can see that physicochemical characterization occurs through SEM, which is scanning electron microscope, through which we can see the surface morphology, and then the XRD, which is X-ray X-ray diffraction analysis, through which we can see the structure of the organism, and then FTIR biomechanical functional groups, and after that 3D bioprinting of skin construct, then induction of pemphigus, and like this way the outcome of this occur. As a result, as it's my review work and I am very new to this project, so I have not a lot of idea about this 3D bioprinting and 3D model. I just started work on this. Coming to the result, we can see that successful decellularization from the review paper. I what I read, that successful decellularization of pig tendon with preservation of extracellular matrix, exposure to the pathogenic antibodies induced pemphigus like intraepidermal blistering, and loss of keratinocyte adhesion means the dermal and epidermal junction, which is also known as retiregis. It mainly form in that part of the skin, and the engineered model closely mimic native human skin, demonstrated suitable for autoimmune pathogens. And coming to the conclusion, pig tendon DCM provides the biomimetic scaffold for 3D reconstruction. 3D skin models on DCM recapitulate key phenotypes of pemphigus and serves as effective platform studies and therapeutic testing. That's all from my side, and thank you for giving me this opportunity.
Thank you, Sreshi. It is nice presentation, and the work you have presented, it is modern one. And can you tell me, can 3D skin models represent the whole, the actual skin? Yeah, what are the limitations of such kind of models if we implement them in our studies?
So
Yeah, what is the
actually,
reliability of this?
it's a lab-based skin. It's a lab-based skin. We try to grow up, and it's a the main skin which has a structure and mechanical structure strong, but a 3D model didn't have that kind of strength and mechanical support.
Okay. And what is the
And
clinical status of these models?
sir, actually, only skin, intestine, and lungs. We see that 3D model only
Did you
performed
find
on the.
any evidence that such kind of studies are being utilized in
Sir,
modern
yes,
period?
sir. I go through with some papers, but I'm not remembering that now.
Actually, you
But
have not cited the references properly. There is no year. That's why I am asking.
oh yes, yes. Mm.
So you are what's your background? You are from pharmaceutics,
No, sir. For pharmacology masters, first year.
pharmacology. Okay.
Mm.
So
Actually, sir, I am currently going through this topic, and it's just a couple of months I am started searching on this 3D printing and 3D modeling. So I'm not a
yeah, I got that. No issue.
okay.
You will be well familiar whenever you will go deeper into it.
Yes, sir.
Um, but just tell me one thing: how 3D bioprinting is carried out? What is the fabrication part? How fabrication is done?
So through ACM, XRD, and FTIR.
That is the characterization part. No
Yes.
issue, dear. Go
So
more
through
deeper into it. It is a
cryomilling,
good
and there are
topic.
many subtypes like cryomilling part and TGA.
Okay. And can you name any kind of formulation which is available in literature based upon such kind of studies? Yeah,
So
drug delivery
ruthenium,
system. Any kind of drug delivery system.
sorry, sir. Ruthenium, is it?
No, it is a very good topic. You must explore it and you can work in your project work for this. So best
Yes.
wishes. Thank you.
Thank you, sir.
Next, please.
Thank you, Sreshi. The next participant is Ankur Day. Ankur?
Yes, ma'am. Am I visible? I'm going to share my screen now.
Yeah, Sreshi. Yes. Ankur, please share your
Your postal
screen
code
with us.
is 08.
Yes, sir. My postal
Okay.
code is 08. Okay. Okay, sir. Is my poster visible?
No, not, yeah, it's visible now.
Okay, sir. So let me introduce myself first. I'm Ankur De. I'm from Department of Pharmaceutical Science and Technology, BIT Misra. And my topic is fucoidan, a marine polysaccharide derived from brown seaweed, a biopolymer for advanced wound healing and dressing therapies. So let me first verse you what is fucoidan. Fucoidan is a marine polysaccharide derived from brown seaweed such as Fucus vesiculus and Nativata. Mainly, fucoidan is cultured from these two species. Fucoidan has been found to have wound healing and anti-inflammatory properties. Wound healing has been distinctly defined in four gradual phases, as you can see. The first is hemostasis phase, second is inflammatory phase, the third is proliferation phase, and the fourth one is remodeling phase. So Fucoidan works in primarily first three phases, while the different pathways, such as in the inflammatory phase, it upregulate antioxidant enzymes, including SOD1 and heme oxygenase, mitigating the oxidative stress, and the proliferative phase in enhanced through fibroblast proliferation and collagen synthesis with the promotion of angiogenesis. Angiogenesis is basically formation of new blood vessels, which brings proper nutrients to the injured site via the AKT, NRF2, HIF1 alpha signaling pathway. And it fucoidan also has a special property it has a role in infected antibiotic resistant bacteria like MRSA infected wound healing. I have tried to mainly focus in three novel formulations using three research articles. That is, first one is hydrogel, fucoid low molecular fucoidan, because we are using low molecular fucoidan because it has better absorption and it moves to at a slow pace. We incorporate our fucoidan into this hydrogel, it adheres on the thermal excision model, and it helps in healing. And here we can see there is a 14-day study in a dermal excision model conducted in rat. The lower one is just a control, and the upper one, the consecutive images of the overall healing property of fucoidan. Second, I have also tried to incorporate nanoparticle formulation in fucoidan with PLG, a polylactic glycolic acid. There also I have tried to incorporate some images there. The control, as we can see in the control, the wound has not healed, but in the fucoidan, the wound has started to heal. And second, there is a MRSA infected wound in rat where fucoidan has adhered to the infection trauma and it has shown considerable antibacterial wound healing properties. But I would like to state some limitations, such as it is currently in preclinical study and not evidence has been shown in clinical trials. As due to lack of evidence in clinical trials, we have not been able to translate the animal data to humans. And there's also difficulties in large scale extraction and purification. And also the brown seed cones occupy large areas, hampering shipping, fitting, and creating our in future prospects, I will like to indicate that synthesis of tailored fucoidan will result in a revolution. We can tailor-make fucoidan according to patient needs in the field natural compound due to diverse pharmaceutical applications. And use in fucoidan in MRSA-infected wounds has an amplifying effect in antibacterial wound healing property. We can also incorporate nanoparticles, nano-emulsion, electrospun nanofibers containing fucoidan, which can improve skin penetration stability and targeted delivery approach. All in all, there's a lot of potential for smartphone dressing using fucoidan, and we can by smartphone dressing. I mean it can respond to pH sensitivity according to temperature, according to the infection. So I would conclude that fucoidan accelerates healing across all wound repair phases through unique molecular mechanisms, including potent anti-inflammatory, antioxidant, pro-angiogenic, and immunomodulatory actions. Its integration in wound dressings such as hydrogels, nanofibers, films, and composite scaffolds enables rapid wound closure, enhanced fibroblast activity, robust collagen synthesis, and improved angiogenesis property. Thank you all. Thank you, sir. And here are my references. I've tried to keep it compact by using reference only of these three research paper that I have produced. Yes.
Okay, Ankur, nice presentation. Can you just scroll up to the title?
Yes, sir.
Just scroll 1 point more. Yeah. So why fucoidan?
Sir, I tried to incorporate fucoidan because it has some unique properties. Because it is
I just want to
polysaccharide.
know. I just want to know how it's better than traditional ones or other ones.
Because that fucoidan works to multiple mechanisms, not one mechanism. So that
Is it
gives you better
evidenced?
chance. Yes,
How
it is
did you
evidenced
came
in
to know? Is it evidence that it is having multiple targeting? It is a multiple targeting molecule. Do you have any reference for this?
Yes, sir. Used in preclinical testing in rat models. It focused on the three different mechanisms that I have just mentioned.
Have you done enzymatic assays for these?
No, sir.
So
it is just
this is just on the literature base you have proposed here, yeah?
Yes, yes. I've not conducted any research personally. I've just put together some points from the other research articles.
Yeah, I got it. So, what is the take home message from this work? What would you like to convey to us or the audiences about this?
Sir, I would like to convey that Foucauldian, it is a new approach. It is a less versed approach. It has not yet been thought about much, but we can put more thought into it, and we can put it is a biopolymer. It is much more biocompatible compared to other molecules. So we can incorporate
Okay,
this.
so what's about the availability of Ficodin? Is it easily available from marine sources?
No, sir. The fact is, Foucauldian has many forms. Suppose the form I'm talking about, the low molecular Foucauldian, it is rather, it is not that much cost effective to culture yet. That's the main issue.
Okay, so can we synthesize it in laboratory? What do you think?
Yes, sir. We can synthesize in laboratory, but according to pilot scale, that if we try to level up the manufacturing process
Just show
to
me the
industrial
structure
scale.
of the drug once, please. Just show me
Yes,
the
sir.
structure. Scroll up.
Yes, sir.
Yeah, we can synthesize it. Okay.
Yes, we can
So it
synthesize
is a
it.
great
But
idea.
yes, sir.
Okay, Ankur, thank you. Nice presentation. You can proceed your research work with this in future. All the best. Thank you.
Okay, sir, I'm leaving now. Thank you.
Next, please.
Yes, our next presentation is Anubhav Datta. Anubhav, are you there with us?
Yes, ma'am. Yes, ma'am.
Yeah, Anubhav, please share your presentation with us. Anubhav?
Yes.
Yeah,
And my
please,
screen.
please share your presentation with us.
Yes, ma'am.
Sir, his serial number is 10.
Sir, is my slide visible?
Yes, your slide is visible. Please put it on slideshow mode and start your presentation.
Yes, start please, beta.
So, good afternoon, sir, and to everyone present over here. I'm Anubhav Datta of BIT Mesra, and I'm currently studying in pharmaceutics. And today I'm going to present a poster on marine-derived bioactive supplements, specifically astaxanthin-based therapeutic strategies. Now, cancer is always a global health challenge with persistently high morbidity and mortality rate. And usage of marine-derived bioactive supplements such as astaxanthin has astaxanthin mainly has a lot of many mechanisms of actions to inhibit cancer cell proliferation throughout the and cancer cell metastasis throughout the body, and it has shown considerable anti-cancer potential in limited preclinical studies such as breast cancer cell lines, colorectal cancer cell lines. It inhibits the vascular endothelial growth factors. The tumor regulates the tumor microenvironment and demonstrates significant anti-angiogenic properties. Various formulations of astaxanthin like nanoemulsions, solid lipid nanoparticles, chitosan or alginate nanoparticles have been utilized till date, out of which liposomes and polymer lipid hybrid nanoparticles have been determined to be the most efficient ones. Sir, in this case, I have shown the molecular structure of astaxanthin, which is obtained from the marine microalgae Hematococcus pluvialis. Astaxanthin inhibits the expression of anti-apoptotic proteins like BCL2, BCLXL, and it upregulates the pro-apoptotic proteins
like BAX and BAD, enzymes like caspase 3 and caspase 9. It also acts via carrier-mediated pathways like in stabilizing IKB alpha protein, which inhibits the NF kappa beta protein, which in turn is responsible for the expression of IL-1 beta and TNF alpha proteins. And it also inhibits the GSK3 beta protein, which further inhibits the accumulation of beta catenin. And the accumulation of beta catenin results in the expression of MYC and cyclin D1 proteins, which are responsible for the cell proliferation, cancer cell proliferation. It also inhibits metastasis by the blocking of matrix metalloproteinases enzymes such as MMP2, 7, 9, and 10, which in turn which degrade the extracellular matrix, and the inhibition of the matrix metalloproteinase result in the extracellular matrix and the cancer cell metastasis. Astaxanthin also inhibits the HIF-1 alpha protein and vascular endothelial receptor, as I mentioned, and it's the angiogenesis process. And there are limitations of astaxanthin as it has a low aqueous solubility, due to which it is poorly bioavailable. And there are extremely limited cases of human clinical studies in cases of astaxanthin-based anti-cancer strategies. Natural and synthetic forms of astaxanthin differ structurally, affecting the biological activity. And coming to the future scopes and improvements, advanced drug delivery like utilization of nanoemulsions, which offer targeted delivery in mitochondria and maintenance of the reactive oxygen species levels for selective cancer cell apoptosis, and liposomes, which improve the stability and the therapeutic efficacy is improved via increasing cellular uptake. Combination therapies include the studies have shown that doxorubicin incorporated combination therapies result in enhanced efficacy, potentially reducing cardiotoxicity. And the production method of astaxanthin can be enhanced by genetically engineering Hematococcus pluvialis and culturing them for increased yields of astaxanthin. And to conclude my topic, I'd like to mention the fact that astaxanthin, it is a marine-derived bioactive compound having significant anti-cancer potential through its multi-targeted anti-cancer mechanisms. However, challenges related to bioavailability, limited clinical evidence, and variable responses across cancer types require continuous research. And the most important conclusion I would like to draw that human clinical trials are needed for astaxanthin related therapeutic strategies. That concludes my presentation, sir. Thank you. Thank you.
Thank you, Anubhav. Nice presentation. Just scroll it up. Yeah, minimize it a little bit. I want to see the title. Scroll it up.
So can you see?
Yes. So your title is Marine Derived Bioactive Supplements as Emerging Anti-Cancer Agents. So
Yes,
you
sir.
have basically focused on astaxanthin-based therapeutic strategies. So
Yes,
you have not cited any, you have just cited some reviews. You have not cited some latest study. So what are the main pharmacological activities of astaxanthin? Broad one. You have shown multiple, it is targeting multiple targets and like anti-proliferative, anti-metastatic, anti-angiogenic. So do you have evidences for specific ones?
Sir, tests have been carried out on cell lines such as colorectal cancer cell lines or
So that data you are having? Have you studied that data?
Sir, I have not extensively studied. I have just obtained a general idea of the required
So
effects.
astaxanthin is a, you can see that it is its structure is like
Yes,
vitamin
sir.
A,
Yes, sir.
and a lot of saturation is there. So I
Yes, sir.
think, do you studies, have you carried gone through the pharmacokinetic of this molecule? Because it is a big molecule and it is, I think it is highly lipophilic and it will definitely cause some bioavailability problems. So
Yes, sir. It is having a low water solubility, aqueous solubility, which is a problem in
definitely its structure is
distribution.
saying that.
Yes, sir.
So you must go for a suitable formulation for it, and you can
Yeah.
you can do the derivatization also. You can reduce the length of the hydrocarbon chain also. So a lot of things
Yes, sir.
can be done
Yes.
for this.
Yes, sir.
So all the best and above. This is a good uh, directive research. You can go longer towards this. So all the best. Thank
Thank you,
you.
sir.
Next, please.
Thank you, Anubhav. Our next presenter is Neha Raj. Neha, are you there with us?
Yes, ma'am. Am I audible, ma'am?
Yes, you are audible to us. Please share your presentation with us. Neha, are you sharing your presentation?
Yes, ma'am.
Okay.
Ma'am, is my presentation visible?
No, your presentation was not visible to us. Yes, it's visible now.
Okay, ma'am. Can
Please
I
put
start?
it on slide share mode and present your work.
1 minute, ma'am. Uh, can I start, ma'am?
Yes, please.
Good afternoon, respected ma'am, respected sir, and everybody present here. I am Neha Ras, and today I'm going to deliver my presentation on the topic cubosomes in nanomedicine, a step towards sustainable pharmaceutical innovation. Starting from introduction, cubosomes are distinct nanovesicles of biocontinuous cubic structure which are formulated by dispersion of liquid crystalline cubic aggregates in aqueous media, and they are characterized by high surface area and identical microstructure to its parent cubic aggregate. They are formulated by certain amphiphilic lipids as like glycerol monolith and phytantriol, which have the ability to self-assemble in water to form cubosomes. They encompass a structure similar to honeycomb structure with a size range of 500 to.
Dear, can you just zoom it in?
Yes, sir.
It
Yes,
is
sir.
not properly visible.
Sir, is it visible, sir?
Continue, please.
Okay, sir. So cubosomes are gaining a special interest as a unique drug delivery system, and recently they have been employed in ocular, dermatological, oral, and cancer therapy. Then the most important characteristic of the cubosome is their structure. So if I talk about the structure of cubosomes, so these are the cubic nanoparticles and have a honeycomb-like structure with lipid and water section arranged hierarchically in 3 dimensions. The dimension of these individual submicron nanostructure usually ranges from 100 to 500 nanometer. In lipid water system, cubosomes show up as square or round dot, which represent the presence of pores with aqueous cubic phase. Cubosomes are considered by their cubic crystalline structure and large interior surface area. Then the composition of cubosomes. Cubosomes are primarily made up of amphiphilic surfactants, polymers, and natural lipid with biocontinuous cubic phases. There are various techniques by which we can form cubosomes, and these includes bottom-up approach, top-down approach, solvent evaporation method, and spray drying method. There are benefits and drawbacks related to each of the methods. If I talk about bottom-up approach, then it requires low energy input, hence it can be used safely for drugs that are temperature sensitive. But its drawback is this method can be preferred only for thermosensitive drugs. Formulations are stable for a short period. Then top-down approach. The benefit of this approach is it reduces aggregation and improves the stability of the formulation for up to 1 year. And its drawback is it requires high energy input to disperse the aggregate into cubosomes. Then solvent evaporation method. The benefit is it provides cubosome of a smaller particle size with high physical stability. But the drawback is due to the large scale mixing of water and ethanol, it high polydispersity index is reported. If I talk about a spray drying method, its benefit is a highly versatile, cheap and scalable method and the best method for drying labile products such as protein and vaccines. But its drawbacks is it is difficult in spray drying or formulation as a cubic phase is formed immediately upon hydration of monoline. There are various applications of cubosomes, like cubosomes are used in cancer therapy because uh, tumor blood vessels are leaky and cubosomes accumulate selectively in tumor tissue. And this is called enhanced permeability and retention effect. So by using cubosomes, we can treat cancer. These cubosomes are also applicable in cosmetic industries, is also applicable in intravenous drug delivery system and also in topical drug delivery system. Because of the cubosomes cubical structure, it is very effective in topical drug delivery system. Likewise, it is also used in dermatological applications. There are various drugs which we can incorporate into cubosomes like erythromycin. If I talk about erythromycin, then it is used for topical delivery of erythromycin in the treatment of acne, and we got the result like the prepared cubosomes were effective in the topical delivery of erythromycin in a non-invasive and sustainable manner. Then simvastatin, it provide enhanced bioavailability of simvastatin to lower bad cholesterol and fat. So the prepared cubosomes enhance the bioavailability of the lipophilic simvastatin when administered orally. Then indomethacin, the objective was the evaluation of indomethacin fabricated cubosome for anti-inflammatory activity. The resulted cubosome is homogenized monoline and poloxober containing cubosome prolonged the delivery of the drug. Then decarbazine. The objective was to reduce the side effect of melanoma, and the decarbazine delivered through cubosome decreased the side effect of intravenous delivery. It also enhanced the drug efficacy, safety, and shelf life. Then timolol. The synthesis of timolol loaded cubosomes and their evolution, and the prepared cubosome showed increased corneal penetration, prolonged precorneal retention time, and enhanced intraocular pressure lowering effect than the commercially available eye drop. Now, if I conclude my presentation, then cubosome represent a highly promising and advanced nanocarrier system with a unique biocontinuous cubic structure that enables the simultaneous encapsulation of hydrophilic, lipophilic, and amphiphilic drug. Their large internal surface area, bioadhesive nature, and controlled drug release behavior and excellent biocompatibility make them highly suitable for multiple drug delivery routes, including oral, topical, parenteral, and mucosal administration. From a therapeutic perspective, Cubosomes demonstrate significant potential in cancer therapy, dermatological delivery, ocular system, and targeted drug delivery owing to their ability to enhance drug solubility, stability, bioavailability by minimizing the systemic side effects. Cubosomes align with the principle of green nanotechnology because they are composed of biodegradable, non-toxic lipid material and require relatively mild preparation methods, making them environmentally friendly and sustainable. However, challenges such as high viscosity, large scale manufacturing difficulties, stability issue, and possible drug leakage must still be addressed before their full clinical translation. Overall, Cubosome can be considered as a next generation sustainable nanomedicine platform with vast potential in modern therapeutics, and continued research and technological advancements will pave the way for their successful commercialization in the near future. So this was from my side, sir. Thank you.
Dear, go to the title.
Okay, sir.
Zoom it out. Yes. So,
Wait a minute.
why nano
Sir,
and
nano,
why cubosomes only? What are the specific features of cubosomes?
sir,
Happy
cubosomes are the first, they are 3D biocontinuous structures, sir. Their structure is the main reason behind its uses in nanomedicine, and they have very high drug loading capacity, and their structure provide large internal surface area for drug delivery. So these are
Uh, Neha, can you name any market formulation of cubosomes? मार्केट में कोई अवेलेबल है क्यूबोसोम्स की फॉर्मुलेशन, नैनो फॉर्मुलेशन?
Sir, sir, I have not read about it actually.
Just see these things, just see its clinical status. क्लिनिकल ट्रायल में भी देखो या पेटेंट्स कोई है वो सारे सर्च करो, है ना?
Okay, sir.
Okay, thank you, Neha.
Thank you, sir.
Thank you, Neha.
Thank you, ma'am.
So moving on to the next presentation. The next one is Nikhil Sharma. Nikhil, are you there with us?
Yes, ma'am.
Yeah, please share your presentation with us.
Ma'am, it's visible.
Yes, Nikhil, it's visible. Kindly proceed.
Good afternoon, sir. So I am the student of MT Institute of Pharmacy, MT University, Uttar Pradesh, Lucknow campus. Today's my topic is delivered to a poster presentation is green nanotechnology for sustainable management of dyslipidemia by gut microbiome modulation. So firstly, I can started with what is dysbiosis. Dysbiosis is abnormal level of gut microbiome. And what is dyslipidemia? Dyslipidemia is abnormal level of lipids. That is increased level of LDL, VLDL, and decreased level of high density lipoproteins. So how the dysbiosis and dyslipidemia can link with each other? So basically, there is a two types of gut microbes, that is healthy gut microbes and pathogenic gut microbes. So healthy gut microbiome consists of lactobacillus, bifidobacterium, Akkermansia mucinifera. That can lead to the SCFA production that consists of acetate, propionate, and butyrate. That leads to the lipogenesis regulation and insulin sensitivity. That leads to the bile acid metabolism into secondary bile acids and bile salt hydrolase activity and cholesterol homeostasis. Second one is a pathogenic gut microbiota. That is dysbiotic gut microbiota that consists of Enterobacteriaceae, D. sulfovibrio, Clostridium. That can lead to the inflammation, that is LPS translocation, lipopolysaccharide translocation, inflammatory biomarkers, that is TNF alpha and interleukin 6 increase, NFK beta activation, metabolic dysfunctioning. This can lead to the liver lipid synthesis, BLDL production. That can lead to the dyslipidemia. That is increase in total cholesterol, LDL, triglyceride, and HDLC. So how the gut microbiota played your regulatory role in lipid metabolism. So firstly, there are the two types of the regulators, that is positive regulator, that is beneficial bacteria, and second one is negative regulator, that is pathogenic bacteria. So beneficial bacteria can help in the microglia in the brain that leads to the satiety, fulfillment, and into the vagus nerve. They can also lead to the brain in satiety. And in liver, they can help in maintain the cholesterol metabolism, and this due to this the lipid metabolism remains balanced. And in pathogenic bacteria, in adipose tissue, they go the accumulation that leads to the lipid storage. Due to this, the lipid metabolism imbalance, and in that epithelium, they affect the tight junction function, intestinal barrier integrity due to the lipid metabolism imbalance, and that can lead to the dyslipidemia. So the interaction between the nutrition, gut microbiota, and lipid metabolism. These are the pathways. So firstly, we can dietary intake, that is polyunsaturated fatty acid, dietary fibers, carnitine. That can lead to the four pathways, that is secondary bile acids, conjugated linoleic acid, short chain fatty acids, trimethylamine oxide. So in these pathways can act on the FXR farneside X receptor in secondary bile acids that can increase the HDL level, increase the TG lipolysis, and decrease VLDL. In second, they can act on the PPR alpha, increase the PPR alpha, and increase the HDL level, increase the TG level, and lipolysis. On the other hand, TMO affect adversely that can decrease the HDL level. So second one pathway is the SCFA. SCFA consists of the acetate propionate butyrate that can lead to the absorbed by the colonocytes, that can decrease the foam cells and inflammatory cytokines and decrease the TREG cells. And on the other, there are PPR delta that decrease the lipogenesis and increase the beta oxidation. That can reduce the risk of atherosclerosis and risk of coronary artery disease. On the other hand, they decrease the HMG coenzyme reductase that decrease the cholesterol synthesis. So the next one is the TMA, that is the adversely affect pathway that can lead to the liver in the TMO increase, that can lead to the increase the scavenger A and CD36, that can increase the LDL uptake and increase the foam cells formation, that can increase the atherosclerosis and risk of coronary artery disease. So now can move on the green nanotechnology. So what is the green nanotechnology? So green nanotechnology is the formulation of the nano formulation by using the eco-friendly methods. Then this the green chemistry can combine with the nanotechnology. So in the conventional therapy, why we need the green nanotechnology? In conventional therapy, we have a systemic side effects, synthetic chemicals, environmental burden, and poor microbiome targeting. Due to this, we can need of green technology that can be targeted dribly, that in biodegradable materials use plant mediated synthesis and nano level dribly. So these are the targeted microbiome modulation that can increase the improved lipid regulation, reduced inflammation, and sustainable therapy. So various targeted therapies that can use is the gut microbiome in the dyslipidemia. So first is gut microbiota directed green nanocarriers that can made by the chitosan based polyphenol nanoparticles that target the intestinal microbiota and mucosal surface. Second one is the polyphenol loaded green nano formulation that can made of tannic acid loaded nanoparticles that target the hepatic lipid metabolism enzyme. Third one is prebiotic functionalized nanoparticles, that is inulin coated biodegradable nanoparticles that target the beneficial gut microbiota, that is lactobacillus, firmicutes. And fourth one is phytomediated metallic nanoparticles that are plant mediated silver nanoparticles that target the pathogenic gut microbes, that is Clostridium species E. coli. And last one is the pathway targeted green nanosynthesis, that is phyto nano formulation that target the various pathways, that is AMPK, actinogen, mitogen protein kinase, farneside X receptor, and PPR alpha pathways. And due to this, they can lead to the sustainability aspects. The various sustainability aspects in this is our plant mediated nanoparticle synthesis, reduced chemical waste, energy efficient production, biodegradable carriers, and lower ecological footprints. So last we can conclude with the green nanotechnology that is offers a sustainable microwave centered and macroscopic integrated strategy for the management of the dyslipidemia. They target the various pathways, that is AMPK, FXR, and PPR Due to this, they restore the lipid balance and minimize the environmental impact. And they represent the next generation therapeutic for the metabolic disease management. Thank you, sir. So this is from my side.
Thank you, Anmol.
Uh,
Nikhil. Thank you,
Nikhil,
Nikhil.
kindly zoom it out.
Okay.
Both. Uh, can you name some examples of gut microbiome approaches or modulation approaches which are already opted by the researchers?
Yes, sir. For these devices, they can operate the encapsulated probiotics. That is Lactobacillus rhamnosus and Bifidobacterium longum. And in nano encapsulated, they can also use the statins. That is atorvastatins and rosuvastatins. And for polyphenol approaches, they can use the resveratrol and curcumin.
Nikhil, what is the correlation between dyslipidemia and microbiome modulation? How these things are correlating with
So
each other?
there are two types of gut microbiome bacteria, that is healthy gut microbiota and dysbiotic gut microbiota. So this gut microbiota can lead to the inflammatory biomarker increase, can act on the inflammatory biomarkers pathway. Due to increase in this, they can activate the NFK beta that can lead to the metabolic dysfunctioning. Due to this, they can increase the LDL levels, VLDL levels, and decrease the HDL levels.
Okay, fine. So what green approaches you are adding to nanotechnology?
Yes, sir. So green approaches, that is gut microbiota target approaches, polyphenol loaded. So they can use the nano encapsulated techniques that can use the statins, rosuvastatins, atorvastatins, and also use the encapsulated probiotics. That is Lactobacillus rhamnosus, Bifidobacterium longum, and also use the polyphenol based phyto approaches, that is resveratrol and curcumin.
Good one. Okay, Nikhil, all the best. Thank you. Next, please.
Yes, sir. The next participant is Anmol Yadav. Anmol, can you please share your presentation with us?
Yes, ma'am. Is am I audible?
Yes, you are audible to us. Yes, Anmol, your slide is visible to us. Please proceed with your presentation.
Ma'am, 1 minute, sir, please.
Yes. Anmol, you can start.
Okay. Proper.
Nikhil, we can see your presentation. Anmol? Anmol, am I audible to you?
Anmol, you are mute. Unmute yourself and start.
Yes, please initiate. We can see your presentation.
Yes, sir. Can I continue?
Yes, please.
क्या हुआ? Your presentation.
Anmol, can you hear us?
Presentation वो होने लगी ना. Show तो है. Okay. Yes, sir. Hello.
Yes, please proceed. We can see your presentation also, and you are audible to us. Kindly proceed.
Okay, ma'am. Thank you, ma'am. Good afternoon, sir. My name is Anmol Yadav from M Farm Pharmacology, and today is my poster presentation on topic gene and RNA-based therapeutics for neurodegenerative disorder, a sustainable biotechnological perspective. So my topic is on the how neurodegenerative disorders and they are treated through the gene and RNA-based therapeutics. As like neurodegenerative disorders are the uh, base of that through which sorry, sir, my slide is not properly.
It was visible previously, now it has been removed. Just reshare again.
Okay, sir. Sorry, sir. It is
Yes,
visible.
continue.
Okay, sir. Okay. The neurodegenerative disorder occurs due to the degeneration of the improper DNA formation or the genetic mutations or mutations occur through the genetic level, such as the Alzheimer's disease, Parkinson's disease, and the ALS, amyotrophic lateral sclerosis. These are the some of neurodegenerative disorders, and their conventional therapies are available in the market, but they are only can treat the symptomatic treatment of basis of the diseases.
Okay, Anmol,
The
can
gene and
you just focus on the significance or advantage of CRISPR technology? Why
yes, sir.
it is why it's better than other techniques in neurodegeneration?
So because the therapies which are present in market is treating the not root cause, but the symptomatic treatment of is available. But in the CRISPR and gene and in the RNA-based therapeutics, it's basically treat the root cause of the diseases, such as the at the genetic level, through which the
Okay.
disease can
And
be
what is the adaptability of CRISPR technique? What is the current scenario?
at the
Is
current
it
scenario.
is anything marketed or is anything available in clinical trials related to this?
No, sir. There is no any adaptability, but there is preclinical trials are running through on CRISPR. But RNA therapies are already in the market, such as antisense oligonucleotides, and which the nusiran is present in the market and FDA approved.
So what's your vision about CRISPR? How do you see this technology after 10 years?
So as technologies are improving day to day, so there is a very good chance of treating diseases which are intreatable through the conventional therapies, because the at we if we treat the diseases at genetic level, so it can be improved in more and more effectively for the uh, uh, treatment of and effective towards that conventional.
So research papers are available on this or not?
Research papers are available, but not in a huge or
You have
in very
not included in this presentation, okay?
yes, sir.
What are would you like to share any kind of disadvantage of this technique, CRISPR technique?
The mainly
Limitation?
the cost effectiveness, sir. It's a very
Okay.
costly technique, and long term disease modification is very impossible to. It's like that it's so very difficult to do that. But
Okay. So Anmol, you can write a very good review on this topic, as you are saying that very less data is available. So you can add your own perspective with the help of your mentor, Dr. Anuradha Mishra ma'am. So you can formulate a good kind of review and you can publish it in good journal. Okay, thank you Anmol. Best wishes.
Thank you, sir.
Next, please.
Yes, thank you, Anmol. The next presentation is Ayushi Singh. Ayushi?
Yes, ma'am. Am I audible?
Yes, you are audible to us. Please share your presentation and kindly stick to the 5-minute time slot which is given to us.
Okay, ma'am. Okay. Ma'am, my screen is visible?
No, it's visible to
Yes,
us.
yes. Yes, start please.
Okay, ma'am. Okay, sir. Good afternoon, everyone. So, good afternoon, everyone. My topic
Yes,
today
yes,
for poster
please.
presentation is targeting the gut-brain axis for ameliorating neuroinflammation using medicinal herbs. So, as we all know that gut is the main, we can say like that gut is the main part which can affect brain as in our day-to-day life, our lifestyle is changing so much so that gut also leads to gut dysbiosis where the bacteria's gut microbiome is present. Gut microbiota gets dysbiosis and the beneficial bacteria are decreased and gut brain axis gets disturbed. So gut microbiome, which is the community of trillions of microorganisms like living in living in our digestive tract. So cognition, or we can say our neural activity also depends on that. There are several or different pathways like HPA axis and immune pathways which can be disturbed due to gut dysbiosis and lead to the neuroinflammation. So one of the major pathway is like lipopolysaccharide pathway or immune pathway, where when the like pathogenic bacteria get increases, the LPS secretion in the gut, which increases, and that leads to the disruption of gut barrier integrity, leading to the leaky gut, and then that leads to the systemic inflammation and activation of different types of cytokines and pro-inflammatory markers like interleukin 6, interleukin 1 beta, and then that leads to the disruption of blood BBB, and then that leads to the activation of glial cells in the brain, and that can lead to further neuroinflammation. So if we talk about medicinal herbs or some natural products which are actually taken in our day-to-day life, like buttermilk and curd, that helps to maintain the dysbiosis and increase in the production of beneficial bacteria, that actually leads to the production of like SCFAs, which helps in maintaining the gut barrier integrity and stability by helping in maintaining the occludins, claudins present in the gut barrier. So there are some therapeutic implications which are also used, like which are herbs like green tea, ashwagandha, ginger, turmeric, aloe vera, that are taken in our day-to-day life. And when we talk about probiotics, there are lactobacillus, bifidobacterium, and prebiotics are also being commercialized in today's world, like inulin and fructooligosaccharides, which are taken, and that helps to support the growth of beneficial gut bacteria, and that is associated in improvement of cognition and declining the neuroinflammation. And
So we can be going in this way and selecting some herbs or phytoconstituents for like improving gut dysbiosis, and then l- like protect and that can help in neuroprotection. So this is from my side, and
Okay,
thank
Aishi, thank you.
yes, sir.
I just want to ask, what is the challenges with the use of medicinal plants in therapy?
So we can use medicinal plants, and the challenges which we can face is the formulation. Like we have to prepare some like oral. If we have to prepare some oral formulation, we have to go for preparing oral formulation, or so that can be the challenge for us when we will be going for like actual research work. So
Many challenges are there. Sometimes mechanism of action is not clear. Bioavailability issues are there. Regulatory guidelines are very much limited upon these, and dose is also an issue. So,
yes, sir.
if you are going to work on natural plants or natural sources, you must work towards resolving such kind of problems.
Yes, sir. Yes, sir.
Okay. Okay, Aishi, thank you.
Thank you, sir.
All the best. Next, please.
The next participant is Sneha Pandey. Sneha, can you
Yes,
hear
ma'am.
me? Yeah, please share your presentation with us.
Okay, ma'am.
And please restrict yourself within 5 minutes.
Then ma'am, is it visible?
Yes, Sneha, it is visible.
Okay, ma'am.
Yeah, please proceed.
Yes, start please.
Good afternoon to all. Today I am Sneha Pandey, student of MPharm, presenting here. The topic is therapeutic significance of magnesium in managing polycystic ovarian syndrome. Let's start with introduction. Polycystic ovarian syndrome is one of the most common endocrine and metabolic disorder among women of reproductive age, characterized by hormonal imbalance, insulin resistance, and menstrual irregularities. Several metabolic and hormonal factors contribute to its development. Recent studies have highlighted the importance of micronutrients, particularly magnesium, in the pathophysiology of PCOS. Magnesium plays a crucial role in glucose metabolism, insulin signaling, inflammation regulation, and hormonal balance. Magnesium deficiency has been linked with insulin resistance, oxidative stress, and chronic low grade inflammation, key features of PCOS. Supplementation of magnesium may improve insulin sensitivity, restore menstrual regularity, reduce inflammatory markers, and correct metabolic imbalances. The present review aims to explore the therapeutic significance of magnesium in managing metabolic and endocrine dysfunctions associated with PCOS and its potential as a supportive treatment approach. Here I mentioned the gut dysbiosis. Gut health deeply involved in PCOS because the trillions of bacteria in intestine regulate metabolism, hormones, and inflammation. The main role of gut is digest food and produce nutrients like short chain fatty acids, which control inflammation, regulate insulin and glucose metabolism. Uh, uh, gut dysbiosis is imbalance in gut microbiota in which translocation of endotoxin LPS enters in bloodstream, and it triggered systemic inflammation, and then it causes the insulin resistance resulting in hyperinsulinemia, irregular GnRH level, hormonal level, which may causes the metabolic dysfunction, which may leads to the hyperandrogenism, and it causes the PCOS symptoms like acne, hirsutism, irregular menstrual cycle, alopecia, etc. Here the table is mentioned. Therapeutic implication of magnesium. First is insulin sensitivity. The role of magnesium enhances insulin receptor activity and glucose uptake in cells, and the clinical implication improves insulin sensitivity and reduce insulin resistance, are key
Yes,
factors
Neha,
in.
these mechanisms are well established. You have presented well, and these five are there. So,
Yes,
in
sir.
which form we can take magnesium if we are managing PCOS? What are the strategies or what are the formulation available in the market, or how you will prescribe magnesium to take by the PCOS as a people suffering from PCOS? What are the sources?
Sir,
Can they take only magnesium supplement or in adjunct to other
they
supplements?
they can include in their dietary sources like green leafy vegetables, cashews, almonds, and they can use in their daily lifestyle, sir.
Lifestyle must be changed. And if we want to take magnesium in the form of any supplement or formulation, will it be okay?
Sir, yes, sir.
Okay, Sneha. So you are B Farm student or M Farm student?
Sir, M Pharm student.
Department?
Sir, pharmacology.
Pharmacology. So you have shown in the table there are five mechanisms
Yes, sir.
through which magnesium plays a pivotal role. So do you have studied the research paper on this?
Yes,
Because
sir.
in the references you have mentioned there is only one cohort study that was conducted in 2019. So you must go through more literature, and this can be a good review too. If you are going to write some kind of review, this will be a good one.
Okay, sir. Thank you for your guidance, sir.
Okay, dear. All the best. Next, please.
Thank you, Sneha. The next participant is Apurva Shukla. Apurva, can you hear me? Apurva Shukla, can you hear me? Are you there with us? Sir, shall we move to the next one? Maybe she is not connected.
Yes, yes.
Okay, so the next one is Devendra Pratap Singh. Devendra,
Yes, Devendra, you can start.
Devendra, please present yourself and be strict to 5 minutes. Devendra, you are not audible to us.
Now, am I audible, ma'am?
Yes, yes,
Yeah.
yes. Please proceed.
Yeah. First of all, a very good afternoon, ma'am and sir. My name is Devin Pratap Singh, a student of MPharm Pharmacology first year. Today, my presentation topic is biomimetic novel metal nanozymes next generation catalytic therapies for the cardiovascular diseases or the CVD. Now, talking about the introduction session, that the cardiovascular diseases are one of the considered to be one of the prominent cause of the mortality or the death across the worldwide, and the cardiovascular diseases consist such as the myocardial infarction, atherosclerosis, and the heart failure. The oxidative stress is considered to be one of the major cause of the cardiovascular diseases, and which is characterized by the over or the excess production of the ROS, that is the reactive oxygen species, and the various example of the ROS are the superoxide anions, H2O2, that is the hydrogen peroxide, and the hydroxyl radicals. And when there is a dysregulation or the imbalance between the body ability to neutralize these ROS and the body mechanism to neutralize these ROS and the level of the ROS, This may further leads to the endothelial dysfunctioning, inflammation, lipid oxidation, and the apoptosis of the cardiomyocytes or the death of the cardiomyocytes. That is, the cardiomyocytes are the cells of the heart. Now, the traditional therapies which are currently used in the treatment of the cardiovascular diseases having the very less bioavailability, and they are not targeted, they not means they are not produce the targeted delivery system. That's why uh, we move to the nanomedicines which having the potential of the targeted drug delivery system. Now, the noble metal nanozymes. Nanozymes are the man-made enzymes like nanoparticles that can speed up the chemical reaction just like real enzymes such as catalase and SODs. SODs are sodium superoxide dismutases. And the noble metals which are used in the formulation of the nanozymes are the gold, platinum, palladium, silver, ruthenium, iridium. Are these are the noble metals which are used for the formulation of the nanozymes, and these noble metals act as a primary catalytic core of these formulations. And now the why nanozymes, because why novel metals are used for the formulation of the nanozyme. The reason is they having high surface energy and uncoordinated surface atoms. This is the main reason why we use novel metals for the formulation of the nanozymes. Now, the role of ROS in the myocardial infarction. Myocardial infarction is basically characterized by the blockage of oxygen or the blood supply to the particular region of the heart due to the blockage of the coronary arteries, which the causes of the myocardial infractions are the ischemia, hypoxia, or the excessive production of the ROS. One of the major reason of the myocardial infarction is the activation of the release of the cytokines such as TNF alphas, which further leads to the inflammation or the oxidative stress. And once the TNF alpha or the cytokines should be released, they will further activate the inflammatory pathway, that is NFKB and MAPK pathways, which further leads to the cardiomyocyte apoptosis as well.
Devendra, can you have you studied some case studies on this? Yeah, do you have some evidences related to activity performed by?
Still, it was in the developing phase. That means the noble metal nanozymes are still in the developing phase.
Okay.
One major, that is gold-based AU nanozymes or the AU-based nanozymes are still in the
Is
trial,
anything
in the
available
clinical
in
trial.
market currently or not?
No, there is no product is available. They are in clinical trial phases, and all gold-based nanozymes are in the
क्लिनिकल ट्रायल में है अभी?
सर गोल्ड बेस्ड नैनोजाइम्स हैं, वो अभी फेज 2 के ट्रायल्स में अभी भी वर्क हो रहे हैं उन पे, स्टिल
Okay, so this
वर्किंग।
is a good field to explore further, huh?
यस सर।
So conclude it.
या so basically they talking about the traditional therapies which are used in the treatment of the cardiovascular disease. They are not they are not provided targeted delivery of the drug, and they having a very less viability, and which was further overcome by these nanozymes or the nanometal based nanozymes, which having the good viability and produce and gives the targeted drug delivery system as well. So that's all
Okay,
from.
this is a good topic, and you can plan your research as well as a review towards this direction. Devendra, you are from M Pharmaceutics?
Yes, I'm from pharmacology, sir.
Pharmacology. Okay, great.
Thank you, sir.
Thank you, Devendra. All the best. Next,
Thank you.
please.
So the next participant lined up is Gargi Mehdi. Gargi, are you there with us? Gargi, can you hear me?
Yes, ma'am. Yes.
Yeah, please share your presentation. Yes, your slides is visible to us. Kindly stick to the 5-minute timings.
Good afternoon, everyone. I'm Gargi Medhi from first year MPharm Pharmacology department. Today I'm going to present on the topic interplay between the engineered nanoparticles and gut microbiota in colorectal cancer. In recent studies, colorectal cancer has been termed to be the third most common cancer worldwide according to WHO, and the mortality rate is increasing, and it is shown that colorectal cancer is the major cause of mortality leading due to cancer. There is evidences highlighting that gut microbiota plays an important role in CRC. It helps in the progression, initiation, and therapeutic responses of the CRC. Colorectal cancer is basically a malignant tumor that begins to occur in the lining of the large intestine, which occurs due to the mutations in the genes, basically the APC gene and the TP53 genes. Gut microbiota plays a dual role. It helps in the maintaining intestinal homeostasis, but in case of dysbiosis, which is the imbalance in the microbial composition, it promotes inflammation, genotoxicity, and the tumor growth. There are various steps, that is initiation, then which leads to propagation of the CRC. The initiation phase begins, which is irreversible damage at the genetic level, which involves formation of DNA adducts, and this formation is due to the carcinogens that bind covalently to the DNA, thus causing genotoxicity and thus in progression of the tumor growth. So, nanoparticles have come into effect because of their physicochemical properties, which helps in treatment or therapeutic responses towards CRC, that is, because of their small size and high surface area. Because of the small size, they penetrate to the biomembrane very easily because of the leaky passages in the tumor cells. And also, because of the large surface area to volume ratio, it interacts with the proteins and DNA easily for the treatment. The engineered nanoparticles, such as commonly are the titanium dioxide, the silver, and the zinc oxide, which are ingested easily from the food, they can be used in targeted delivery. And the targeted delivery that can include and that is marketed are 5-fluorouracil. 5-fluorouracil is given as nanopolymer with chitosan nanoparticle, and it can be used in the treatment because it has targeted directly to the tumor cells rather than giving 5-fluorouracil solely, because it will target not only the tumor cells but also the normal cells. There are specific bacteria such as bactericides, Fucobacterium, and also E. coli, which can help in tumor growth. E. coli produces colibactin, which is a genotoxin, and it causes DNA damage. So basically, the gut microbiota promotes CRC by DNA damage and also inflammation and metabolic dysfunction. Nanoparticles and the gut microbiota can come into interplay in CRC because the engineered nanoparticles exhibit intrinsic
Gargi,
antimicrobial properties.
your topic is very good. Can you tell me some combinations which are already available in market with nano formulation and some kind of gut microbiota?
So nanoformulations that are marketed are
Nano formulation with these probiotics or prebiotics.
Sir, chitosan based 5-fluorouracil, it is a nanoparticle which is available because 5-fluorouracil was the first line drug which was used in CRC.
Is there any prebiotic or probiotic loaded with it?
No, sir. Because clinical trials are still going on and research articles are still being...
How many clinical trials are going on? Have you studied this?
Yes, sir. In animals,
because this approach is very much promising nowadays, and so many people are working on it.
Yes, sir.
So I think success rate will be soon. Soon there will be a drug in the market containing both of the things.
Yes, sir.
So you can also design your work, but you are a pharmacology student. If you want to prepare some kind of formulation, you can go toward this direction too.
Yes, sir. Yes.
So can you tell some challenges toward this direction? दोनों को combine करने में कोई challenges हो सकता है?
Sir, challenges uh, there are challenges in preparation of the nanoparticles because
नहीं, nanoparticle तो बन जाएंगे easily.
हाँ सर.
But gut microbiota और nanoparticles को stable रखने के लिए there is some problem. इनका stability issue generally होता है, so you can work toward that direction also.
Okay, sir.
References you have included. Can you name some gut microbiota? कुछ माइक्रोऑर्गेनिज्म के नाम बता सकते हो?
Sir, healthy gut microbiotas are lactobacillus, the common one, and
Okay.
the other gut microbiotas are Bacteroidetes fragilis, then Fucobacterium nucleatum.
Because this field is very highly explored in case of ulcerative colitis, which ultimately leads to colorectal cancer. So this is a good approach, and you must work towards this direction. Go and search more and more literature and plan something new for your further research. Okay, all the best, dear.
Thank you, sir.
Next, please.
Sasank Bajpayee, you are with us? Sasank?
Yes, sir.
Am I audible, Sasank?
Yes, sir. Yes, sir. Sir,
Yes, please.
Hello.
Yes, please share your presentation.
Okay, sir. Okay, sir.
1 minute, Sasank. Hello?
Yes, sir.
Sasank, am I audible? Please
Yes,
share
sir.
the
Yes, sir.
presentation.
Okay, sir. Okay, sir. Sir, give me 1 minute.
Yes, please. Shashank, start. Shashank, start your presentation.
Sasank, please start your presentation.
Sir, can we take next one? Meanwhile, he is getting stable.
Yes, sir. Yes, sir. Uh, Manorama Patel.
पता है
Manorama Patel. You are with us, Manorama Patel. Manorama, am I audible?
It's Sasank.
Please start your presentation.
Yes, Shashank, please continue.
Okay, okay. Sorry, sir. It's a mistake from my side.
Yes, please continue.
Sir, good evening. My name is Shashank Bajpai and I am from MIT Institute of Pharmacy. Today I am presenting my poster on the topic neuroprotection targeting apoptosis in Parkinson disease. Sir, firstly, I want to brief about the what is the Parkinson disease. Parkinson disease is a progressive neurodegenerative disorder which mainly characterized by degeneration of dopaminergic neurons in the brain. Sir, the current treatment of the Parkinson disease is totally depend on the sympathomimetic treatment, which is unable to cure the root cause of the Parkinson. And therefore, my work is focused on the targeting apoptosis, a key mechanism responsible for the neuroprotection care. Sir, firstly, I wanna again discuss about what is the apoptosis. Apoptosis is the programmed cell death, which follow by the which follow up by the all cell and neuron for the degradation. About to 10 million cells and neurons follow this pathway to over their whole life. Sir, mainly the two pathways are in the apoptosis. First one is first one is intrinsic pathway. Second one is extrinsic pathway. In the intrinsic pathway, here mainly caspase 9 works about it, and that's why it is totally cured by sir, intrinsic pathway is also known as a mitochondrial pathway, where the caspase 9 is works to cause the death of neuronal from brain. And sir, targeting apoptosis is one of the most cared because of mitochondrial environment activate apoptosis caspase and alpha cyclonuclein, which is a protein in brain that regulate the neuronal changes in the brain that is also maybe treatable about this. Hello?
Yes.
Sir, audio आ रही है ना?
Yes. You are audible. Please continue.
Okay, sir. Okay, sir. Sir, again, the symptomatic treatment and the present with what I am working, that is a huge difference between it. So the main difference between that, the symptomatic treatment is unable to cure the root cause of this, and it is short acting. But at apoptosis, when we able to cure it, then it will be the long-term activation, long-term activity provided in the disease of Parkinson, and it will be.
Shashank, I got the message from your this poster. I just want to know from your side that what kind of therapeutic agents can be used to protect apoptosis in
Sir,
Parkinson disease, or
yes,
you have
sir.
already summarized all the things, all the phenomena taking place in this Parkinson disease. So what kind of,
Yes, yes.
how will you, if you are a medicinal chemist or if you are a scientist, then how will you design your drug?
Sir, firstly, I
Okay, what is the
study
current
about the
scenario towards
pathway.
this direction?
The current scenario is that the antioxidant
Okay.
like coenzyme Q10 and sir, a totally gene-based therapy, tolinirib. This drug is mainly under the phase 2 clinical trial, but is not giving more activity and advancement about the Parkinson disease. So, sir, at present time, I can say that there is no present drug is available for the therapy of Parkinson disease.
ठीक है शशांक, ये जो आपकी पार्किंसन डिजीज है, इसकी पैथोलॉजी है बहुत कॉम्प्लेक्स है, इसमें बहुत ज्यादा टारगेट इन्वॉल्व होते हैं। तो क्या कोई ऐसा मार्केट ड्रग बता सकते हो
Yes, sir.
जो अवेलेबल है ऑलरेडी, जो एक से ज्यादा टारगेट पे काम करती हो?
सर ऐसा ड्रग अभी तो सर यही एंटीऑक्सीडेंट जो हमारा कोएंजाइम Q10 बना हुआ और टोलिनिबी ड्रग चल रही है जो सर क्लिनिकल ट्रायल में है, अभी नहीं वो आ रहा है मार्केट में। दैट सर ओनली आई
तो
नो।
अगर तुम फ्यूचर में इस पे इस डायरेक्शन में काम करना चाहोगे तो तुम्हारा क्या प्लान रहेगा? हाउ विल यू सॉल्व दिस प्रॉब्लम?
Sir, by the use of BCL2 family proteins and P53, I will I मारा
अगर
maintain
तुम्हें
those.
कोई ऐसा कंपाउंड ढूंढना पड़ेगा जो मल्टीपल टारगेट पे काम करता हो, एक से ज्यादा टारगेट पे काम करे,
Yes, sir, yes, sir.
ठीक है, तो इस पे और लिटरेचर देखो एंड और स्टडी करो कैसे इसको किया जा सकता है, ठीक है?
Okay. Thank you, sir.
ओके शशांक, थैंक यू, ऑल द बेस्ट। नेक्स्ट प्लीज।
Okay, Shashank, you are leave now. Next is the Monorama portal. You may leave now, Sasank.
Good evening, sir.
Yes, yes. Present your slide.
Shashank, you remove your slide that's so that next can be taken in. Yes.
Sir, it is visible to you?
Yes, yes, please start.
Okay, sir. Good evening, sir. I am Manorma Patel and I am from MIT University, Lucknow. And today I am presenting my poster on sustainable nanocarriers for treating postmenopausal metabolic disorders and bone disorders. As postmenopause is a natural stage in woman's life that occurs due to decrease in estrogen level hormones. This hormone imbalance can lead to metabolic disorders such as obesity, and insulin resistance, and cardiovascular risk, as well as bone disorders like osteoporosis. Conventional treatments such as hormone replacement therapy may cause side effects and long-term risk. Therefore, there is a need for safer and more targeted treatment approaches. Sustainable nanocarriers are advanced drug delivery systems that can improve drug sustainability, enhance bioavailability, and provide controlled release. While reducing side effects, these systems also focuses on eco-friendly and biocompatible material, making them safer for long use. Uh, sir, can I explain my poster?
Yes, yes, carry on, carry on.
Okay. First of all, here I want to inform you about what is nanocarrier. A nanocarrier is a teeny drug which size is approx 1 to 1000 nanometer, which transport medicine.
You just go to the importance of nanocarriers in menopause.
What, sir?
Just go to the section applications of nanocarriers in postmenopausal disorders.
Okay, okay, sir. Sir, it improves metabolic disorder, hormone delivery, calcium, vitamin deficiency, and reduce drug toxicity, and it provides sustained drug release.
So what kind of specific nanocarrier you must use, or is there any nanocarrier available in the market to treat such disorders, postmenopausal metabolic disorders? क्या मार्केट में कोई है ऑलरेडी या क्लिनिकल ट्रायल में कोई है?
यस सर, सर अभी मार्केट में एक ड्रग है ऐसी जो सर हार्मोन थेरेपी होती है, मतलब उसमें जो पेशेंट होती है उनको ये काफी लॉन्ग टर्म है, बट यस सर, बट मतलब अभी उतना मतलब एडवांस नहीं है कि मतलब ये पूरा डेवलप नहीं हो पाया है, बट या सर इस पे काम चल रहा है, ट्रायल हो रहा है. हेलो.
अभी ये मतलब प्लान स्टेज पे ही है आपका, या आपने कुछ लिटरेचर इस पे देखा है, या स्टडीज ऑलरेडी चल रही हैं?
यस सर, जैसे कि लाइपोसोम्स वगैरह हैं जो यूज करते हैं नैनो कैरियर की तरह.
मार्केट में है कोई बोन डिसऑर्डर्स के लिए या पोस्ट मेनोपॉजल के लिए?
यस सर, इस्ट्राडायल समथिंग, इस्ट्राडायल है शायद. सर पोस्ट मेनोपॉज के लिए या फिर ऑस्टियोपोरोसिस के लिए?
जो भी, कोई भी, दोनों में से कोई भी डिसऑर्डर हो.
ओके सर, बेसिकली जो इसमें होता है पोस्ट मेनोपॉज में होता है कि जो एस्ट्रोजन का लेवल होता है वो डिक्रीज हो जाता है, तो उसके लिए.
हाँ, दैट दैट आपने वो बता दिया. क्या मार्केट में इसके लिए है कोई अवेलेबल ड्रग
यस सर,
जो
मैं
नैनो
उसी से आ रही.
फॉर्मुलेशन में हो, नैनो फॉर्म में हो?
यस सर, उसके लिए सर इस्ट्राडायल यूज होता है, और जो ओस्टियोपोरोसिस है उसके लिए एल्डोड्रोनेट यूज होता है.
नैनो फॉर्म में है वो?
यस सर.
ओके. So what's your plan? Yeah, what is the take home message to the audience from your this poster? What do you want to convey?
Sir, my message is to convey that as women is already having so many disorder and disease in their whole life, सर मेरा जो मोस्ट पर्पस है इस टॉपिक का कि नैनो कैरियर का जो डेवलपमेंट है इस फील्ड में भी होना चाहिए.
Okay. So you can design a project towards this direction and definitely you can work towards this.
यस सर, क्योंकि इसमें सर ऑस्टियोपोरोसिस और मेटाबॉलिक डिसऑर्डर पे रिव्यू पेपर तो अवेलेबल है, बट पर्टिकुलरली इस पे मतलब पोस्ट मेनोपॉजल और ऑस्टियोपोरोसिस रिलेटेड टू दैट काफी कम है और इस पे उतनी स्टडीज नहीं है.
Okay, okay. So you can design. All the best. Thank
Thank you,
you.
sir.
Next, please.
Manorama, you may leave now. Vaibhav Singh, you are with us. Okay, present your slide.
Yes, sir.
Vaibhav, share your slide.
Yes, sir. So just a second. Sir, the screen is visible now. Hello.
Not visible.
Not visible, sir. Right. Okay, sir. Yes. Is it visible now?
Yes, yes.
Yes, sir. So, sir, good afternoon, sir. My name is Vaibhav Singh, and I am going to present my topic, that is nanotechnology-based therapeutic strategy for hepatotoxicity. So, sir, first of all, we start with
Vaibhav, make the slide share mode. Slide share mode.
okay, sir. Sir, is it fine?
Okay,
Hello?
okay, present.
Yes, sir. So the first of all, we start with my introduction of my topic, that is hepatotoxicity. Hepatotoxicity means the damage to the liver caused by the chemicals, drugs, toxins, or infections. Drugs like PCM, alcohol abuse, viral infection like hepatitis virus. This can lead to the hepatotoxicity. So we are going to for the nanotechnology. Nanotechnology helps in hepatotoxicity by protecting liver cells, delivering drug directly to the liver and reducing the side effects. And so why nanotechnology is more conventional by comparing the other technology overcomes
वैभव,
poor targeting
तुमने
of
ना इसमें
conventional
एक
therapies.
टेबल टेबल बनाया है, वो टेबल एक्सप्लेन करो बस. बाकी तो टेक्निक मैंने देख लिया है.
Yes,
इस
sir.
टेबल में क्या दिखा रहे हो?
Sir, these are therapies which are currently running into the market, but only patisiran is only a drug which is approved by which is only approved in the market, and by and other
Okay.
drugs are and other therapies are under the clinical trial.
अगर हम पैरासीटामोल के नैनो फॉर्मुलेशन बना दें तो फिर
Yes,
हिपेटोटॉक्सिटी
sir.
नहीं करेगी? अगर पैरासीटामोल के नैनो फॉर्मुलेशन बना दें तो हिपेटोटॉक्सिसिटी नहीं होगी?
सर वो डिपेंड करता है सर, आप किस अमाउंट में और कितने टाइम तक आप पैरासीटामोल का यूज कर रहे हैं सर.
नहीं तो पैरासीटामोल, सपोज किसी को 5 दिन बुखार आ रहा है लगातार,
Sir, more than 4 gram of paracetamol may cause the hepatotoxicity. So, sir,
नहीं नहीं वो तो मैं समझ रहा हूँ,
nanotechnology, sir.
बट क्या कोई ऐसा
Yes, sir.
एविडेंस है तुमने कोई स्टडी करी है जिसमें ऐसा कुछ दिया हो? जैसे तुमने ये टेबल में दिखाया है, ये आपने कहाँ से उठाया डाटा जो टेबल में है?
सर ये जो करंटली पेपर पब्लिश हो रखे हैं सर उसके अंदर से।
वो है ना, ये तो इसका मतलब हो रहा है वो। तो
यस सर।
एक तो एमआरएनए के लिए है, एसआईआरएनए के लिए है। कोई मुझे ड्रग का नहीं दिख रहा इसमें। कोई ड्रग का एग्जांपल है जिसमें ड्रग की नैनो फॉर्मुलेशन बनाई हो और हिपेटोटॉक्सिसिटी प्रिवेंट की हो?
सर अभी तक तो नहीं हुआ सर।
अच्छा अच्छा। तो तुम भी बना सकते हो। अगर इस तरह का कोई डिजाइन है जो हिपेटोटॉक्सिसिटी ड्रग करती है, उसकी नैनो बना के आप उसको बाईपास कर सकते हो।
यस सर, इज इट पॉसिबल? बट सर स्टडी इसकी अभी चल रही
फ्यूचर
सर, तो
पर्सपेक्टिव में क्या
रिसेंटली।
दिखा रहे हो, बोलना जरा।
यस सर।
व्हाट इज द फ्यूचर पर्सपेक्टिव?
सर फ्यूचर पर्सपेक्टिव में सर जैसे कि थर्मोनाइस्टिक नैनो पार्टिकल्स दे रखा है सर यहाँ पे, तो सर इसके अंदर क्या होता है, जो हम नैनो पार्टिकल यूज करते हैं बॉडी के अंदर, पहले ये तो जाएगा लीवर के पास, फिर वहां पे वो चेक करेगा कि कौन से लीवर साइट्स हमारे डैमेज हो रखे हैं। देन आफ्टर दैट सर वो उसकी एक इमेज क्रिएट करके देगा, फिर हम लोग वहाँ पे जो अपना ड्रग है वो इंट्रोड्यूस कराएंगे, तो थेरापेटिक रिस्पॉन्स वहाँ पे प्रोड्यूस करेगा।
तो क्या
और सर
सारी की
ऐसे
सारी ड्रग्स के लिए पॉसिबल है ये?
नो सर, ओनली फॉर स्पेसिफिक ड्रग्स। तो सर
हाँ,
अभी
वही
उसके
मैं
ऊपर
एग्जांपल
रिसर्च
पूछ रहा
बेस्ड
था, किस
वर्क चल
टाइप की
रहा है।
ड्रग्स, कौन सी ड्रग, कोई ड्रग का एग्जांपल है तो वो बताओ।
सर सॉरी सर, अभी मुझे नहीं पता है।
चलो इसका लिटरेचर देखना, और इस डायरेक्शन में आप काम भी कर सकते हो। ये एक अच्छी अप्रोच है,
ओके
ठीक है। ओके,
थैंक
थैंक
यू
यू।
सर।
नेक्स्ट प्लीज।
शिवम कुमार। शिवम। Shivam, am I audible?
Yes, sir. Yes, sir.
You
I
present your
share my
slide.
screen. Are you showing my PPT?
यस यस।
Okay. Good afternoon, everyone. Everyone, my name is Shivam Kumar. My topic is green synthesized small nanotechnology studies for Alzheimer disease, innovative, eco-friendly approaches to treatment challenges. So, firstly, start with introduction. Alzheimer disease, as you know, that a most common progressive neurodegenerative disease is measure 55 million people affected by this.
ठीक है बेटा, ये इंट्रोडक्शन मेरे को पता है। आप ग्रीन सिंथेसिस पे आओ जरा। ग्रीन
Okay, sir.
सिंथेसिस में कैसे करोगे?
This is the green synthesis where different types of nanoparticles are available in this, which is definitely target the pathological hello markers like amyloid beta plaque and tau protein hypophosphorylation,
नहीं,
neuroinflammation
इसको
and
आप ग्रीन क्यों कह रहे हो? ग्रीन सिंथेसिस का क्या इन्वॉल्व कर रहे हो आप? ग्रीन सिंथेसिस
it
कैसे
is
है ये?
because it is synthesized from a green plants like flavonoids flavonoids, epigenin, curcumin, which is effective for the treatment of Alzheimer disease.
तो उसका एविडेंस है आपके पास कि फ्लेवोनॉइड्स आर यूज्ड इन द थेरेपी ऑफ एल्जाइमर डिजीज?
Yes, sir. Like epigenin, novel synthetic flavonoids,
वो
catecholins.
चीजें तुमने
These are
इंक्लूड नहीं करी हैं पोस्टर में कहीं भी।
yes, sir.
और ग्रीन सिंथेसिस के लिए क्या क्या अप्रोच होती है? एक
Uh,
तो बता दिया कि ग्रीन आप नेचुरल प्रोडक्ट से ले लोगे।
yes, sir.
इसके अलावा
To,
ग्रीन सिंथेसिस की क्या क्या अप्रोचेज होती हैं?
uh, it's like metalline silver, and
बेटा बड़ी सारी टेक्निक्स होती हैं, जैसे आप माइक्रोवेव असिस्टेड सिंथेसिस कर सकते हो, अल्ट्रासोनिफिकेशन से कर सकते हो, ग्रीन सॉल्वेंट यूज करके
yes
कर
sir.
सकते हो। बड़े सारी टेक्निक्स हैं, उससे आप ये नैनो फॉर्मुलेशन बना सकते हो जो कि अल्जाइमर डिजीज को ट्रीट करने में आगे यूजफुल होगी। तो इसमें और लिटरेचर देखो जरा, इस पे काम करो, टेक्नोलॉजीज देखो कौन कौन सी ऑलरेडी ये है और कौन सी ऑलरेडी एस्टेब्लिश्ड है, वो और थोड़ा लिटरेचर देखो, ठीक है।
Okay sir.
नेक्स्ट प्लीज।
Shivam, you may leave now. Akanksha, am I audible? Akanksha,
Yes, sir.
share your slide.
Good afternoon, sir. It is visible.
यस यस प्लीज कैरी ऑन।
So my name is Akansha. Today I am here to present my presentation on the topic of advanced nanotechnology-based target therapies for psoriasis, progress from preclinical studies to clinical development. Let's start from introductions. Psoriasis is a chronic autoimmune disease which is characterized by red scaly patches on the skins. It affected on 64.6 billion people at this time. People with psoriasis may also suffer from diabetic heart problem, depression, and anxiety. Let's go on the pathophysiology. The disease begins with activation of dendritic cells in the skin. These release interleukins 26, which stimulate TS17 cells. Then is produced T inflammatory cytokines such as interleukins 23 and interleukin 17. Keratinocytes, hyperproliferation, and abnormal differentiation, formation of red scaly patches, itchy patches on the skins. Let's go on the rationale for nanotechnology in the psoriasis management. It is used for enhanced skin penetration and retentions, provide controls and sustain drug release, reduce systematic side effect, improve drug stability and bioavailability, enable target delivery in inflamed skin cells, enhance therapeutic efficacy with lower dose. Let's go on this schematic representation of nanocarriers mediated therapy in psoriatic skins. These are the nanotechnology for treatment for psoriasis skin. It is treat low side effects and very low times. Types of nanocarriers and outcomes. There are 4 types of nanocarriers: liposome, solid lipid nanoparticles, NLC, and microemulsions. That's conclusion. Advanced nanotechnology-based target therapies offer a promising strategy for improving the management of psoriasis by enhancing the drug penetration, controlled release, and site-specific action while minimizing these systematic side effects.
ठीक है बेटा आकांक्षा, आप इसको एडवांस
you,
क्यों
sir.
बोल रहे हो? एक नैनो टेक्नोलॉजी है सिंपल, एक एडवांस नैनो टेक्नोलॉजी है, दोनों में डिफरेंस क्या है?
Sir, can you speak?
हेलो
Sir, can you repeat?
मैं ये पूछ रहा हूँ बेटा, व्हाई यू हैव यूज द वर्ड एडवांस नैनो टेक्नोलॉजी? एक सिंपल नैनो टेक्नोलॉजी है, एक एडवांस। एडवांस में क्या क्या आप इनकॉर्पोरेट कर रहे हो? आपने इसमें दिखाया लाइपोसोम्स और ये सब, ये तो ट्रेडिशनल ही है, तो एडवांस क्या हुआ इसमें?
सर टू ड्रग्स को मैच करके भी मैंने यूज किया, जैसे सर रेस्वेट्रॉल और एपिजिनिन को यूज करके ट्रांसडर्मल पैच बनाएंगे और उसको ट्रीट करेंगे। दैट्स
मतलब 2 ड्रग्स को एक में इनकॉर्पोरेट करके उसकी नैनो फॉर्मुलेशन बनाएंगे, ऐसा?
यस सर।
अच्छा,
सर एज लाइक
क्यों नैनो
ट्रांसडर्मल
क्यों बनाना
पैच।
चाह रहे हैं आप? नैनो का क्या एडवांटेज है?
सर बिकॉज enhance skin penetration retention, provide controlled and sustained drug release, reduce systematic and side effects.
तो सोरियासिस तो एक टॉप अपनी लोकल डिजीज ही है, जिसको आप टॉपिकली अप्लाई करोगे तो उसमें तो टॉक्सिसिटी वाला ज्यादा कोई चक्कर रहेगा नहीं वैसे।
यस सर, बट सिस्टमेटिक भी अप्लाई कर सकते हैं।
सिस्टमेटिकली ठीक है बेटा, इस पे थोड़ा और डाटा देखो। प्री क्लिनिकल का तुमने इंक्लूड नहीं किया इसमें, प्री क्लिनिकल इंक्लूड करो और स्टडीज और करो, है ना?
Okay.
ओके, नेक्स्ट प्लीज।
Stepna,
Yes, sir.
just share your slide.
Yes, sir. Sir, is it visible?
यस,
Hello?
विजिबल, काइंडली स्टार्ट।
Yes, sir. A very good morning to one and all, a very good evening to one and all present over here. My name is Stefna Verghese, and the topic that I'm going to present on is green synthesized nanoparticles for targeted cancer therapy, pharmacological mechanisms, and clinical potential. So first of all, we need to understand why do we need such a type of therapy? Because cancer treatment continues to be a cause of concern due to its systemic toxicity, and the major disadvantage with the current traditional cancer treatments is that along with the cancer cells, it also destroys the healthy cells. So that is a big disadvantage that we are facing right now. So to tackle with that, scientists have been working on eco-friendly nanoparticles to enhance the therapeutic effect by regulating tumor cell pathways, which enables the destruction of cancer cells without harming healthy cells, leading to reduced side effects and overcoming
So
drug
what is
toxicity.
the green thing in this?
Sir, the green thing here is that we use metallic nanoparticles. So, reduction of that metallic nanoparticle is done using plant sources like leaves, flowers, seeds, and even microbial culture. So, first of all, we make the extract out of leaves or flowers of a particular plant. And in those extracts, we add metallic salts like
Okay, you are
silver
adding
nitrate.
metal ions as catalyst. Okay, it's fine. And you are
Yes,
using
sir.
flavonoids, polyphenol, terpenoids as a drug. Okay.
Uh,
So why
yes,
not? Suppose if I am going for preparation of synthetic drug and I
yes, sir.
am preparing nanoparticles of a synthetic drug, and
Mhm.
you are saying that we are preparing flavonoids
Mhm.
nanoparticles. So व्हाट्स अबाउट द एक्टिविटी? फ्लेवेनॉइड ज्यादा एक्टिव होंगे या जो हम ड्रग बनाएंगे वो, उसको कैसे इंश्योर
सर
करोगे आप?
सर एक्चुअली फ्लैवेनॉइड का रोल यहाँ एज अ रिड्यूसिंग एजेंट एंड अ कैपिंग एजेंट की तरह है, वो एज अ ड्रग की तरह नहीं काम कर रहा है, हम ड्रग को
नहीं,
इनफूट
आपने
कोट
लिखा है ना
कर।
फॉर टारगेटेड कैंसर थेरेपी,
यस सर।
तो वो
यस सर।
कैंसर सेल को कैसे टारगेट करेगा फिर?
Sir, actually, the main thing is that tumor tissues are have a very weak and leaky vasculature. So, because of that, its increased porosity and the small size of the green nanoparticle, it is able to penetrate easily and accumulate inside the
नहीं नहीं,
uh,
दैट आई
cancer cell. Yes,
अंडरस्टैंड,
sir.
बट व्हाट इज द थेरेपेटिक एजेंट? ड्रग कौन सी है इसमें?
Sir, सर ड्रग तो अभी मतलब सर ये वाला जो मेथड है इस पे अभी काम चल रहा है, तो अभी तो
कोई ऐसा लोगों ने बनाया है ऑलरेडी, लिटरेचर में है ऐसा कुछ?
नो सर। सर अभी नैनो पार्टिकल्स जो बने हैं वो गोल्ड के हैं, सिल्वर के हैं एंड जिंक ऑक्साइड के हैं, बट अभी प्रॉपर कोई मार्केटेड ड्रग नहीं बनी है इसके ऊपर, अभी इस पे काम चल रहा
कोई
है। इट्स
क्लिनिकल ट्रायल
बेसिकली
में कुछ हो?
सर प्री क्लिनिकल ट्रायल्स में अभी है।
क्योंकि जो फ्लेवेनॉइड और पॉलीफिनोल आप बोल रहे हो और ट्रेपिनॉइड्स, इनका स्ट्रक्चर बहुत बड़ा है। ये आपस में
यस
कहीं ना
सर।
कहीं आपकी थेरापेटिक ड्रग से ना रैक करके कुछ इंटरफेरेंस कर सकते हैं, तो इस डायरेक्शन में कुछ देखना, कोई इंटरफेरेंस ना हो।
यस सर।
बाकी फ्लेवेनॉइड खुद एंटीऑक्सीडेंट है, तो वो कहीं ना कहीं उसमें हेल्प करेंगे एंटी इंफ्लेमेटरी में, बट दोनों के बीच की कंपैटिबिलिटी का इशू हो सकता है, वो आप चेक करना एक बार, ठीक है?
यस सर।
ओके, थैंक यू स्टेफना।
ओके सर, थैंक यू।
नेक्स्ट प्लीज।
Saurian,
Yes, sir, I am here. At first, good afternoon to everyone. I am Soren Gupta from Infam Pharmaceutics. My topic is in situ hydrogels of acetylcholinesterase inhibitors for nose to brain delivery and approach for Alzheimer's disease treatment. So, Alzheimer's disease is a progressive neurodegenerative disorder which is characterized by cognitive decline and cholinergic dysfunction. Acetylcholinesterase inhibitors such as donepezil and rivastigmine remain the first
बेटा
line
थोड़ा इसको
symptomatic
जूम करोगे आप?
Okay, sir.
बस कंटिन्यू नाउ.
Okay, sir. The acetylcholinesterase inhibitors such as donepezil and rivastigmine remain the first-line symptomatic treatments. However, their clinical efficacy is limited by poor blood-brain barrier penetration and exceptional fast-pass metabolism and systemic side effects. Intranasal nose-to-brain delivery has emerged as a promising non-invasive strategy to bypass the BBB via olfactory and trigeminal pathway. For that, we have prepared in situ hydrogels. In situ hydrogels are basically a polymeric formulation that exists as a liquid or salt form before administration and undergo gelation at the site of application due to the physical stimulus such as temperature or pH or can be ionic strength. For intranasal delivery, this formulation is administered as low viscosity solution into the nasal cavity where it converts into
Sorry,
a
just explain the route. How
Hmm.
nose to brain delivery is, what is the pathway from nose to brain delivery? How it is happening? Just focus on this.
Okay, sir. Actually, sir, there is mainly two pathway from nose to brain delivery. One is olfactory pathway and one is trigeminal pathway. In olfactory pathway, in olfactory pathway is sensory neurons in the nasal roof directly connected to the olfactory bulb through the cerebriform palate foramina. Drugs diffuse across the olfactory epithelium into perineural space surrounding these axons. And rapid transport of frontal cortex regions and the time is 15 to 30 minutes it takes, and this pathway achieved 8 to 12% direct olfactory deposition with the highest drug concentration. And other pathway is trigeminal pathway, uh, which is which facilitates slower cellular transport to the brainstem, cerebellum, and hinder brain. And this takes time 30 to 60 minutes. And trigeminal route predominates from nanocarriers like NLC, and 20-fold higher concentration is in trigeminal pathway is seen than the olfactory pathway.
Is there any market formulation available already in the market?
Sir, there is some acetylcholinesterase inhibitors of rivastigmine and donepezil are there, which is marketed in the name of Ixoron in tablet or capsule form. And there is also one nano formulation. I think it is transdermal patch, liposomal transdermal patch. These are already available, and the tablet and capsules are available in 1.5 mg, 3 mg, and 6 mg dose. And
Okay,
the
just scroll down.
okay, sir.
You have shown some preparation methods, physiological parameters. Scroll down. So
Sir.
what is the message you want to convey to the audience?
Sir, in this time, it is one of the hot topic for the nose to brain delivery because there is some limitation in brain delivery because of
But you
the
are saying that already a lot of things are available in the market. So
no, sir.
what new
Actually,
you have planned?
sir, actually conventional dosage form are available like tablet and capsule, but there is no nano formulation, and
Okay.
one nano formulation is marketed, but that is not so much effective.
Okay. So what point you are adding to make is effective?
Sir, if we want to make it effective, then we have to easily cross the blood-brain barrier, or we have to increase the retention time of the drug or targeting.
Okay. Okay, it is a good plan, Sorin, and you can seek towards this direction in future too. It will be good work.
Obviously, sir.
Okay, thank you. All the
Thank
best.
you.
Next, please.
मुझे पता था.
Aman Yadav,
Yes, sir.
share your slide.
Yes, sir. Sir, is it my poster is visible?
Yes, visible, beta. Start.
Yes, sir.
Yes, start, bit.
First of all, good afternoon, one and all present here. My name is Aman,
and I'm here to present my poster on the given topic, Stimuli Responsive Drug Delivery System for Site-Specific Drug Release. As we know, the Stimuli Responsive Drug Delivery is the one of the smart advanced drug delivery system that release drug only at the specific site of action when triggered by the certain stimuli. Unlike the conventional drug delivery, where the drug is spread throughout the body may cause a side effect to the neighboring cells, this system releases the drug in controlled and targeted manner. There are many triggers a stimuli can be. pH changes, such in the case of tumor where the pH slightly change, temperature, yes, of course, the second example, enzyme, redox conditions, light, magnetic field, and ultrasound. What is the need of the site specific? It targets only disease tissues like tumors, reduce the damage to the healthy cells, provide controlled and the sustained release, useful in chronic cancers like diabetes, arthritis, and cancer. Types of stimuli, internal stimuli, sir, pH, enzyme, redox conditions, and temperature variations. Uh, Types of stimuli responsive nanocarriers. First of all, pH.
So,
Yes, sir.
how these stimuli can be produced if you are going to prepare dendrimers? So, what will be the stimuli in this case?
Yes, sir.
How to trigger the action of the drug in case of stimuli? If you are using some dendrimer, what will be the stimuli?
Sir, in the case of
अगर आप डेंड्रीमर यूज कर रहे हो तो उस केस में डेंड्रीमर को एक्टिवेट करने के लिए या उसके लिए स्टिमुलाई क्या हो सकता है? आइदर इट इज पीएच और डॉक्स, एनी काइंड ऑफ रिएक्शन जो बॉडी में होगा वही है.
सर
मेरा क्वेश्चन समझ रहे हो? मैं कह रहा हूं अगर आपने कोई भी ऐसा नैनो कैरियर बनाया है जो आपकी बॉडी में जाएगा तो उसको आपको पीएच सेंसिटिव बनाना पड़ेगा या पीएच रिस्पॉन्सिव बनाना पड़ेगा
सर पीएच रिस्पॉन्सिव
या कुछ थर्मल टाइप के होंगे या कुछ अलग टाइप के होंगे?
सर पीएच सेंसिटिव बनाना होगा
सभी को?
नो.
ये तो मैंने अलग अलग टाइप के दिए हैं इसमें, पीएच सेंसिटिव, थर्मो सेंसिटिव, पॉलीमेरिक,
हाँ
डेंड्रीमर्स.
हाँ सर.
So how dendrimers will be activated? चलो नो इश्यू. मार्केट में अवेलेबल है कुछ इसका?
हाँ सर, थर्मोडॉक्स है, टेम्परेचर सेंसिटिव लिपोसोम,
10 मिनट.
जिसमें डॉक्सीरुबिन को इंकॉर्पोरेट करते हैं. दूसरे में सर डॉक्सिल है या कैलिक्स है, पिगाइलेटेड लिपोसोमल डॉक्सीरुबिसिन है. तीसरा है सर मैरिक्यूबो, लिपोसोमल विल केस्टिंग फॉर्मुलेशन.
ओके, तो कंक्लूड करो जरा. कंक्लूजन में क्या लिखा है आपने? मल्टी यूजिंग जीन थेरेपी, इम्यूनोथेरेपी, एप्लीकेशन आर वाइड. सो क्या मैसेज आप कन्वे करना चाहोगे ऑडियंस को?
Sir, extremely responsive drug delivery is the one of the promising field where we can smartly treat the disease with the help of extremely responsive, and it have a lot of work going on. That's all.
Okay, dear. Thank you. Next,
Thank you, sir.
please.
Aman, you may leave now. Okay.
Okay, sir.
Soham?
Hello, sir. Am I audible?
Yes,
Yes, yes, please.
Okay, sir. Sir, is my screen visible?
Yes.
Okay, sir. Thank you. Good afternoon, everyone. My name is Soham Das, and I'm a student of MIT Institute of Pharmacy. Today, my topic is green synthesis of nanocarriers and approach for targeting glioblastoma. Glioblastoma is the most aggressive malignant brain tumor in adults and is associated with rapid proliferation, diffuse infiltration, frequent recurrence, and very poor survival despite surgery, radiotherapy, and chemotherapy.
So, Home, just tell me which green approach you are going to prepare for nanocarriers. Just scroll down.
Yes, sir. Sir,
Just
here
scroll
are,
down.
yes, sir. Sir, here are several
What
types
is the
of
greener part in your approach?
So greener part of my approach is that in my green nanoparticle system, the parts of various plants, microorganisms are used for the synthesis of nanoparticles, which
So
targeted.
how they will focus to the glioblastoma only?
Sir, there is a mechanism of action how they are targeting glioblastoma. Here we can see the nanoparticles enters into the cells where on the left side different types of nanoparticles are shown. These nanoparticles enter the cancer cells through
I
cellular.
got it. I got this figure also.
Yeah.
But I am asking how you are making your nanoparticle target specific, that they will target only glioblastoma, not others. How?
By targeting to the brain delivery system.
How you will target to brain? What
Uh,
thing are you adding?
So by targeting from nose to the brain delivery system, where we can focus on the olfactory trigeminal pathway, and we can also modify the surface that can helpful the nanoparticles from treatment of glioblastoma, for the treatment of glioblastoma.
And what you are presenting in table number 4? What is these are the reported ones?
Yes, sir. These are the reported ones that which plant parts are used and which metals are used for the targeting glioblastoma.
Okay. So all the shapes are spherical. So I
Yes,
think
sir.
it will also,
Sir,
the shape is also useful for it.
Yes, the shapes are also useful for it. Some all are not spherical. Some are cubic. Some are triangular, and some are rod also. Rod
What
shaped
are the
also.
challenges?
So challenges are that it is a plant-derived metal nanoparticle synthesized by green methods, but incorporation metals raises concerns regarding potential toxicity. But metallic properties may lead to bioaccumulation and adverse biological interaction. These are the limitations and challenges there.
So what is your plan? Will you work in this direction or something else?
Sir, I am working kind of same direction, but not this type of research. My target
Okay,
is
so
also
you are not following the green synthesis?
sir, yeah, not likely green synthesis, but nanoparticles.
Okay. Okay, dear, all the best.
Thank you, sir.
Try to apply greener things also in your synthesis.
Okay, sir. Thank you. I will mind it. Thank you, sir.
Next one.
Vedita, am I audible?
Nivedita. Yes, Nivedita, start. Nivedita, start your presentation.
Vedita, am I audible? Please start your presentation. Nivedita, sir, I think technical glitch maybe.
So we can take the next one, sir.
Next one,
Meanwhile.
yes. Meanwhile, good time. So, Akshat,
Yes, sir. So, I am audible?
yes.
Okay.
Nivedita, you just now remove your slides.
Yes, sir. Yeah. Someone already shared.
Yes, share now, Akshat. Yes, please, Akshat, start your presentation.
Yes, my name is Akshay Gupta, and my topic is cardiovascular implication of hypothyroidism and hypothyroidism. First, start with introduction. Thyroid hormone T3 and T4 regulate the cardiovascular system. T3 is active form and directly act on heart muscle cells. Any imbalance can disturb normal cardiac functions. Then mechanism of action: T3 enter cardiomyocyte through a transporter, bind to thyroid receptor in nucleus, then regulate gene for beta adrenergic receptor. This increases the contractility and heart performance. It also has a rapid non-genomic
अच्छा
effect.
अक्षत, मैं सीधे सीधे तुमसे क्वेश्चन पूछूं? ब्लड प्रेशर अगर हाइपरथायराइडिज्म है तो ब्लड प्रेशर पे क्या इफेक्ट होगा?
सर इंक्रीज होगा.
इंक्रीज होगा. और हाइपोथायराइडिज्म में?
सर डिक्रीज होगा.
ऐसा क्यों?
सर हाइपरडायनामिक सर्कुलेशन की वजह से.
कार्डिक आउटपुट पे क्या इफेक्ट रहता है?
हार्ट रेट इंक्रीज होता है सर. हार्ट
रेट इंक्रीज होगा कब?
सर तो हाइपरथायराइडिज्म होगा.
हाइपरथायराइडिज्म में. अच्छा, अब हाइपरथायराइडिज्म के रीजंस क्या हैं, क्यों होता है ये?
सर थायराइड ग्लैंड प्रोड्यूसिंग एक्सेसिव हार्मोन्स.
हाँ, वो तो ठीक है. कोई इसका ट्रिगर होता है बॉडी में? कुछ अल्कोहल या कोई किसी डिजीज या समथिंग एल्स कुछ? अच्छा, व्हाट इज आर्टेरियल फाइब्रिलेशन?
टाइप ऑफ हार्ट अरिथमिया.
कैसे, क्या इसमें स्पेसिफिक क्या है आर्टेरियल फाइब्रिलेशन में?
इंक्रीज हार्ट रेट, रैपिड हार्ट रेट.
तो आप क्या मैसेज देना चाहते हो कि लोगों को अपना थायराइड लेवल नॉर्मल रखना चाहिए?
यस
अगर कम होगा तो भी कॉम्प्लिकेशन होंगी और ज्यादा होगा तो भी कॉम्प्लिकेशन होंगी. बट तुमने एक चीज नहीं बताई इसमें कि नॉर्मल रखने के लिए क्या क्या चीजें हम करनी चाहिए. बता सकते कुछ? हाउ कैन वी मेंटेन अवर थायराइड लेवल्स हेल्दी?
सर सर अगर मेंटेन करना है तो दवा दवा ले सकते हैं.
कौन सी?
सर थायरोडमाइड और बीटा ब्लॉकर्स. और सब डाइट भी होते हैं. सी
बीटा
फूड.
ब्लॉकर तो फिर हार्ट रेट के लिए हो गया ना.
यस सर.
ये पढ़ो बेटा, थोड़ा लिटरेचर है ना.
यस सर.
क्योंकि अभी आपने ओवरव्यू दिया है, ये लगभग सबको पता है. आप उसको कैसे कंट्रोल कर सकते हो, उसकी थेरेपीज करंटली क्या चल रही हैं, थेरेपी नहीं लेनी है तो उसके नॉन थेरापेटिक्स मेजर्स क्या हैं, ये सारी स्टडी करो. ठीक है बेटा?
यस सर.
ओके, ऑल द बेस्ट.
थैंक
नेक्स्ट
यू सर.
प्लीज.
Nivedita, am I audible? Okay. So Aditya Ras,
Yes, sir.
you share your slide.
Yes, sir.
यस आदित्य, स्टार्ट.
Yes, sir. Sir, good evening, sir. Myself, Aditya. Today, I'll be presenting on my topic, which is basically Just a second. Which is basically tau and oxidation begins in Alzheimer's disease. Basically, the Alzheimer's disease is a heterogeneous multifactorial disorder characterized by accumulation of amyloid plaques in the
बेटा इसको स्क्रॉल डाउन करो नीचे.
Yes, sir. So,
करो करो और करो. और करो स्क्रॉल. पाथवेज के अलावा आपने और क्या इसमें आप दिखा रहे हो? पाथवे
sir,
एवरीबॉडी
we
नो
are
की
basically
ये सारे होते हैं, इन्वॉल्व होते हैं उसमें. आपके टाइटल में है द प्रॉमिस ऑफ मल्टी टारगेट डायरेक्टली लाइगेंड्स इन एल्जीमर थेरेपी.
yes, sir.
हाँ, वो उसके बारे में बताओ जरा.
Basically,
मल्टी टारगेट आप कैसे करोगे?
basically we get drugs are available. They are basically of symptomatic treatment. So what we can do is that if we can make drugs bind on all three enzymes, basically the alpha secretase, beta secretase, and gamma secretase. Basically, artificially engineer monoclonal antibodies, which is given to the patient that goes and binds to the
Aditya, you are a
microglia.
pharmacology student or pharmacology student you are?
Yes, sir. I'm a pharmacology student.
Pharmacology?
Yes, sir.
So can you name any drug which is used in the treatment of Alzheimer disease and which is targeting more than one target?
For example, monoclonal for lecanemab, donanemab,
मार्केट में कोई ऐसी ड्रग है जो एक से ज्यादा टारगेट पे बाइंड करके अल्जीमर को ठीक करती है?
yes, sir. Yes, sir.
तो एग्जांपल बता सकते हो?
Lecanemab is the name. Hello.
जाना तू? जाना?
हम तो चले गए नहीं फिर नीचे. Excuse me, sir. Excuse me, sir.
Yes,
Sorry,
yes.
sir. Sir, today till this other next participant will continue for tomorrow.
Okay, sir.
So, dear delegates, this presentation will continue tomorrow. Today till this. So, thank you very much, sir. I would like to sincerely appreciate the valuable contribution as a evaluator in our international conference and your time, expertise, and constructive feedback. I justly hearing you given the nice feedback, and although they are the MPharm students, but yet to they start their projects, but your feedback definitely help them
Yeah,
to
yeah,
both
definitely, sir. Students
condition.
are very good.
Yes,
They
sir.
have put their best concepts, and I ensure that they will work in good direction in the second year also. And thank you
Yes.
so much for providing me the opportunity to evaluate the
Sir,
students.
sir, one token of appreciation in recognition of the your effort. Just one, I'll request to the IT team to arrange a small that appreciation, that is the certificates. So I request to IT team, please display the certificate for Dr. Rohit sir. Abhishek sir.
Yes, sir. Yes, sir. Yes, sir. I am sharing, sir.
Thank you so much for the honor, sir.
And thank you very much, sir.
Thank you,
Thank
sir.
you so
I
much, sir.
hope for future collaborations with you, Thank you, Abhishek sir, for providing this opportunity. And definitely we will
Thank you
collaborate in terms of research and all the things, sir.
thank you so much, sir, for giving us your valuable time. I know, sir, you are very busy. Thank you so much, sir.
Okay, sir. Thank you.
Thank
All
you.
the best to all the students. Do well and get your good publications. Thank you all.
Thank you, sir.
Thank you, sir. Thank you.
Thank you, sir.